Guys, remember that. Good morning, good afternoon, good evening. Because since it's a cross night, that's why I said good morning. Because since it's a cross night, that's why I'm saying good evening, eh? Because now we are in evening, but we're gonna go up to morning. However, we shall be having some breaks so that you refresh your mind, and then we see that we get our distinctions. The reason why we are here is to see that everyone is able to score high. I pray that you guys, what you wrote, the examiners are not that much difficult on you. They mark you nicely and then you pass. There is no need for you to repeat. There is no need. There is no need. Ne? Yeah. So guys, if things don't go according to the plan, ne? Don't lose hope. That's not the end of the world. Ne? You rebuild it, and then you go up to, there is a child I taught she did her first metric in 2000, I think 17. She came back 2018. She just got the course she wanted last year, and she's there in University of Cape Town. She said, if I don't get University of Cape Town, I'll repeat until I get it. So she repeated until she got it. I'm telling you. I'm telling you. She was very determined. Eh? Yes. Yeah. She got it. She's there in University of what? Yeah. So it means that if things don't go right, they all don't go according to the plan. Stay focused. Ne? Things will come right at one time. Yeah. However, you must always have with the plan B. Ne? If you see that you don't have that patience to keep on repeating the same thing, have plan B. Still, when you're going to Joburg, you can pass in a different route or another route. It can be longer, but still you reach your what? Yes. So, and lastly, you always know me. I say this and I'll repeat it. Not the best in the class is the best in the world. Yeah. So make sure that if you know something, don't become selfish. You won't be the best in the world. Never. So make sure that you help others so that they also what? Succeed. Thank you so much. Let's start. We know that the first topic is DNA code of life, isn't it? DNA. You see now. DNA. I want to see if you see. If you don't see, I increase the thickness. Should I increase the thickness? Okay. DNA. Can you see now? All right. Thank you so much. DNA code of life. When you say DNA code of life, guys, there are some things which you need to talk about. So we're going to try to do different questions. Our focus. But I'm going to brainstorm you. And then I see. I have my specific time here on this chair. There will be another teacher who will come still on this chair, and another teacher who will come on this chair, meaning that you are 3 teachers who are supposed to take you over this cross night. Are we there? Yes. Describe the structure of DNA. Who can describe for me? Is a double 6 marks. 6 marks. Describe the structure of DNA. 6 marks.
So we think it's 6 points.
Yeah.
So the first one is it's double-stranded.
It's double-stranded. It's saying double-stranded.
Double helix.
Double helix.
Twisted.
Huh?
Coiled.
It's coiled.
What mark I say goes not to a large.
I told you. You know, remember now I'm reminding you, ne? Huh? Onele. Where's Onele? Yeah, you want to say something? Eh? You want to say something? Always to talk. Hey. No. Okay. DNA double helix. When they ask you, describe the structure of DNA, you draw the structure of DNA. You look at it and you describe what you see. Isn't it? For example, remember, DNA is made up of what? Is made up of what? A nucleotide. Is a mark. Are we there? A nucleotide is made up of what? Phosphate. Describe the structure of DNA. And then deoxyribose sugar. Ne? Deoxyribose sugar. Don't just say sugar. Sugar what? Deoxyribose or ribose is made up of a phosphate deoxyribose sugar, and nitrogenous bases. You tell us which nitrogenous bases are those. Is thymine, uracil, isn't it? Oh, I think you are not in class. Yeah, it's thymine, adenine, and guanine. Oh, and cytosine, isn't it? That's a mark whereby adenine goes with thymine, and then guanine goes with cytosine. This is also another mark. Is one mark, ne? You mentioning the mark. You mentioning the bases is a mark. You mentioning how do they combine is another what? Another mark. Is it clear? Then it has two strands, whereby these strands, one strand is joined with another strand by what? By weak. Say guys, weak hydrogen bonds. When you say hydrogen bonds, we won't give a mark. Guys, please don't do this. Don't say hydrogen bonds. It's the same thing which I said yesterday, that don't say perpendicular. And then someone was asking the same question when you're busy talking about the memorandum. I use perpendicular. This is not maths. It is what? It's life science. So weak hydrogen bonds. People who are doing physical science, don't say hydrogen bonds. Is it clear? Yes. It forms two strands, whereby these strands are joined together by weak hydrogen bonds. Is it clear? It twists around to form a double helix. Do you see? Don't talk about coiling. Please. You want to get a mark for coiling? Ne? Yeah. It twists around to form a double helix. What is the natural shape of DNA? What's the natural shape of DNA? We only have one answer. Double helix. Nothing else. The natural shape of DNA is what? Double helix. Is it clear? Yeah, I don't want to waste too much time. We're going to go to the question so that we smash them. Ne? Who discovered the structure of DNA? Who discovered the structure of DNA? Who discovered the structure of DNA? Watson and Crick. Watson and Crick. We have Watson, Francis. No. And then you have Crick, Rosalind. No. Is who?
It's James Watson.
James Watson and Francis Crick. Don't say Watson and Crick. We have so many Cricks. We have so many what? Watsons. You understand? You say James Watson. Is it clear? Next question. Who discovered the structure of DNA using X-ray diffraction pictures? People online who are on live, you tell us the answer. Yeah, double helix. Yes. Watson. Chief, make sure that you say James Watson. You see, even people online, they are saying Watson. Ne? James Watson. Is it clear? Who discovered the structure of DNA using the X-ray diffraction pictures? Nakato, I don't hear you. Rosalind, Franklin, and Morris Wilkins. Are you there? Who said, which scientist concluded that DNA is double helix with complementary base pairs? Who concluded that DNA is double helix with complementary base pairs? Morris. Is it Morris? You don't know. You don't know. Now you have to know today. Yeah, it's James Watson, and it's James Watson who concluded that DNA is double helix with complementary base pairs. Is it clear? Another question. When was the Nobel Prize given? Yes. 1968. 1962. Yeah, you are here to remember. Ne? Yeah. If you don't know something, guys, make sure that what? You note it down. Ne? Yes. We got the next question. Should we go? Right. Now, If you have DNA, DNA is made up of 1000 bases. Yeah, 1000 bases. And then 100 is guanine. What is the ratio of cytosine to thymine? Did you hear me? If you have DNA, calculate. I want the answer from your books. Yeah. I want to see. I want to see. I want to see. I want to see. DNA is made up of how many bases? 1000. Only 100 is what? Is guanine. What is the ratio of guanine to cytosine? The ratio of cytosine to thymine. Nana, do you get it? Do you know how to get it? Okay, let's find it out. Ne? I hope you are done with it. Let's calculate it together. We are saying that, remember, you need to complement them together, isn't it? You have guanine goes with cytosine, isn't it? If guanine is 100, cytosine is what? Is 100. So I have 100 plus 100, which gives me 200, isn't it? So it means I'm going to say, because the total number of bases in DNA is 1000, so I'm going to say 1000 minus what? Minus 200, which gives me 800. Wabona? So 800 will be for cytosine and it will be what? It will be thymine. It will be thymine and adenine, isn't it? So if I divide by 2, I'll get 400 here, 400 here, isn't it? So now what I'm going to do, I'm going to say guanine, they want cytosine, cytosine to thymine, whereby cytosine is what? We said the cytosine is 100, and then thymine is what? 400. So divide by 100, divide by 100. Wabona? And then now I get the answer to be is 1 to 4. So do you know what's going to happen if they bring such questions? The first objective, they will say A, 4 to 1. B, 100 to 400. I see. Then they say C, 1 to 4. I see. Then they say D, 2 to 8. Because you calculated and you found 1 to 4. The first objective is 4 to 1. You think that's the answer. You get it wrong. So please make sure that you do not rush. You do not what? You do not rush. We continue next question. Yeah, someone was saying 1 to 1, you see. Yeah, thank you so much, guys. I think now you can get it. All right, when we continue, guys, let's continue, guys. Another question. Give me two DNA, two locations where you can find DNA in animal cell. Mitochondria and the nucleus. Name two types of DNA in the animal cell. Mitochondrial DNA and the nucleatic. Nuclear DNA. Do you see how the question can be treated? Mitochondrial DNA and nuclear DNA. Is it clear? All right. We go to the next question. Sorry, guys. I'm not a meme. Okay, we continue. Someone is calling me. Hello, ma'am. All right. Yeah, we continue, guys. Here is a simple diagram of DNA, isn't it? What is A? What is one? What is one? Guys, talk. What is one? Phosphate. What is two? Deoxyribose sugar. What is three? Weak hydrogen bond. Three. Those lines in between. Eh? Three is this thing. These lines. Weak hydrogen bonds. What is G? 1,9. And then what is that? X. Cytosine. What is F? What is F, guys? Is here. What is F? Cytosine. Because already we have seen it here. So when they bring, you know what happens? They won't ask you what is this. They won't ask you what is this. They'll ask you what is this. So that we know you have to use this knowledge that this one goes with this. You understand? Yes. How many nucleotides does this DNA has? How many nucleotides? 1, 2, 3, 4, 5, 6, 7, 8. Isn't it? Yeah. So DNA, guys, is not that a big deal. It's not that a what? A big deal. I'm gonna fire you a question now. What is DNA replication? What is DNA replication? Process whereby DNA makes exact copy of what? Of itself. When does it occur? In the nucleus. Where? I said, when does it occur in the nucleus? When does it occur? During the interphase. I see. When? Where does it occur? In the nucleus. Why does it occur? To double the chromosome? Yeah, you are still on track. You are still on track. Shh. How does it occur? You describe. Who describe? How does DNA replication occurs? Need someone? Samantha, are you okay? Are you okay? Yeah. Sharon, describe for me. Yeah. Describe the process of DNA replication.
The DNA double helix unwinds.
And
the weak hydrogen bonds break as it unzips.
Weak hydrogen bonds break as it unzips, okay?
Then what happens as
You asking me?
And then two templates are formed. Two
templates are formed. Let me add you before you go so that we correct. Two separate. The word separate is the marking point. If you don't say two separate, you are gone.
Two separate templates are formed. And then free DNA nucleotides from the nucleus.
Wait. It's better to say two separate strands are formed. You understand? They might give you a mark there. You saying separate. Then you say each acting as a template. You get another mark. Wawana. Two separate strands are formed, each acting as a template. Guys, are you hearing me? Yes. So you get another mark, meaning that you have 2 marks there. Continue.
DNA nucleotides from the nucleus attach to their complementary nitrogenous bases. And then there are 2 DNA strands. A new DNA strand is formed that is similar to the previous one. That makes sense. I'm just paraphrasing.
Okay, you better say that. You better say that 2 new DNA are formed.
Yes.
2 new DNA are formed. Each one with original strand, each with one original strand and one new strand. 2 new DNA are formed. Each has one original strand and one new strand. Is it clear? Yes. So guys, that's it. No enzymes.
Enzyme.
The process is controlled by? Maybe. All right. Okay. Okay. Let's go together here. What is you? What is you? What is you? DNA. DNA what? DNA strand, eh? What is V? Look at V. What is V? V is adenine. Nitrogenous base, what? Adenine, eh? Are you sure? Yes, V is adenine. Guys, I told you that always, when it comes to the bases, always find out where you're gonna get a click, where you're gonna get a link. You understand? Look at this. They say this came from here, meaning that this is thymine. I see. Then automatically this is what? Adenine. Is it clear? Yes. What is Z? Hydrogen bonds, isn't it? They're saying molecule W. We have seen molecule W, ne? Let me see. Yeah, what is molecule W? This one. Is a what? Is a nucleotide, isn't it? Bond Z, we saw nitrogen base, we saw it. Where in the cell does the process takes place? Where does in the cell this process takes place? Where? Nucleus. Name the phase of the cell cycle where it takes place. Interphase. You see that now, since we have gone through there, we are able to answer any question concerning about this, isn't it? All right. Let's see if we have covered everything here. Yeah, we talked about that. We talked about that, isn't it? Yeah, we talked about that. Guys, do you have any question with the base calculations? Yeah, let's go to transcription and translation. Here I'm gonna focus more on mutations. What is transcription? Formation of RNA from Carapita. Here. Oh, you must come closer to me here. You don't want? Carapita. Must be closer. I don't want you to do anything, ne? Want you to be grasping, ne? All right. What was the question? What is transcription? Formation of messenger RNA. Is it clear? Yeah. What is translation? There is a simple definition I want to hear. There is this. Oscar, I want a simple definition, because sometimes they brought this question in prelim, and then people, they messed up because they don't know that this time they use the simple definition, and they ask you describe the formation, the process which lead to the formation of a polypeptide chain. People are describing transcription. Formation of polypeptide, that process is called translation. Yes. It's supposed to charge. All right, guys. We continue, ne? They're saying name the phase of the cell cycle. We are done with that, ne? Do you see what I'm saying? This is what I'm saying. This is what I what? I'm saying translation is the formation of what? Look, formation of what? Messenger RNA. While translation is the formation of a what? A polypeptide chain. So when they ask you to describe the process which lead to the formation of a polypeptide chain, what do you describe? You describe translation. You understand? When you mention that process, ne, you get a mark. You only don't get a mark if they mentioned that. You understand? But if that question, that process is not mentioned, then you get a mark for that. Is it clear? Yes. What is A? What is A? Okay, there is no A. What is Y? What is Y? Amino acid. What is Z? Huh? Ribosome. Yes. What is W? What is W? What is W? Huh? Guys, what is W? How? Check it critically. W, not mRNA. Whenever you see something coiled, like twisted, that's DNA. This one is DNA. DNA. Is it clear? What is X? mRNA. Is it clear? What is this? Transfer RNA. Is it clear? So the process which occurs in the nucleus is called transcription. Oscar. The process which occurs in the nucleus is called transcription. The process which occurs in the cytoplasm is called. You are lying. You are busy talking. The process which occurs in the cytoplasm is called. Hey. I got you now. Process which occurs in the nucleus is called transcription. The process which occurs in the cytoplasm is called seroma. It's called what? Seroma. It's called guide translation. Yeah. All right. Which organelle? Listen carefully. Which organelle? Where the messenger RNA aligned itself during the process of protein synthesis. Ribosome. So in most cases, we have two organelles. We have the first organelle they usually ask is the ribosome. Is it clear? The second organelle is the nucleus. So your answer must be related to that if they ask you that question. Is it clear? We continue. Give me two differences between DNA, nucleotide, and RNA nucleotide. DNA? DNA is double stranded? No. It has ribose sugar. Deoxyribose sugar. And then here, RNA has ribose sugar. Isn't it? Then DNA has thymine, RNA has. Is there any other? No, because they are talking about nucleotides. Second question, give two differences between transcription, replication. Two differences between DNA replication and transcription.
Only one strand.
Only one strand. It serves as a what? A template. In what? In transcription. Meaning that here we have one strand acting as a template. While in DNA replication, you have two strands acting as a what? A template. The second one, transcription, forms the mRNA. And then translation, transcription forms messenger RNA. Don't say it forms RNA because we have different types of RNAs. You are born. So you have to tell us it forms messenger RNA. While DNA replication, you form DNA strand. You understand? Yes. A strand or molecule? DNA molecule. Is there any other answer? Yes, the other answers. What is the similarity between DNA replication and transcription? What is the similarity between DNA replication and transcription? Check. What is the similarity between DNA replication and transcription? Nakato, give me answers. Give me answers. Give me answers. Hey. Most of them, they form what? Template strands. Okay, look. What is the first statement in each? What do you say? DNA double helix in DNA replication. DNA double helix unwinds. What about in transcription? DNA double helix. Meaning that if you have, you can say that in both processes, DNA double helix unwinds. Wabon? Yes. The next one. In both weak hydrogen bonds break. I see. You check the process and you find out. Ne? Yes, check the process and do what? And find out. By the way, guys, your parents are watching. That camera is working. You see? You see it? Yeah. So please don't disturb. You disturb, you are recorded. Is it so? But feel free. So your parent is there. If you're busy disturbing here, your parents busy watching there on a big screen on the TV. All right, let's continue, guys. Let's continue, guys. Yes. Can we continue? Hey, guys, don't forget the reason why we are here. We want marks. Ne? We want what? 1 marks. Describe the process of transcription. Describe the process of transcription. Let me even stand. Describe the process of transcription. Okay. Yes. You want to talk? Yeah, Given. Yes. Talk. Yes, the words and why? With hydrogen bonds break, to form two separate strands, one strand acts as a template, to form mRNA strand, using free floating mRNA nucleotides, from the nucleoplasm. Complemented using complementary base pairs adenine with the uracil and the cytosine with the guanine. Then it moves out of the nucleus to the cytoplasm on the ribosome. Thank you so much. Thank you so much. Nana, are you interested? All right. We got the next question. Okay. Let's continue. Let's continue, guys. Let's continue, guys. Translation. 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 When I stand up. When I stand up. Stand up. Stand up. Take your chair. Come sit this side. This side. I told you if you start making noise, I will separate you. I'm starting with that one. Next one is coming. Make sure you don't block the people behind. Okay. Let's continue now, guys. Guys, we are here to look for marks, Yeah, not catching up. Not catching up. I'll give you time to catch up during break. Yes. All right. Let's continue, guys. Let's continue, guys. Describe the process of translation. Guys, you cannot go to the paper without knowing the structure of DNA, without knowing DNA replication, without knowing transcription, without knowing translation. Wabona? Yes. Please don't make a mistake. So when you go back on Saturday, sleep. On Sunday or Saturday night, read these processes, because these other questions, we will be done with them. You only go back to read the questions, the long questions like DNA replication, transcription, those ones. Is it clear? Yes, but these other questions, you will be ready to write. So, transcription, translation, you talk about according to the messenger RNA. You can use any other language, as long as we find the key points. According to the messenger RNA codons. Once we hear the word messenger RNA codons, we give you a mark. The transfer RNA with the complementary, that word complementary, anticodons, we give you a mark. It brings the required amino acid. That word required amino acid is a mark to the ribosome. Sometimes even the ribosome, they give you a mark. The amino acids are joined together by these bonds, which we call peptide bonds. Guys, we don't have polypeptide bonds. I repeat, we don't have polypeptide bonds. Onele, we don't have what? Polypeptide bonds. We have peptide bonds. So amino acids are joined together by peptide bonds to form a required protein. Is it clear? Yes. How do you call the bonds between two amino acids? How do you call them? Peptide bonds. How do you call the structure? A structure which is made up of 20 amino acids.
Protein.
How do you call the structure which has 20 amino acids? It's not a protein. It's a polypeptide chain. We only call it a protein when it has 50 amino acids and above. Is it clear? Yeah, we only call it a what? A protein if it is having 50 amino acids and above. Then we shall say that it is a protein. Is it clear? Yes. Now question. Let's go to the calculation before to go. We got the mutations. If I have 21 bases, ne? How many? Are you hearing? How many anticodons do I have? Codons do I have? I have 21 bases. How many anticodons do I have? If I have 21 bases, how many anticodons do I have? You're saying 7. Meaning that I have, remember, each anticodon is made up of 3 bases. You understand? Each anticodon is made up of what? 3 bases. You see that each anticodon, it has 3 bases. You see? 1, 2, 3. Yes. If I have 21 bases, codons, how many anticodons do I have? If I have 21 codons, how many anticodons do I have? 21. Because the number of anticodons are equal to the number of codons. Now, if I have 21 codes, 21 codes, yes, how many amino acids do I have? Yeah. If I have 21 codes, how many amino acids do I have? I skipped it. How many amino acids? Guys, I'm still waiting for the answer. I should check online. You're passing it to the online. If guys online, they are saying that give us the answer. Give us the answer. Don't just keep quiet. Give us the answer. We are waiting answers from you, guys from online. The class is saying. They are saying how many? They are asking me also. Guys, okay, let me explain it like this so that you understand it better. Because you will not understand it unless you know these concepts. Ne? Yeah. You have code. Code are 3 bases on DNA. Is it clear? I see. You have codons. Codons are 3 bases on messenger RNA. You have anticodons. These are what? 3 bases on transfer RNA. Is it clear? Yeah, so it means that you have 3, 3, 3, 3. Wabona? Meaning that these are 3 bases, 3 bases, 3 bases, 3 bases. Meaning that if I have 21 code, I have 21 codons, I have 21 anticodons. Isn't it? But each code codes for 1 amino acid. You understand? Each code, each one code, each one codon, each one anticodon code for 1 amino acid because each code has 3 bases. Each one amino acid code for 3 what? Bases. You understand? So if I have a code, I'll have 1 amino acid. If I have a codon, I have 1 amino acid, isn't it? If I have 1 anticodon, I'll have 1 amino acid. Now, if I have 21 code, it means that I have 21 amino acids. So this concept will help you to interpret the graph, the table where we are going now. Is it clear? Let's go. Let me look for that. Yes. This is the table I'm talking about. Nakato, they are saying that they are also waiting answers from you guys. Yeah. So if you expect you get answers from online, even people from online, they are waiting answers from you. Yes. Yeah, ask. Yeah, what? Code is for DNA? Yes. Yes. Under DNA? Yeah, the O is 33333. Yes. Uh-huh. Can we continue now? You need to know how to interpret this. Okay. Let's continue. The sequence of amino acid in the protein molecule is coded by DNA and RNA. The table below shows some of the messenger RNA codons and the respective amino acids. Okay. Are we there? Yeah, we are saying that according to the table, how many codons are coded for phenylalanine? You see, how many codes? What does it mean? When they say codes, codons, it means that it's linked to messenger RNA. That's why here they said messenger RNA codons. I think so, but they're saying how many zoom. Yeah, can you see now? They're saying how many codons code for phenylalanine. This one. How many are they? Are you sure? Yeah, it's this one and this one. So there are two, isn't it? Do you know what they say? Do you know what sometimes they say? Uh, yeah, actually, phenylalanine is here. Phenylalanine is this and this. So look, these codes, because these are codons, eh? The first one is this, eh? And the second one is this. This codon is different from this. If this was UUU and also this one was UUU, then it would have been one, isn't it? But because now this one is UUC and then this is UUU, so they are different. So we have two codons. Is it clear? Is it clear, Nakato? Yeah. What is anticodon for? What is the anticodon for glutamic acid? Glutamic acid is here. So they are saying what is the anticodon, meaning that you have to convert it into messenger RNA. So it's gonna be C C U C. Yeah, I see. C U C. I see. Eh. Section of messenger RNA has the following base sequence. Is it clear? Huh? Don't put your head down. Wow. Are we together? Or you want to come close to me? They're saying that section of messenger RNA has the following base sequence. Are we there? And is read from left to right. Okay? You have it here, isn't it? DNA base triplet for the last codon on the section of messenger. Give the DNA base triplet for the last codon. They're saying last codon. But they want the DNA base triplet, meaning that you have to link the codon. The codon is found on the messenger. Is it clear? Oscar, is it clear? Yeah. The codons are found where? On the messenger. So now, how are we going to convert it? You said last codon. So 1, 2, 3, 4, 5, 6. They're talking about this, isn't it? So it's gonna be what? It's gonna be T GG. Is it clear? The first amino acid coded by this section of messenger RNA, the first amino acid. So what's gonna be the answer? So remember, look, the trick is here now. This is what I wanted. Guys, look, remember they're saying the section of messenger RNA has the following base sequence and is ready from left to right, isn't it? And then they're saying the question, the first amino acid coded by this messenger RNA. Wabona? But what is here? Is a messenger. I think. So am I going to change this? No, because they say this is messenger, is a codon, is messenger, which means that this is a codon, isn't it? And what they're asking here, they're also asking the messenger. I think. And what is in the table is also messenger, meaning that I don't change anything. Unless they were saying that this is transfer RNA, tRNA, and then what is in the table is a messenger. Guys, stop making noise. Is it clear? So then that's when I would have changed. Guys, always, I repeat, always try to link them. If they are not coming together, like code in the question and in the table is not code, is anticodon, it means that you have to change it into codon, then you change it again into anticodon. You understand? If they say it's DNA and what is in this table is transfer RNA, then you change it 2 times. If it is DNA and what is in the table is DNA, then you don't change it. If it is DNA and what is in the table is messenger RNA, then you change it 1 time. Is it clear? Please, because these are the marks you can't miss. Even if you don't revise, you can answer those questions from there. You don't need to revise to get the mark from this. Guys, paper 2, if you know that you made it in paper 1, you're gonna make it in paper 2, because the paper 2 is simple. I'm telling you, you will see, you'll tell me. When you're choosing DNA, the yes. The DNA. Then it uracil goes with adenine, so they remain the same. Yeah, uracil goes with the adenine. If you are changing, unless you are changing back to DNA, then uracil will go with. Actually, still uracil will go with the adenine, but adenine will go with the thymine now, not uracil. Yes. All right. Let's continue the next question, guys. Yeah, here is your friend. Here is your friend. The one you don't like. A mutation occurred which resulted in the... Are we there? A mutation occurred... Are you there? Oh, a mutation occurred which resulted in the following base sequence on the messenger RNA molecule. You have this, you have this, you have this, you have this, you have this. But we need to know what changed. We don't know what? Yeah, this is the same, So we take it, isn't it? This is the same, This is the same. This one is not the same. I see. What about this? And then this. Now, guys, when they give you this question, the first step, you need to know to find out what is the mutation. Then you write it. So now you have identified the mutation. That's the first step. The second step, you need to understand what does this code for, which amino acid does it code for, and what does this one code for. Wabona. If you find out that this one codes for X and this one also will have no effect on the protein, But if you find that this is X and you find that this is Y, then it means that the mutation will have an effect on the protein. Is it clear? Did you get it? I said that if you find out that the amino acid coded by this is X and the amino acid coded by this is also X, it means that this mutation will not have any effect on the protein form. But if you find out that this one now codes for X while this one codes for Y, it means that that mutation will have an effect on a protein because a different amino acid has been formed. Is it clear? Now we need to identify that and you explain it. Let me clear up here because now we know what all right. So we said this and that. Describe the mutation that occurred. So now we are going to describe what happened here? What happened here? Because now we are describing. When we describe, first identify it. What is it? It's a gene mutation. A gene mutation is a mutation whereby it results in the change of the genes. Tinile. Back to class. Number 2, you need to tell us what changed. It resulted in the change of the third base from C to U. You are done with description. This is a gene mutation whereby the third base C was replaced by U. Is it clear? Yes. Sometimes gene mutation, we call it point mutation. Is also correct. Point mutation. Because it's just happening on a point. Is it clear? Yes. All right. We go to the next question. How will the effect of mutation described above will have a resulting protein? Will have resulting protein. Now we need to find out which amino acid does it code. AGC. We said that this is codon, isn't it? So we don't need to change. AGC is what? AGC is serine. AGC is serine. And then AGU is also serine. I think, meaning that this will code for the same amino acid which is serine, isn't it? Therefore, it will have no effect on the protein. Pastor, I'm gonna remove you from there. I'm telling you. I'm telling you and your friend. Yes. Speak louder, I can hear. If they ask, explain the effect, yes.
You just compare, you don't change anything.
Yeah. The reason why we didn't change is because they say that this is a messenger RNA. You see it here? This thing is messenger RNA. And then also, they say this is messenger RNA. So we don't, it's messenger RNA. When you go back to the table, what did they say here? It's messenger RNA, meaning that we don't need to change anything.
It's a different thing. Let's say here you change.
Let me give you an example, another example. Look at this question. I sing it. They're saying gene mutation. Okay, we are done with that. What will happen if a mutation changes base triplet number 1 from ATG to ATA? You understand? AT is what? T is what? It's thymine, which is found where? DNA. I see. Meaning that, but what is in the table? Codon. Codon is, it codes for what? Is for what? Is messenger RNA. You understand? So in this case, because what is given here is DNA, but what is here is messenger RNA, therefore you have to change them first before you identify. So it means that you're gonna say ATG is gonna be U, because we are taking it to messenger RNA, and then A, and then C. Then I go and then look for it. UAC. UAC is what? UAC is tyrosine. I see. And then ATA. ATA. I see. ATA is gonna be what? UAU. I see. So UAU, what is it gonna be? It's gonna be also, you see, tyrosine. It means that there will be no effect on the protein. But don't just say there will be no effect on the protein. You have to tell us that this change to this, which code for the same amino acid tyrosine, therefore there will be no effect on a protein. Is it clear? What about now this? They code for the same amino acid, and then you mention that amino acid based on the question. Is it clear?
Yes.
Concierge, I need to see you. I need to hear you. Is it clear? Cariopita. Cariopita? No, Cariopita is my daughter. Eh? You are my daughter. Cariopita. All right. We continue. Now let's take this another this example. We say that T. Let me clear it up. Let me clear, guys. Even if you are dozing, even if you are doing what, don't make a mistake to go to the paper. Onel, please. Yeah, don't worry. Where is Karabo? Karabo is not here again. But Karabo. Seroma, are we together? Seroma, I don't want to see you in the face because you're gonna do like this. All right. Okay. We are saying that, please, even if you are dozing in the paper, please make sure that you answer that question because it's simple. Okay, let's see this one. T-T-A. So it's gonna be what? It's gonna be what? A-A-U. So A-A-U code for what? A-A-U code for what? Valine. Ne? Alright. And then it's gonna change to T-T-T, which is A-A-A. Is it clear? Which is what? Proline. Meaning that, guys, keep quiet. Tini, this is the last time. Mem, come take away Tini from here. Go there. Go there. Go there. Don't talk to me. Go there. I don't care. Go there. Yeah, because you are disturbing. I've been warning you. Yeah, go sit there then. So, let's continue, guys. Hurry up, I'm waiting for you. Let's continue, guys. To the next question. Yes. You want to say something? All right. So we are saying that. So we said that this one codes for valine, and then this one codes for proline. Is it clear? Therefore, we're gonna say that this one changed to this. You understand? Of which AAU code for valine. Is it clear? And then AAA code for proline, which is a different amino acid. You understand? Therefore, there will be a different protein. So please make sure that you caught what changed and you caught what formed. Is it clear? Yes, because if you just caught what formed and you don't caught what changed, you lose marks. And I don't want you to lose those marks. Do I need to talk about DNA profiling?
Yes, you want
DNA profiling, everyone must pass it. Okay, it's fine, it's fine. Let's just get 1 or 2 questions. Okay, let's smash this one. Oh, this one is similar to the book we did, ne? There is another question which they ask, ne? I think I should ask you also. But before I ask you that question, let's just go through this and then we go to profiling and then we can start meiosis. What is this? What is W?
DNA.
DNA. DNA, double helix, DNA molecule. Ne? Yeah. What is X? What is X? What is Y? Tinil. What is Y? What is Z? What is Z? Ribosome. What is process B? What is process A? What is X? What is X? This one. This one. This one. This thing. Nuclear membrane. Are we there? And then what is the hole in the nuclear membrane? Nuclear what? Nuclear what? You know why I'm laughing? I'm just remembering what you were saying yesterday here when I was saying 4S, the sweat duct, the sweat gland, then the blood capillaries, and then boom, the same question, boom. All right. Hey, people don't remember. All right. Okay, thank you very much, guys who are online. Your answers, I see them. Nuclear power, good. Hey, when you have a good name, dog. Hey, okay, dog. Back and I see. Yeah, it's a dog. You call yourself a dog, Mama. But dog, you're giving me correct answers. Thank you so much. I like your name. You remember yesterday there was someone who said what? Kids online. Yeah. All right, guys, let's go to the next question. Let's go to the next question. Let's just smash this one. Describe how a mutation. This is the question which I wanted. Ne? It's a mutation, but it's a general question. Ne? It's a mutation, but it's a what? It's a general question. Ne? Describe how a mutation on molecule W will affect the structure of protein formed by process B. Hey, will you see? Do you see now? They're saying describe how a mutation on molecule W first identify. W is DNA. They're asking how is the and then process B. Yes. Who can tell me the answer? 4 marks. Yes, ask. You're asking me, don't you explain or you're gonna explain?
I'm going to explain, but then I need clarity.
Okay?
You need to explain from like the DNA to the mRNA to the tRNA.
Exactly.
Okay, so if the DNA strand is altered with, it's going to affect the complementary strands that is going to form of the mRNA. The mRNA is and the mRNA codons are going to choose. I wanna change the anticodon.
Okay.
mRNA codons are going to change the anticodon so that it's going to match with
Online, you must also tell me.
the ribosome. Get that right. So should I continue?
Continue.
And then the anticodon, which is going to affect the protein that's going to
Yeah, man. Yeah, it's true. She's saying that what happens if there is a change in the DNA sequence, it will affect the sequence of messenger RNA formed. You understand? Of which this sequence will also alter, will also alter the sequence of amino acids. You understand? Then it will be a different protein. Change, now this is the correct answer, the perfect one. Change in the base sequence of DNA, change in the, yes, I'm coming. Change in the base sequence of DNA will result in the change in the base sequence of messenger RNA. Wabona? Will result in change in the base sequence of messenger RNA, which will result slow but show. All right, which will result in what did I say? Only thing is not memorized. I said that change in the base sequence of DNA will result in a change in the base sequence of messenger RNA, which will result in the sequence of tRNA, bringing the amino acids. I think it's the same thing. Yeah, which will result in a change in the sequence of messenger RNA with the amino acids. You understand? It's the same thing. I said what? Yeah, tRNA, not messenger RNA. Sorry. Yeah, tRNA with the amino acids. Hence a different protein. So basically, it's like a chain, bond. Change in the sequence of bases of DNA will result in a change in the sequence of bases for messenger. You understand? Yeah, you use your English, but it must have those sequence for what? Which will result in a different sequence of transferrin A with its amino acids. Wabona? Yes, meaning that brought to the ribosome. You understand? In a different sequence of amino acids or transferrin A with its amino acids. You understand? Yes. Hence a different protein. Yes. Do you understand? But sometimes, guys, the question does not start from DNA. It starts from messenger RNA. And then now you see the fire. So it starts somewhere. So when it starts somewhere, what does it mean? You also start from there. Change in the sequence of messenger RNA will result in a different sequence of transferrin A with its amino acids, hence a different protein. You understand? Yeah, so it depends on where it has started. Consion. Ne? Are Peter? Are you here? All right. Yes. Yeah? Wait, your question. What you are saying? Which mutation? We have two different, we have two types of mutation. Mutation, what we talk about, both of them we talk about them. The first one is the gene mutation. We have different mutations, but based on our curriculum, it's only two. The first one is a gene mutation, or what you call point mutation. Is as a result of, it can be a deletion, it can be an insertion, it can be duplication, it can be, you understand? Meaning that the bases, you understand, can change. The sequence of the bases change. It's an error. What causes error? What causes an error? Yeah, sometimes when it is transcribing, there are some errors. There are some enzymes which are there which try to rectify that error. Sometimes those enzymes, they skip that error, and that error is maintained. You understand? Whenever you are born, when you are manufacturing things, there are some things which come out right and some things which come out not right. For example, I'm going to give you a best example, but I'm not demarketing them. There are some things which go to Woolworth. You call it what? Whatever. I don't care. Ne? They sell their nice clothes, what. But there are some clothes which have some damage. They send them to specific shops. You understand? So those clothes which have damage, those are the mutations now we are talking about. So maybe the process going and then something happens accidentally. That's a mutation. When DNA is forming, a simple mutation occurs, meaning a simple mistake occurs. Instead of G, C is gone. And then now it comes in. It's random. No one can detect it, but you can introduce it to make sure that those mistakes are there. We call it induced mutation, but we don't leave it. Yes, guys, the second one is called chromosomal mutation. Chromosomal mutation, that is Down syndrome. The whole chromosome change. It can be removed, or it can be added, or it can be modified, but the whole chromosome must change. That's why you call it chromosomal mutation. Sometimes, scientifically, when you go to the other side of ours, we can call it chromosomal aberration. Hey, guys, you must ask question which is related to. Yeah. We are looking for marks today, ne? Yeah, not knowledge now. Just marks, ne? Yes. Akira, I said you didn't stop. Okay, what is it? Kuluma. Okay. Conditions for gene mutation, like what? No. They just want to know gene mutation or chromosomal mutation. That's it. Chromosomal mutation occurs in meiosis, Down syndrome. We have even other syndromes, but we don't talk about them. Yes. Okay, guys. Who is the most likely suspect? Huh? Zoom. Yeah. Which one was the most likely to be the rapist? How do I identify it?
You can see by it.
You can see by eyes, ne? You see this is a semen. Can't you watch semen? Sperms, ne? Shh. Nana. Am I fine when I'm saying mutation? Okay. Suspect 3, ne? Because you see that, make sure that yeah, I think it's suspect 3, ne? Yeah. Explain your answer. How do you explain your answer? I want to hear specific words from you guys, because there are some specific words we don't mark. I want to hear from you first. Eh. That's what. Where are you? What's what? Wisdom. You want to give me the answer? Wisdom. You can't say that. What are you saying, Nakato?
I think that the blood from the skin.
The blood from the bars.
Yes.
You mean these ones? The bars. Yes.
That's
They match with
suspect 3.
suspect 3. Okay. The DNA profile of the semen matches with the DNA profile of what?
You are saying what?
Yes. The DNA profile of the semen matches with the DNA profile of the what? Of suspect 3. Mm. Mm? Mm. Oh, okay. Eh, can we continue, guys? Hey, guys, shh. When's that? Where are you going? Hey, guys, I don't want too many people going to the toilet. 1-1. 1-1. This business of going to the toilet and then you start talking there. We are here to look for marks. All right, let's continue. Yes, ask. This one it doesn't have too many questions. Okay, let's finish up. Give one reason why the evidence may be considered reliable. Give one reason why, meaning that, guys, look, shh, they can ask you, why is DNA profiling can be considered reliable, or it can be considered conclusive. You understand? Is a conclusive evidence when they do a DNA profile of your father, a DNA profile of you, and they see that there is no match, they can use it to conclude that is not your father. Why is it that DNA can be used to conclude? What makes DNA profiling to be a reliable source? That's what they are trying to say. Hey. Yes, you're saying so. Yes, tell me the answer. Yes. Uh-oh. Samantha. Kulum. Yeah, DNA profile. DNA. Guys, each individual has a unique DNA. Even identical twins, they don't have the same DNA. You understand? That's why we say that. But on only using one, like if you use a large sample, yes, then DNA is unique. Unless you use a small sample. Look, you'll find out that there are some bars from here which will match with other suspects. Meaning that if you use that small sample, it means that you won't be able to, you won't get the actual person. But if you use a large sample, you understand, and then they don't match, they don't match, then it means that it can be conclusive to conclude that this is reliable results. So the answer is we don't have the same DNA. So each individual has unique DNA. You understand? Yes. No, large sample. For example, what you are going to use. If you are going to use blood to test for DNA, blood must be a lot, enough. It must be enough, not just one drop or very small, of which that may not be able to be used to conclude that you are the person. Yeah. Other cases, who say that they use small samples? No, when they talk about small sample, a hair can be enough. You understand? Yes. But we are talking about in the language of DNA. Yes. That's why they have to look for even other evidences, not just one. All right. Eh.
So Nina was this question.
I want to go to meiosis, guys. Eh.
Father is first, but then that's your answer. You'd have to be like the father is most likely to be the father because the DNA samples of the males kind of similar. So is there a way we find there is a unique DNA but it's not completely unique. It can be similar to somebody else's DNA.
Eh?
I don't know how that is.
You know what they do? The other question I wanted to talk about. Eh? In most cases, it happens if it's the father and the mother. You understand? Listen carefully, guys. When they bring this question, because I wanted to conclude with that. Ne? I don't know whether that's what you're asking. They bring the father. They bring the father here. Ne? They bring the child. Then they bring the father. They bring another father. Then they bring the mother. You understand? Then they say one. Let me just use numbers so that I'm fast. 1, 2, 3, 4, 5. You understand? If these are the bars, these are the bars, ne? And then now here, this one has 1, 3, 5. You understand? Then this one has X, 2, Y, 4, Z. I see. Then this one has 1, 8, 10, 11. I see. So then they ask you, who is the father? Maybe there is an X there. Who is the father? If this is the father 1 and the father 2, and then here is the mother, and then this is the child. You understand? Then they ask you, who is the father? You're gonna say F1. Then give a reason for that. Yes. So it means that you have to first identify which bars match with the mother. You understand? You find out that this matches with the mother. You understand? And then this matches with the mother. The remaining bars of the child, it must have that one. The remaining bars of the child must match with those of the father. I see. If there is any one bar here which is not matching, that's not your father, that's your relative. Maybe the neighbor did something. Hey, hey, are you sure your father is your father? Hey, hey, hey. That thing, yeah, it makes me not sleep. When they ask you a question, are you sure your father is your father? I'm not sure. Seroma, I'm not saying anything. So you have never gone somewhere and then they tell you that you resemble someone. Eh? Sharon, you have never gone somewhere and they tell you that you resemble someone. Sharon, don't lie. Sharon, don't lie. Hey, hey. Hey, you want to sue me? You don't. Listen carefully. Let me tell you something. The things you say. You say, my brother from another mother, my brother from another father. What does it mean? What does it mean? My brother from another mother, my brother from another father. What does it mean? And you know that it's not your brother. What does it mean? What does it mean about your mother? What does it mean? You see? I don't know anything. Hey, live. All right, let's continue, guys. Yeah. Life is good. Hey, life is good, Baba. Hey. Actually, you are reporting to us that something happened before you came. Amen. Yeah? Yeah? Even if the same age. Oh, the father can work here and can work there. Hey, same day. Same day, Baba. Hey, same day. let's go to meiosis. Yeah. You make me laugh, ne? Guys, let's go through meiosis very fast. Maybe do you want to first come? For we should go for meiosis. They go for the short break. Then we come back for this. Isn't it? The first one is short. The second one is the problem. You know what I'm scared about, ne? The moment you go outside, ne? And then the air comes. That is the problem. You're gonna come back when the eyes can't see. That is the problem. All right. Let's continue, guys. Let's go to meiosis. Meiosis. Let's use this. Ask you questions. Then we continue. Okay. Where does meiosis occur in animals? Where does meiosis occur in animals? Where? Where does meiosis occur in animals? Ovary, ovary, and testes. And then in plants, anthers and ovaries. Yes. So remember, in animals, ovaries to produce ova, testes to produce spermatozoa. Yes. And then ovary in plants to produce ovules, and then anthers to produce pollen grains. That's done. What is the function of meiosis? Give me two functions of meiosis. Produce gametes. That's number one. Number two? Have the number of chromosomes. Number three? It maintains the chromosome number. It maintains the chromosome number from generation to generation. You are forgetting something. Variation. That's why I'm asking you. Are you sure your father is your father? No, it's a question. I'm not insulting anybody. It's a question. The answer is yes or no. Then we go. No, that simple. We don't know. I like it. I like it. Yes. No, there's a daughter in blood and the daughter in blood. Yes. Okay, guys, we continue the next question. Can I play something? I'm so shocked. When someone says that I look like Kevin Hart, why not his mother? Why not his father? Why? I don't know. I don't know. Before my mother, after my mother died, my dad was my son. So, you must they have confirmed. Before you die, you come. Come, I tell something. You know what? I lost your father. That guy, the neighbor, is your father. Alright, guys, let's continue. Anyway, you might not resemble your father or your mother because of variation. Is it clear? You don't need to resemble any of them to be belonging there. Actually, you can even have someone who resembles them, but they don't belong there. Since we came from the same individual, Adam, meaning that we have some genetics which we share. Maybe that one came out. Ne? Yeah, so don't stress about it. Ne? Yeah, we continue. Guys, now we come back to the business. Replicated chromosome and replicated chromosomes. Remember, we say that, guys, when you have this anaphase, how do you call this? These are chromatids. Okay. But the moment they reach telophase, they are what? Chromosomes. They are called what? Single-stranded chromosomes or unreplicated chromosomes. Please, when they are in telophase, they are chromosomes. But when they are anaphase, they are chromatids. Is it clear? That's why we have this when we say that unreplicated chromosome or replicated chromosomes. Is it clear? When DNA replication takes place, you form the double strands. All right. Describe. Don't want this. Describe the karyotype of a human. 5 marks. Yeah, describe the karyotype of a human. 5 marks. Karyotype of a human. Karyotype of a human. Wait, 5 marks. Karyotype of a human. Zones, of chromosome 1 mark. From pair number 1 to pair number 22 is called autosomes. Is it clear? The 23rd pair is called gonosomes. Is it clear? The 23rd pair is responsible for sex, not sex, sex determination. Is it clear? Yes, gender. Which is made up of seroma. Which is made up of what? Which is made up of X and Y. The X chromosome is longer than Y. Is it clear? Are we there? Actually, and don't forget that these chromosomes are arranged in homologous pairs. Is it clear? Yes. So that's why here you are finding here human cells made up of 46 chromosomes or 23 pairs, and then each are different from each other by size. All right. 1 to 22 is called autosomes. The 23rd pair is called gonosomes, which is made up of X and Y. X and X for female, X and Y for male. X is longer over Y. That is, you are describing a karyotype. If they ask you, 5 marks. You have a distinction material book, Win. Yeah, it's there in the distinction material book. Yeah. You're saying what? Hey, don't do that. Don't do that. Sell it. Sell it. If you bought it, sell it. I'm telling you. We got the next question, guys. In which phase does crossing over takes place? Prophase 1. Prophase 1. The word 1 must be there. Don't say prophase. When you say prophase, 0. Describe crossing over. Yeah. Onsion. Describe crossing over. Yes? Homologous chromosomes. Pair. 1 mark. Chromatids of what? Chromatids of homologous chromosomes overlap. They overlap at a point called chiasma. They exchange genetic material.
That causes variation.
Causes variation. So you form 2 chromosomes which are genetically different. Thank you so much. Shh. Nana. Don't do that. Don't do that. Shh. Can we see the questions now? Let's go to the questions. Draw a diagram to show the cell during anaphase 2 of meiosis. What is this? Which phase is this? Which phase is that? Which phase is that? Look at the chromosomes. Guys, look at the chromosomes. The chromosomes, what are they? What's happening to them? Look at them. It's single. They are single. It means that it's meiosis what? Meiosis 2. Because they're not homologous. If they were like this, then it's meiosis what? Meiosis 1. But now they are not homologous. They are single, you see? Then this is meiosis 2. Prophase 2. You understand? So they want you to draw a cell under anaphase 2. Therefore, you have to draw a cell. Guys, listen carefully where people lose marks. Ne? When they ask you to draw, please pay attention on the variation. Ne? Alright. Pay attention on the what? On the variation. For example, if you look at this chromosome, it has a white one and it has white and a dark part. I see. Then you come to this one, it's dark, all of it. Yes, and then it has a white and most of it is a dark one. You understand? You see it, this one. So that thing will give you a mark if you see that you have it. So the variation are very, very, very important. Don't forget the caption, the title. And I said, don't forget, listen carefully. In paper 1, they didn't bring anything related. Listen, I promise to you, paper 2 is going to be easier. You're going to draw. You're going to draw a pie chart or a bar graph or a line graph. Paper 1, it was not there. You're going to draw. Guys, those marks, don't lose them. In paper 1, they brought the percentage increase. You won't find it in paper 2. That one is done. Put in the pin. Hey. All right. We continue. Don't discuss about paper 1 now. Sir. 300. Yes. All right. People were there talking about 75 cents. Which was oh yeah. All right. Okay. We got the next question, guys. The diagram below shows. Shh, guys, come back. Waswa, you're gonna come close to me now. Waswa. You want me to start you, ne? Akira is sitting in your place. I'm telling him. The next time I tell you, you are also okay. Let's continue, guys. Shh. Oh, what is A? What is A? Come back, guys. Come back to class. Guys, you are making noise for people who are online. Yeah, come back to class. What is A? What is A? Spindle fiber, non-spindle, spindle fibers. What is B? Centrio. What is C? C. Cell membrane. What is D? Chromosome, non-homologous chromosome. What causes these variations in the chromosomes we see? Crossing over. Which phase is that process? Give the advantage of the process you mentioned above. Variation. Leads to variation. Yes. They can even ask you, how is this process biologically important? How is that process biologically important? Meaning that it leads to variation which which brings about different survival rates. You understand? We will survive differently. Don't you see that all of you are here in class? When the paper comes, you'll get different marks. That is variation. You understand? Yeah, that is what? That's variation. All right, we continue now. State two characteristics of homologous chromosomes. What? Eh? Like structures. Thank you for trying. Huh? The what? They're in pairs. Thank you for trying. Characteristics of homologous chromosomes. Guys, they want the characteristics of homologous chromosomes. You talked about the karyotype here. Hey, say it. Same size. Is here. Thank you. They said it here. You see, different from each other by shape. So meaning that they have the same shape, same size. You understand? Same genetic composition. Do they have the same genetic composition? Yes, but different locations. No. I'm talking about the positions. Like chromosome for I, they have the same I. Chromosome, like, let me show you. If this is chromosome X, and this is chromosome Y, X and chromosome Y, and these two chromosomes are homologous. And this chromosome, the gene for big nose is here. Even here, the gene for big nose will be here. It can't be here. You understand? Okay, same locus of genes. Better that way. Same locus of genes. Everything is the same in terms of the structure. But inside, it's not the same. How do you know what's the answer? How do I know what's happening inside? I said same size. They have the same size. They have the same shape. They have the same locus of genes. Same locus or same loci of genes. You understand? The genetic composition could be different, but the same loci of genes. Meaning that the genes, they have the same positions. Loci. Locus is one. Loci are many. You don't say locuses. Same loci. Same locus. Yeah. Any other question? We continue to the next question. They're saying that identify A. We know that. How many? The problem. Lee, you see behind you, there is a camera. Just behind you. You'll find this video in your parents' group. Hey, I'm like that. If I see you or someone disturbing me, I'm like that. All right, guys. Let's continue. How many each of the following chromatids present? How many chromatids, guys? 8, and then centromere 1, 2, 3, 4. Yeah. Draw the H they want to draw, Draw the diagram to show what where chromatids here 8 and centromere is what? 4. Chromatids are 8, you see them. Draw the labeled diagram to show one cell that would be formed at the end of telophase 1. They are saying at the end of telophase 1. Meaning that you're going to draw this. Guys, which phase is this? Yeah, which phase is this? Anaphase what? Anaphase 1. Meaning that you're going to have that. Wabona? Yes, that's what you're gonna what? You're gonna have. Anaphase 2, the chromosomes will be chromatids. They will be chromatids. They will be like this. Look here. They will be like this, you see?
Yes, sir.
It will be like that. Like that. So it won't be whole chromosome. But here, once you see them, they are still whole chromosomes. It means that they are still in they are still in meiosis one. You see, they are still in pairs. Are we together? Wisdom. Are we together? Yes. We continue the next question, guys. Hey, where's my question? Can we smash? Let's smash this one. Should we smash this one? Yeah, I'm looking for questions we must smash. Yeah, let's smash this one, eh? It looks to be ugly. Study the diagram of the phase during meiosis, ne? Guys, you're gonna describe for me non-disjunction. That's the last question which is remaining. Non-disjunction. Yes. Yeah, after this, and then I want you to tell me the chromosome number in each cell. Study diagram of meiosis during study diagram of the phase during meiosis. All right. Now they're saying identify the phase in the diagram above. Guys, telophase 2. Good. Telophase 2. Isn't it? Give 2 visible reason for your answer. 4 daughter cells, that's 1. 4 daughter cells, that's number 1. Unreplicated chromosomes, you see them? Yes. That's number 2. Yeah, basically the unreplicated chromosome. They are even, you see, even the nuclear membrane is back. Wabona. Contraction of a cytoplasm is called what? Contraction of a cytoplasm. Contraction of the nucleus. Karyokinesis. Okay. Karyokinesis. Karyo means what? Means the nucleus. That's why you say karyotype. Waswa, which kind of nucleus do you have? Which type of nucleus do you have? That's why I call it karyotype. Wabon. Yeah. Tell me something. I want to call you Nana. Nana, it means, to me, it means someone I care about. If I call you Nana, you know that I care about you. Okay, I'll call you Nana. Yes. Eh? Eh? Sharon. Sharon in Nana. All right. All right. They're saying that how many? Bona. Eh? It's about this. Eh? Wait. Let's first finish this one. Yes. Guys, come back here. Can you repeat? Are you here? Can you repeat? Are you here? Eh? Say that you are here. Okay. If you are here, let's record you. How many chromosomes? Eh? Onel? Eh? Always 2. Hey, don't worry. You don't need my pen. Hey guys, how many chromosomes are present in each diagram? We said how many? You go back. Shh. Guys, look here. This question, you always fail it. How many chromosomes are present? Sorry. Give me that phone. Give me the phone. Give me the phone. Give me the phone. Give me the phone. I'm not starting before that phone is here. You'll get it in the morning. Eh? You know the principle. You know the principle. If you don't know how to use your phone, I'll teach you how to use the phone. Eh? Yeah. Come back. I'm not taking your phone. I just want you to concentrate, and then you get marks at the end. Eh? Yes. All right. We continue. If they don't say that this is a human cell, you count the number of chromosomes you see. Is it clear? But if they mention it's a human cell, now you answer it in a different aspect. You answer it depending on the phase in which that cell is. If it is in meiosis 1, it will be 46 chromosomes. But if it is in meiosis 2, it is 23 chromosomes. When so better way. Yeah. Actually. Oh. Then they're saying that number 2, they're saying that now we need to find out. They didn't mention here that it's a human cell. Okay. So you're saying that how many chromosomes are present in each cell in the diagram? 20. Did I say human cell? It's not a human cell, meaning that it is 2. 2. We see 2. 1, 2. What were present in original cell at the start of meiosis? 4. The cells in the diagram are not identical. Name 2 processes during meiosis that led they say during meiosis. So you don't mention anything outside the meiosis. So crossing over, not crossover. Yeah, crossing over, number 1. Crossing over, random. Fertilization is not in meiosis. Random alignment of chromosomes. Random arrangement of chromosomes. When you say that, you will get 0. When you say random arrangement of chromosomes, you will get 0. Where? When? Random arrangement of chromosomes during metaphase. That word must be there. During metaphase. You have to tell us which phase is it. Ne? Yes. I told you always fail these questions. These last questions. Explain the significance to the species. This is what I said. Explain the significance to the species of the cell being different from each other. This is what I said. You see? Why don't you resemble your brother? You have different fathers. No. You don't resemble your brother or your sister because of variation. So that you survive differently in this world. Yes. So the cells are different so that they can survive differently. It brings about different survival rates.
Yes.
Very clear?
Yes.
Lastly is this. Crossing over. How many pairs of chromosomes occur in the human cell? You see now, the question is linked to what? Human cell. How many pairs of chromosomes occur in the human cell? 23. You see? 23 pairs. But even if you say 23 is fine because they mentioned the word pair. You understand? Label X. What is X? Centromere. What is Y? Area Y? Chiasma. Then I say name the organ in a human female where meiosis occur. You see now? You're able to answer it. And they said female. So it means that you must be keen. They're saying name the process in diagram B. Crossing over. Then they're saying the phase represented in the diagram above. For phase 1, because it is crossing over is taking place. Ne? Yes. Type of cells that would result from meiosis of this cell.
Gametes. Cells.
Haploid cells?
Gametes.
Gametes. You understand? Yes. They're saying arrange the letters A, B, C to show the correct sequence of the events.
CBA.
It's gonna be C, B, A. Because here it shows that the variation has taken place already. Are we together? Yeah. So guys, the last question, this one is about karyotype, ne? What? Oh, why did I say gametes? Because meiosis results in the formation of gametes. The question is saying that the type of cell that would result from meiosis of this cell. And they already mentioned there that is a human cell. Yes. Guys, they're saying that what is A? What is A? This one. What is A? What is A? Autosomes. Homologous. Oscar. You want me to sing for you a song? Please don't judge me. I won't judge you. You are so. So which one are you? You are so. I didn't say that. You are. So I don't want to sing for you that song, ne? All right. Okay. It's fine. Yeah. Okay. Come back, guys, ne? Come back. How many? Label A, it is homologous chromosomes, ne? How many autosomes are present in the nucleus of this cell? Did I say the human cell? They say the karyotype below shows the chromosome of a person. Yeah. And then, guys, listen carefully. You can, I repeat, even let me stand here. You cannot just give your answers from the head. When they ask you this question, count them. Let's count them. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23, 24, 25, 26, 27, 28, 29, 30, 31, 32, 33, 34, 35, 36. 36. Ne? Where is gone? 37, 38, 39, 40, 41, 42, 43, 44, 45, 46, 47. 47. You said 40. Hey, they said autosomes. So it's what? Minus 2. 45. Yes, because this one there are 3. Wabona. Hey, so you don't just answer. Eh? You count because sometimes it might not reach there. I remember they brought a question whereby it was 15. Yes. And we're now, because we're used to 43, 45, 47. Buzo. Why do I count them as 1 non-homologous chromosomes? Now the question will come to you and ask you that how will you count this? So it's better to count them as 1 because there you can get even the prime number or the odd number. But if you count them as pairs, you only get even numbers. Wabon. Do you understand what I'm saying? All right. Then they're saying that, hey, I should not abandon the honorary kids. Homologous chromosomes. Yes, man. Yeah. Hey, dog is saying autosome pair. Yeah. Dog is saying autosome pair. Yeah, it's a homologous chromosome. Dog. Yeah. Dog. Why that dog? You have celebrities. You're a celebrity now, ne? You have fans here. Say hello to the dog.
Hello, dog.
Ay. Eh. La, la, la. Okay, guys. We are continuing. Shh. Name the type of mutation. Guys. In fact, come back. Do you see that question? That question properly? Let me zoom it. They're saying name the type of mutation represented in the diagram above. So because here, this mutation is the whole chromosome. You have an extra chromosome here. You can't call it point mutation. You can't call it gene mutation. So it is a chromosomal mutation. Is it clear? Dineo, are we together? Yes. Okay. All right. Yes. Is an error. Remember, mutation means an error. Hey. It's an error. Let me show you. They're saying describe the event that lead to Down syndrome. That's what I'm talking about here. The last question, the last point. So that we go for a short break. Definitely. All right, guys. All right, guys. Shh. Guys, we are saying that, remember, we are describing Down syndrome, ne? Oh, how does non-disjunction lead to Down syndrome? Guys, failure of the chromosome to separate during anaphase 1 or anaphase 2 is called non-disjunction. So you have chromosomes. Let me just use 2 chromosomes. Ne? These chromosomes are under what? Metaphase. Is it clear? So now what happens to them? What happens to them? They have to separate. You understand? This one, they have to separate. Meaning that this one has to move to this side. Then this one has to move to this side. You are born. And then now, this one also is supposed to do the same thing. Are you seeing? But because of non-disjunction, yes, this one doesn't separate. Therefore, what happens? The whole chromosome will move to one side. Are you seeing? The whole chromosome will move to one side, and then this side, there will be nothing. Are you seeing it? So this one will result in this cell. Yes, this cell will have one chromosome, then even plus these 2 chromosomes. You understand? While this one will have only one chromosome, only this one. Are you seeing it? So it means that you're supposed to have 2 chromosomes here, even here 2 chromosomes here. But now you have one, meaning that you are short of one here. You understand? And then here you have an extra. Is it clear? So if you have 4 here, you have 23 chromosomes from a normal sperm, and this is an ovum. When they fuse, yeah, meaning that this one is at 24 now because it has an extra. So 23 plus 24, you're gonna form 47. When this one fertilizes a normal ovum, it's gonna become 23, yes, plus now this one is gonna be 22. So it's gonna give me 45 instead of 46. So these conditions are called Down syndrome. But in our exam, we prefer using an extra instead of short of a chromosome. Is it clear? Yes. So that's what you are supposed to describe, or this is what they are talking about here. Bring me the characteristics of Down syndrome. Yeah. It may lead, check this non-disjunction, one nuclear contain 22, while others contain 24. When either of these resulting gametes fuse with the normal gamete, you understand, result in 45 or 47. This is what I've described. And then the chromosome, 47 chromosomes. Shh. Shut up, man. 46, meaning that this is what we are talking about. This may lead to Down syndrome. Why do we call it trisomy?
21.
Because it has 3 chromosomes instead of 2. Because tri means 3. Some means a body. It's a body. Some. Ribosome. You understand? Yes. So it's a trisomy. Is a body having 3. Is it clear? Yes. So guys, unless you have a question, we're gonna break. Yes. Ma'am, how long are we going? 10 minutes? I like it. I like it. I like it. Ma'am, it is supposed to be short because when they go there, the air is gonna go into them and then 15. So ma'am is saying 15. Okay, we are going for 20. Yeah. But you didn't I say, do you have a question? So guys, we are going for 20. We're gonna ask. Let me first. Hey guys, who are online, you are going for a short break for 20 minutes. Ne? Yeah. So we are coming back to genetics.
Yes. So,
Yes, guys, we are back. We are back. We are back. We are back. So let's start with the genetics. But yeah, yes, man, we can take over.
Okay. All right, guys. We are now online. All right. Welcome back. We want to start with the biological terms. We just want to start as a refresher with the biological terms. Can you see, guys?
yes.
Can you see? Right. So we're just going to do a few of these biological terms, then we move on to the content. Right. Let's start with the first question. Question 1.1. Give the correct biological term for each of the following description. Write only the term next to the question number. So we have 1.11. It's saying a genetic cross involving 2 characteristics at a time. So what we're going to do is I'm going to call your number so that we have order. Call your number, you say the biological term. Okay? The number that is in front of your computer. Right. 1.11. Let's focus. Let's focus. Let's focus. 1.11. A genetic cross involving 2 characteristics at a time. Right. I'm checking on computer number 29. Computer number 29. Those who are facing computer number 29, may I have the answer, please? A genetic cross involving 2 characteristics at a time. Right. Dihybrid cross. We agree, class? Right. Those who are online, you agree with us? All right. Okay. Let's move on. 1.12. Thank you for being quiet. Yes. 1.12. The exchange of genes between homologous chromosomes that bring about variation. The exchange of genes between homologous chromosomes that brings about variation. Computer number 26. Computer number 27. Computer Crossing over. Yes, it's crossing over. Right. Crossing over. So here we said that's okay. Dihybrid cross. Okay. Then here we have crossing over. Right. Okay. Crossing over. Not crossover. It's crossing over. Right. Let's move on to 1.13. All the genes in all the chromosomes of a particular species. What do we have? All the genes in all the chromosome of a particular species. Computer number 14. Computer number 14. No, nothing. Computer number 10. Nothing. Computer number 20. Okay, no volunteer. Okay, so we call this a gene pool because we're saying all the genes in, so it's the gene pool, right? Okay, right, gene pool. Let's just check. So when it's individual, it's called genome, okay? All right. I don't know what's going on with my pen. Okay, it's fine. Let's move on. Right. 1.14. An allele that is not shown expressed in the phenotype when found in the heterozygous condition. An allele that is not shown expressed in the phenotype when found in, right, recessive allele, okay? Right, recessive allele. I'm trying to write here. Not sure what's happening. Right. Okay, it's fine. Right. Then all genes, let's move on to 1.15. An allele that is always expressed in the phenotype. Yes, okay, I'm trying. There was a problem, an error here. All right. Gene pool. So when it's individual there, it's genome. Gene pool, yes. Because we're saying all. When it's all genes, it becomes what? But when it's individual, that's a genome. You need to understand this one. But when all genes, it's called a gene pool, right? Then we have an allele that is not shown. What did we say on 1.14? Recessive, right. Recessive allele. Recessive allele, right. Okay. Right, then let's move on. An allele that is always expressed in the phenotype. An allele that is always expressed in the phenotype. What do we have? 26. Please answer because you are busy there. 26. Okay, let's move on. Yes, dominant allele, right. Dominant allele. Right, that's a dominant allele. Don't worry about my handwriting there. Dominant allele, right. Okay, dominant allele, right. Then the position of a genome on the chromosome, the position, right. So what is the locus? What is the loci? Right. 1.17. Let's move on, be quick. The process by which genetically identical organisms are formed using biotechnology. Genetically identical. We have what? Cloning. Yes. Cloning here. Right. Then let's move on. Let me just write it here so that you can see. Right. That's it. Then 1.18, a segment of a chromosome that codes for a particular characteristic. What do we have there? A gene. This one is very simple. Wonderful. You're doing good. 1.19, type of inheritance where none of these two alleles dominate over the other. An intermediate phenotype is produced. Yes, the moment you see intermediate, we're saying there's something different that is produced, right? So that's we are saying incomplete, right? That's incomplete. That's incomplete, right? Incomplete. Let's move on to the next 1.10. An individual having two non-identical characteristics for an individual having two non-identical alleles for a characteristic. What do we have there? An individual having two non-identical alleles for a characteristic. What do we have? Computer number 10. Computer number 10. I need the answer. No answer. Computer number 18. Computer number. No one wants to answer. Okay, what do we have, class? Hetero, because it's non-identical, okay? So when it's identical, it's homo. When it's non-identical, it's hetero, right? So we have heterozygous here. Okay, we are done with this one. 1.11, alternative forms of a gene in the same position on the homologous chromosome. Alternative form of a gene, right? We have this one. The allele, right? The allele, right? Let's move on, class. The process of finding a desirable gene, isolating it, then moving it into the cell of another organism. What do we have? What do we have? The process of finding a desirable gene, isolating it, and then moving it into the cell of another organism. What do we have? The process, listen, the process of finding a desirable gene, isolating it, and then moving it into the cell. That's genetic engineering, into the cell, okay? Artificial, you are just selecting, you are not moving it into the cell, you're just selecting the preferable characteristics, okay? But now you are moving, like, for example, the production of insulin using bacteria. So it becomes what? Genetic, yes? What is the difference between, come again? Genetic? Right. Genetic engineering. There is a question who wants to answer. The difference between genetic engineering and genetic modification. What is the difference between genetic engineering and genetic modification? What is it, class? Right, genetic modification. Like, for example, we have the presence of GMOs and then genetic engineering. What is the difference? Can I hear from you? You're the one who writing, Mus. Yeah. Yeah. Let's hear from you, class. Let's participate. Genetic engineering and genetic modification.
Can I ask?
Yes.
Since genetic engineering, like inserting something, inserting genetic material, then genetic modification, you just telling with the genetical what of that particular thing. I don't know if you get my point.
I'm getting you.
But I'm not sure. Like genetic,
Right.
taking it from another species, and then genetic modification, you don't take it from another species, you just change, like, alter the. Yeah.
Okay. Now listen. Are you hearing what she's saying?
No.
Can you speak up?
Okay, I'm not sure, guys. So I think genetic engineering is like taking like a specific gene from another species and then putting it into another species to find what you want. And then genetic modification is like you're not taking it from another species, you're just changing the one that you have. Like,
You are modifying.
yeah, you're
Thank you.
altering.
Right. Thank you. From the word modification, you are modifying. You are modifying. Right. Right. You are modifying. Now, in this case, on genetic engineering, you are changing, you are taking the nucleus, like the DNA of a certain particular, like, for example, I talked about insulin, okay? Like the production of insulin using bacteria. Then on genetic modification, you're just modifying, you are changing to suit the certain environment that is there. Right, let's move on to the type of inheritance involving alleles that equally determine the phenotype of heterozygous. What do we have there? Right, the type, yes, thank you. The type of inheritance involving alleles that equally determine the phenotype. So in this case, we are saying if it's red and white, we are seeing red and white. Both, they are expressed equally, okay? Right. So we call it what? Codominance. Right. Codominance. Wonderful. Right. Then we move on. Organisms having two identical alleles at a given locus. Identical, what do we have? Identical, what do we have? Right. What is the difference between homologous and homozygous? What's the difference between homologous and homozygous? Yes, these are identical alleles. Remember, there's allele here. There's allele here. There's allele here. So it can't be homologous. It's homozygous. It's homozygous. Because we have
Yes, yeah.
yes, because here it's identical allele. So it can't be homologous. It's homozygous. Yes. It's homozygous. Thank you on that one, right? Now we move on to a genetic cross involving one characteristics. Monohybrid. Wonderful. Monohybrid. Right, that was a refresher. Now let's move on to, I want you to open your Saidi book. You have your Saidi book. Introduction there on genetics. We can't talk about genetics without talking about Gregor Mendel, the father of genetics. The father of genetics. And he came up with the three principles, right? He came up with the three principles. Yes, we still have, we are checking here, we have chromatids, chromosome. Let's move on. We want to move on to the three principles. Remember now we are focusing on content. All right, we want to move on to the three principles of Mendel's theory. Right. Mhm. The laws of genetics. We want to start with that, then we go on to check the questions that we have on the laws. Right, while I'm scrolling down, who can tell me the first one? The first law? The law of dominance. Yes, what does the law of dominance say? We have, okay, let's name them law of dominance. Then the second one, law of segregation. The third one, law of, yes, random assortment. And then, right, we have the third one. Right, let's start with explaining the first one. What does it say? The law of dominance. Do we have anyone who's find where we are having the three laws? If you are there, please shout out. It's not there. Okay, right. Let's go with the independent one. Okay. Here in our book, it's not there. Let's just quickly state them. Then we go back to what we need to do. Yes, dominance. Yes. Can you please come again? Louder. Louder, please. Louder, please. Yes. So what are we saying when we're talking about masking the recessive allele? What are we saying in terms of the phenotype? In terms of the phenotype, when they say masking the recessive allele, what exactly are we saying in terms of the phenotype? You said the dominant allele always mask the recessive allele. So I'm saying, what are we saying in terms of phenotype when you say masking? What is a recessive allele? Yes. Expressed on the phenotype. Okay. So, for example, you are saying, right, wonderful. For example, you are saying when we have tall, right, for example, here, let me look for a monohybrid that we can have. Monohybrid that we can have. For example, right, guys, let me look for the monohybrid that we have here. Then we check, right. Right. Yeah, I'm okay. Okay, right. For example, let's say we have tall. For example, here, an allele that is expressed in the phenotype when found in the heterozygous. So, for example, this condition T, we know that in genetics, the capital letter T means what? Dominant, right? The capital letter, whatever variable that you are given, the capital letter means what? Dominant, right? And then the small variable means what? Recessive. So we are saying now if we're having a heterozygous condition, for example, this one that we're having here, we have T, we have heterozygous T. So which means T, let's say T is tall, is going to dominate. Then we're going to see someone who is tall on the phenotype that we are going to see, right? Yes. Okay. You answered. Right. Let's move on to the second law, the law of segregation. The law of segregation. Anyone want to explain the law of segregation? Noctenda? The law of segregation. Hmm?
I can read it.
Yeah, you can explain, you can read whatever.
Okay.
Mm-hmm.
Each space is controlled by two vectors situated on homologous formula.
Mm-hmm.
Gametes formed through meiosis. The two vectors are separated or segregated. The gamete contains one of two vectors from each family. I read
Okay, right. Before we move on to segregation, let's just link to complete and incomplete dominance. What do you understand if you check the law of segregation in terms of complete and co-dominance, incomplete and co-dominance? What do you understand about the law of segregation? What do you understand about the law of segregation? What are we saying? What do you understand in terms of co-dominance and incomplete? No one wants to say something. Hmm? You are tired. Come on, guys. The exam is still coming. The exam is still coming. The exam is still coming, whether you are tired or not. The exam is still coming. Yes. I am saying from the law of segregation, can you link to, right? I'm going to give you two co-dominance and incomplete. What exactly is being said by the law of segregation? Can you tell us that this is the talking about complete or it's talking about incomplete? Yes. But what?
Incomplete or complete?
Yes, it's talking about what? Incomplete, right? Let's move on to incomplete. I want to talk about incomplete by explaining the law of segregation. Right. Incomplete dominance. What exactly is incomplete dominance? What exactly is incomplete dominance? What exactly is incomplete dominance if you are checking on Mendel's law of segregation? Mm-hmm.
incomplete. None of them.
Yes. We don't. let's use the word intermediate. Very good. Let's use the word intermediate. Yes, let's use the word intermediate. Right, let's use the word intermediate. So when we have incomplete dominance, so what can you say as a science learner that is writing? What I'm saying about the linking the Mendel's law of segregation and incomplete dominance? What exactly are you saying? Because here we have the diploid organism passes randomly selected allele for a trait to its offspring, such that the offspring receives one allele from each parent. So what are we saying there? If you are linking the law of segregation into incomplete dominance, what are you saying? What do you say? You don't know. Okay. Okay, let me check here. Right. Okay, let's move on because we don't have time. Right. These three laws, law of dominance, law of segregation, and law of independent assortment, okay? Right, we have the explanations that we have. But in other ways, if you check very well, here we're talking about diploid organism, definitely there is variation, okay? So that's why we are saying there is production of an intermediate characteristic or intermediate phenotype. Then if you check the law of independent assortment, in other ways, we are saying there's independent, which means each and every character is shown. Okay. So that's in other ways, we're explaining co-dominance. Right. Okay. Now let's move on. We have a diagram here, format of representing genetic cross. We have format of representing genetic cross, but I have questions that I want us to tackle. I here. I need page 66. Let me just quickly scroll down page 66 on this page. Right, quickly, I can punch it down. There's a question that I want us to check together. This is the document. I'm sure I'm opening the correct document. No, don't worry. I think I'm there. The questions. Right. Mhm. Right. Okay. Guys, let's move on to question 2. Let's move on to question 2. A boy's mother, can you please pay attention? A boy's mother has a patch of white hair called a white furlock, which is caused by a dominant allele H. The mother is heterozygous for this trait. His father does not have a white furlock. The symbol, yes? You can't see. You can't see. Oh, should I what? Zoom. Oh, this one. That plus. How many percent? Oh, I'm supposed to zoom, right? Let me see. Is it clear? Can you see now? Are you sure? Okay, thank you. Thank you. Right. Let's go on. A boy's mother had a patch of white at the back there. Can we see? People are sleeping. Right. A boy's mother had a patch of white hair called the white furlock, which is caused by a dominant allele H. The mother is heterozygous for this trait. His father does not have a white furlock. The symbol for the recessive allele is H. So which means H is recessive, right? Then represent a genetic cross to determine the possible genotype and phenotype of the children. Right. Yes. Okay, what do we do first when we are presenting a cross, genetic cross? What do we do first? First, we write the P1, right? Computer number 26. 20, that one. Right.
That means that the matrix is
Right. Okay. Let's start. What do we write first? P1, right?
Yes, yes.
P1. Right. So, what is P1 generation? What do we have first? What do we write first? P1 generation. What is supposed to be written here? Phenotype. Phenotype. So we have the boy's mother. We have also the what? The father, right? So the mother is heterozygous for this trait, and the father does not have. So what is the phenotype of the mother? The mother, the boy's mother had a patch of white hair called a white follicle, which is caused by a dominant allele. So which means this white patch is caused by a dominant, okay?
Yes.
It's caused by a gene that masks over, okay? Then the mother is heterozygous. So which means the mother is also carrying the recessive gene, right? Or recessive allele, okay. Right. So now phenotype, what do we say? The mother has or the mother doesn't? Yes, okay. So mother with mother. I hope you can see, because always my handwriting is clumsy. It's a doctor's handwriting, right? Yeah. Right. Then we have what? The father, right? Okay. Father does not have, okay? Right. So father, no. Right. After phenotype, we move on to what? Genotype, right? So because the mother is heterozygous, what do we have here? Yes. The father, what do we have? Small h and small h. Right. Now we are done with this one. Thank you. Let's move on. What is the next step after this? What is the next step? Because remember, if you just put meiosis, meiosis, right. Auntie, if you want to come here, you can come and teach. Thank you. You can come here and teach. And teach us, yeah? Right, yeah, very good. You can come here and teach us the whole night. Very good. Right. So we have meiosis. Then what do we have on meiosis? Gametes. Gametes. So we can put our gametes and put a circle, right? Right. Right. Then what else after the gametes? Where do I put a circle? I want to know. Should I circle them together or I circle one? Each here. Right. Now it's not clear. Let me just rub it. Let me just rub it here. Yeah, let me just. Okay. This, oh, the blank page. Ah, better. Thank you. Thank you. At least. Right. Yeah, thank you. Right. Okay, right. Oh, thank you, Sam. Okay, right. Let's move on. Okay, let me start again. We have the phenotype. Phenotype. I have P1 here. Phenotype. We are saying mother with white follicle. Then we have father No. What next? Genotype? Hmm? Small h. Right. We are done with this one. Next is meiosis, right? Then we have the gametes. We write them. Hmm. Right here. Hmm. Right. What next, class? We have our gametes there. What next? Fertilization. Right. After fertilization, we move on to what? Can you focus, please? Because I know that in exam you just put crosses. We have been marking your prelim. You just put crosses without. Can you focus? No tender. Please focus. Right. Fertilization. Oh, right. So now after fertilization, what do we put next? Right. You do the foil. I know you've been taught by the foil method or whatever, but just know that each gamete should go twice, okay? Whether I use the foil method or whatever method that you're going to use, just know that each gamete should go twice. Let's do together, right? The first one goes with the second one. What do we have there? H, right? And also it goes again. What do we have? Right. So you are done with the first gamete. Let's move on to the second recessive gamete. The second recessive gamete can go here, and then it goes with the first one. We have the homozygous here. Then also it goes again with the last one, right? Okay. Now we have the results here. What do we call this? It's F1 generation, right? Yes, F1 generation. And what is this? Is this a phenotype or the genotype? Genotype. Thank you. We are together there. Right, we have the genotype. So when we move on to the phenotype, what do we have here on the phenotype? What is this heterozygous? It's white. White. Recessive. One white followed, then there's two. There are two, right? Yes. Yes. Mm-hmm. So it's 50-50 because also white follow is 2, right? So what is the percentage here? What is the percentage? Mm-hmm. Right. So the ratio is 50% or 1:2? Right, 1:1. Right. So after this, you are getting your marks here. You are getting your marks. You are getting a mark here. You are getting a mark here for P1 and F1. You are getting a mark here for crossing. People think that the marks are on crossing. The marks are for naming. Examiners need to know if you know what is a P1. Examiner need to know if you know what is a genotype, the gametes fertilization, meiosis, you are marked here, and then 50:50 also the ratio. Right, we are done with this one. Let's move on to the next question. All right, let's move on to the next question. Right, Question three, we are still on the first one. We are still on the first topic. The topic of complete, right? Question three, it says a gray G male rabbit was mated with an albino female rabbit. The entire F1 generation was gray. Use the genetic cross to show the phenotype ratio of their offspring. If one of the males of the F1 generation was mated with an albino female. At the back there, are we all together on this question? That side? Are we focusing? Right, okay. Thank you, guys. Let's move on. So here, what exactly is the question saying? A gray male rabbit was mated with an albino G. So we have, the moment you see a dominant allele and a recessive allele, you know that the result that is coming is going to be what? Complete, right? Because we have one allele that is dominant over another. Right. So now, the question says, use a genetic cross to show the phenotypic ratio of their offspring. If one of the males of the F1 generation was mated, do we have to do it again? Or is the same as the first one? It's the same as the first one. Do we have anyone with a challenge? We are done with this one. Okay. So we are done with the topic of complete dominance. Do we have anyone with a challenge? No, right? Okay. Computer number 11, they have a challenge. Computer number 11, we have challenge. Are you sure? Right, let's hear the challenge. Let's hear the challenge before we move on to incomplete dominance. Let's hear the challenge before we move on to incomplete dominance. I'm waiting. Okay, no problem. So we are sorted, we are done. Now let's go on to incomplete dominance. Right. Computer number 28 and at the front there, you are not focusing. Thank you. Right. Let's move on to incomplete dominance. Right. Remember we said earlier on when we were doing biological terms, remember we said when we're talking about incomplete dominance, we are talking about where we are having a result, an intermediate, okay? Intermediate phenotype is produced, okay? So, when we are saying an intermediate, for example, we are doing a cross between a red flower and a white flower, okay? Right, what is produced is a pink flower. A pink flower is not red. Oscar, focus. A pink flower is not red. A pink flower, neither is it white, right? So which means we're having a different, an intermediate phenotype that is produced, okay? So, which means the genes are dominant. Hence, they produce an intermediate that's incomplete. Right. Let's check, for example, here we have the first P1 generation. We have red and white. Let me just quickly go through with you here. The alleles for red are dominant and the alleles for white is also dominant. So we have the red color here and the white color that is also dominant, okay? Right. So when we are saying when homozygous red crosses with homozygous white, using a genetic cross to determine the possible genotype and phenotype of the flowers produced, what do we have? We are going to have an intermediate offspring. Right, let's quickly go through here because I have a question that I have that I want us to tackle. On incomplete dominance. Right. Here, for example, we're given an example. We have the red and white there. We have homozygous R and homozygous W. Then what is produced here? Is pink, right? What is produced is pink. So which means, yes, are we together? We are here? Right. So what is produced is pink. As I said before, pink is not red, pink is not white. So when it's incomplete, we are saying we are producing a different, that's an intermediate, a different. I always give an example when we're class. I always give an example, though we are not talking about people. I always give an example of a colored, okay? Right, it's not white, it's not black, okay? Right, it's colored. Okay, right. Now, let's quickly just go on to codominance, then we'll tackle the questions once. Codominant. Codominance, most learners get confused with codominance and incomplete. Why? Because on the genotype, when I keep quiet, which means you are talking, guys, you need to focus, okay? Thank you. Right. Most learners get confused with codominance and incomplete. Why? Because on the genotype, we will be having the similar what? Alleles or the similar genes. Now, the difference is on the phenotype. The difference is on the phenotype. On codominance, we are saying both the characteristics are expressed equally. If it's red and white, we are going to have red patches and white patches. Are we together? If it's red and white, you're going to have red patches and white patches. Right. Okay. So I'm going to go on. Do we have a question before we move on to the questions? Anyone with a question? No question. Right. Okay. If we don't have a question, let me move on to the questions that we have. Right. Question 5, we'll come back to it also. We need to focus on this one. We come back to it. For dihybrid. They said this one. There's a lot. Okay. About how many percent? 74 is fine. 135. 135. Hmm? Wait. This question that yeah, this one. Okay, guys. Let's move on to question 16. Let's move on to question 16, all of us. Let's move on to question 16. There. Right. In plant species, 2 characteristics of flower color and plant height were studied. Each of these characteristics has 2 variations. Flowers may be red or white in color, and the plants may be tall or short. Plants that are heterozygous for flower color had red flowers, and plants that are homozygous for plant height are short. The allele for each characteristic are shown in the table. Right, we have the characteristic there. We have the dominant allele. We have the recessive allele. Flower color, F. Recessive allele, F. Plant height, H. Recessive allele, H. Right. What is the term given for a genetic cross involving two characteristic dihybrid. This one we have mastered it, right? Okay. The next question. Give the dominant phenotype for flower color. Give the dominant phenotype. Take note of phenotype. Dominant phenotype. Okay. Can I repeat again? In a plant species, two characteristic flower color and plant height were studied. Each of these characteristics has two variations. Flowers may be red or white in color, and the plants may be tall or short. Plants that are heterozygous for flower color have red flower, okay? Plants that are, I'm repeating again so that I'm going to write something here. Plants that are heterozygous for flower color have red flowers. So which genotype are we having if we have a plant that is red but heterozygous? When we have a plant that is red but heterozygous, plants that are heterozygous for flower color have red flowers, and plants that are homozygous recessive for plant height are short. So here, what do we have as heterozygous red flower? Do we have heterozygous F, or what do we have? Is this correct? Class, I'm talking to you. Is this correct? Right. Okay. Now let's move on. You go on to say the dominant phenotype for flower color. What is it? What is the dominant for phenotype? At the back there, can I have the answer, please? Hello, guys. What is the answer? What is the answer? No answer. Here, what is the phenotype? No answer. Here. Red. Right. Then recessive phenotype for plant. Recessive phenotype for plant. Height. Recessive phenotype for plant height. I'll go with you again. Plants that are homozygous recessive for plant height are short. Then recessive phenotype for plant height is what? Short. We are together on this one. Then we move on to C. The phenotype of a plant that is heterozygous for flower color and homozygous dominant for plant height. The phenotype that is heterozygous for flower color definitely is red, right? I'm now talking to myself, class. You're not focusing. So simple. Okay, so let me have the answers and then we quickly move on. Let me have the answers quickly. Very good. If it's simple, let me have the answers. Then we move on. If it's so simple, let me have the answers. Yeah, let me have the answers, guys. It's so simple. Let me have the answers,
Let's go because
yeah, because if it's so simple, let's have the answer and move on to the difficult ones. Because you are saying it's so simple, but you're not giving me the answers. But see, right, phenotype of the plant that is heterozygous for flower, red and tall. Yes. Okay, right. The genotype of a white flowering short plant. The genotype of a white flowering short plant. White flowering is recessive, right? Short plant is also recessive, right? So what do we have? Correct, right? Let's move on. 1.63. State Mendel's law of dominance. We did this one, right? Can we just quickly go through it? What does it say? Law of dominance. I had an answer there. Who was speaking? Can I have a name? Kopeka. Yeah, Kopeka. Can I have the answer? Yes. Yes. No one wants to give me the law of dominance. Yes. When two individuals. Okay. So take it slowly. Remember, I'm writing. I need to hear from you. So we are saying when two individuals all right. When two individuals come again with contrasting pure breeding, when two individuals with contrasting pure breeding, yes, characteristics, across, when two individuals with contrasting pure breeding characteristics across, what happens? They the individuals will display okay, the characteristics of their dominant right. Correct. What did I write? Come again. Okay. Thank you. Sorry. Yeah. There you go. Right. Thank you. Right. Okay. Right. We are done with this one. I need us to move on to dihybrid. There's a question for dihybrid that I want us to focus on. I'm scrolling up. There's a question of dihybrid that I want us to focus on. Another question. Let's just quickly go on to dihybrid and we finish once. Then we'll move on to pedigree and sex-linked. Right. There's a question on dihybrid here. This one. Question 5. Right. Do we have some people who are sleeping there? Oh. Okay. Please don't sleep. My voice is audible. Okay. Let's move on to question 5. We want to see if we can answer this one. Mendel observed some characteristics of the pea plant, the Pisum sativa, which he suggested were controlled by inherited factors. He conducted a series of experiments in which he crossed the pea plants with contrasting phenotypes to obtain the offspring of F1 generation. At first, his crosses were simple and involved only one pair of characteristics. Mendel counted the number of offspring showing each of the variation. His results are shown in the table below. So we have the seed there. We have the seed there. We have the pod. We have the flowers. We have the stem. The characteristics that we have seed texture, seed color, pod texture, pod color, flower color, location of the flower on stem, height of stem, P1 generation, round and wrinkled, yellow and green, full and constricted, green and yellow, violet and white, axial and terminal, tall and short. The filial generation, all round, all yellow, all full, all green, all violet, all axial, all tall. Right, let's move on to the question. Okay, give the term for the inherited factors that Mendel referred to. Give the term for the inherited factors. What are we talking about? Give the term for the inherited factors. When they say inherited factors, automatically you know it's an allele, right? Because remember here, the difference now between the gene and the allele, here we have a variation because we have seed color, we have seed texture, okay? So it becomes what? Allele. Right, anyone with a different answer? No. Right, give the term inherited factors that Mendel referred to. You don't understand? Okay. Right. If you are saying you don't understand, we are saying what carries the characteristic? This is what the question is saying. What carries the characteristics? So that's why I say inherited factors. Where do we find those characteristics? On allele, okay? Because now we could have said gene, but because now we have variation. Remember, an allele shows the variation, okay? The variance or the variant gene. So now we are saying our answer becomes allele. What carries these factors? What carries those characteristics? That's the answer there. Right. Okay. Then this one is a repetition. A crossing involves only one characteristic. Yes. Yes. A characteristic, a cross involving one characteristic. Monohybrid. Thank you. Someone said monocotic. Are we now doing dicots and monocots? Right. Name the female structure of the flower. Can you focus, please? Name the female structure of the flower where meiosis occurs. It occurs in the sex cell, right? So, name the female. It becomes what? Ovule. It's an ovule. It's the flower. It's the flower.
If it's over, I don't know. Then it becomes an ovule after meiosis.
Listen to the question. Female structure of flower. Guys, check the question. Check the question. Flower. Not human. Not human.
Ovaries.
Not human. Not a woman. Not human. Check the question. Flower. Did you see the question? Yes. It becomes the ovule. The sex cells. Listen. Okay, listen. Ovum is for the humans. Okay? Yes. So the question is saying flower. Flower.
What is it, ma'am?
It's ovule for the flower. Yes. This.
It becomes an ovule. That's actually
Mm-hmm.
that is over.
Over. Hmm?
The show one. Then that's an ovule.
We have two answers. We have ovule. We can say pistil race. But the answer here they becomes ovule. Remember, it's the flower, not humans. It's the flower. It's the flower, not humans. Yeah. If it's the human, yes, you can say that. Okay. Yes. What did you say? Where meiosis occurs, name the female structure of the flower. It's the ovule. I can take pistil. I'm saying this because there's this question. This question once came, is it November 2018, paper 2? If you check very well. November 2018, paper 2. November 2018, paper 2, if you check very well. It preferred pistil and ovule. Paper 2, 2018. Yes, pistil and ovule. Ovary. They wrote ovary there. But the question paper, okay, we can now take ovary. But what I'm saying is we are focusing on the plants. Pay attention. We are focusing on the plants, and we are also focusing on what the examiners were. 2018, check paper 2, 2018, if I'm not mistaken. Check this. Right. Okay. So let's move on to the next one. Use the information in the table above to give the number of each of the following. 5.3. The characteristics of pods, the number. How many are there? Characteristics of pods there? Number. What do we have? We have pod texture and pod color. So we end up having what? 2. Then let's move on to B. The allows for seed characteristics. Remember earlier we are talking about a variant gene. So we are counting here. Let's count together. 1, 2, 3, 4. Are we together? Yes, 4. So we are having 4, right? Okay. Now let's move on. Give the characteristics that is dominant for flower color. Let's go here. Give the characteristic that is dominant for flower color here. What do we have here? What do we have, guys? Yellow. Okay. So we have our answer there becomes yellow, guys. Right. Then we move on to B. Recessive for stem height. Recessive for stem height. Let's move on to height here. Recessive for stem height. What do we have? Short. Right. Then let's move on to the next 5.15. If the individuals of F1 generation are crossed, how many phenotypes of seed color would be expected in F2 generation? If the individuals of F1 generation are crossed, how many phenotypes for seed color would be expected in F2 generation? Right, let me have the answer. If the individuals of F1 generation, this is here. Then we move on, that's our F1 here. If the individuals of F1 generation are crossed, how many phenotype for seed color will be expected in F2 generation? Right, seed color here. So, what do we expect? How many colors will we expect, guys? Why are you saying 1? Why are you saying 1 here? Why are you saying 1? F2 generation. Why are you saying 1? Because remember here, this is the product of yellow and green, right? We go back to complete dominance, right? This is the phenotype of all yellow. This is what we see. Which means the green one is what? Mass, right? Can we have some people here? Right. Let's start. Let's do together. Yellow. Right. Phenotype. Right. Yellow. Let's start with P1 and then green, right? Right. On yellow, what do we have? Okay. Just a quick one. Let's see what is happening. Right. Please focus. Please focus. Just going to take one. Yes. Okay, guys. Let's go on to this one. Just what I wanted. Can you please focus? Right. So, all yellow, right? I just want us to focus on the color. All yellow. So, which means this is the genotype of all yellow, okay? This is the genotype of all yellow. Are we together? Right. Now, the question says, just to check, if the individuals of F1 generation are crossed, how many phenotype for seed color? So which means this genotype, I'm not going to write everything, meiosis, whatever. I just want to show you something. So we're saying if the individual, right, of F1 are crossed, this is what you are going to cross, right? This is what we are going to cross. Because remember we said P1 generation is this one, yellow and green. Yellow and green. To give us F1, these are the genotypes that we are going to have, right? Then we are saying this F1 generation, I'm doing a shortcut. F1 generation is crossed. Because here, remember, we have homozygous and homozygous. So definitely, we are going to have 100% of this. So if F1 generation is crossed, let's just quickly go and see what we have. Right. We are just doing a recap. Right. What are we going to have here? Yellow, right? Yes. Just for you to see. Okay. So what do we have here as the phenotype? Yellow, right? Yellow. And here we have also what? Yellow. Then here what do we have? And then here what do we have? Green, right? Now the question goes back to say, if the individuals of F1 are crossed, how many phenotypes of seed color will we have? F2 generation is what? Can I have the answer now? 2. Simple. So this one was just to work to show out how we are getting the answer. Right. Okay. Right. Let's move on to the pedigree. Yes. Not sure. All right. Yeah.
Hey guys, ne? Let's finish up the pedigree. Let's finish up the pedigree. Let's finish up the pedigree. And there is no hiding. Yeah. I need to see your face. Even let me remove so that I see you clear. In the face, yes.
Thank you.
How many generations are there? How many? Thank you so much, man. How many generations? Name. Name. The time. How many generations are there? 3. 1, 2, 3. Done. Eh? Uh-huh. Who is Linda? Who is Linda? Who is Linda? Male or female? Male or female? Affected or not unaffected? Affected. So guys, listen carefully. You need to understand this. If you want to get the marks regarding to pedigree and some bit of genetic cross, shh. Listen carefully. All right. You need to identify whether is caused by a dominant allele or is caused by a recessive. Please. Yes. You want to ask something? Yeah. So is it caused by a dominant allele or recessive allele? If you come back to our question, who is it? Was this caused by dominant allele or recessive allele? They are saying caused by recessive allele. Wabona. So, who is poor? Is it homozygous or heterozygous? Homozygous. Heterozygous. Why heterozygous? Because they produced a kid who is what? A kid who is what? Someone who is affected, isn't it? Meaning that this one, Gabi or Gabi, whatever, I don't care. Yes, is capital H, small h. Even this one is capital H, small h. So that these two can come together and produce this baby, isn't it? So, so you need to know that. Is it clear? Let's go to this one and then find out. The pedigree diagram below traces the inheritance of hemophilia in the family, isn't it? You are having it. What is happening here? They said unaffected female, affected male. Sometimes they don't give you these boxes. We say that if it's a box, is a male. If it's a circle, is a what? Is a female, isn't it? So how many generation? 1, 2, 3. How many kids are for 1 and 2? How many kids are for 1 and 2? 2. Which one and which one? 4 and 5. Now who is 6? In-law. Meaning that okay, 6 married, 5. Ne? Yes. So now I think we can see it, and then because of that, we can know. Let's find out, is it caused by a dominant allele? Hemophilia is always caused by a recessive allele, isn't it? So now, once you have a baby who is affected, it means that the parents are always heterozygous. Is it clear? Yes. So that one is sorted. Now, what about, it's here. Use the possible allele X and determine the genotype of the following individual. Individual 1, where is individual 1? So what is individual 1? Look, do you know the meaning of this? Do you see it? Do you know the meaning of this? It means that it's sex linked, but they didn't mention it in the paper, in the question. It must be mentioned there that is sex linked or is not sex linked. If they don't mention it, they give you a clue which show that it's a sex linked. Guys, if they don't mention that it's a sex linked, do not bother yourself. Even if they say Karabo married Kabelo, whatever. Kubega. Yeah, Karabo married Kubega, whatever. Even if they say that, Seroma married whatever. Even if they say that, Seroma, please do not use X and Y unless they say that is sex what? Alright. So we continue. So because of this, we are able to know that now, where is it? We are able to know that this one is what? Is it? So this is capital letter H, small letter h, and then this is, because this is normal, is only capital letter H, isn't it? So what does it mean? It means that because this is a male, the Y came from there, and then the small x came from there, and made this individual to be what? To be sick or to be affected. Guys, please do not use the word affected just like that. They will ask you affected with what? HIV. So you say affected with hemophilia, or you say hemophilia. Wabon?
Yes.
Hey. No. Are we together? Huh? Alright, we go to the next question. Let me look for, if I can find it, I will be happy. Look at that question. It's caused by what? Recessive allele. Isn't it? Let me check if I can find a dominant allele anyway. People are what? Who cares about people are laughing? Don't care, man. Don't care, man. You know what is gonna happen? 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 Let me give you another question here. You know what is gonna happen? I know you like the gossip. Okay, let me use this one. Yeah, let me use this one. Let me show you this one. Yeah, here is a question. Senzo, where are you? Senzo is not here. Even Senzo, this one is not Senzo I want. Okay, let's find out here. How many grandsons are James and Teboho? How many grandsons? How many grandsons do James and Teboho have? How many? James is here. Teboho is here. Grandsons. How many are they? 3. How many? 2. Which one and which one? This one and this one. This one and this one. Yeah, grandsons means second generation, isn't it? Ah. What is Grace's phenotype? Shh. Shut up, man. Nagato. Huh? What is Grace's genotype? What is Grace's phenotype? Where is Grace? Is affected. Affected what? Let's find out. The pedigree diagram below shows the inheritance of albinism, meaning that Grace is what? An albino. Hey, you understand? Is Anna what? Albino. They're saying that. So when you say affected, affected with what? Albinism. Then you go to shh, Oscar. Pastor. They're saying Johnny's genotype. Where is Johnny's genotype? John. Johnny, yes, Papa. Eating sugar. Telling me lies. Open your mouth. You ain't open the mouth, man. Yeah. So Johnny's okay. Johnny's genotype. What is Johnny's genotype? Did they give us the letters to use? No. They didn't give us the letter, ne? Meaning that if they're saying Johnny's genotype, John gave a baby who is sick. Meaning that if albinism is indicated by maybe small a, you understand? Meaning that John had a small letter and also Johanna had what? Small letter. Meaning that, they gave it here. You understand? So it means that Johnny's genotype is capital A, small a, because they gave a baby who is albino. Not infected. Albino. You understand? Yeah. Please, don't use that word. When are you fighting for others? How can you do that? People online, they are watching you. Okay, we go to another one. John and Johanna wish to have their children. What is the percentage chance of this child that the child will be a girl? Guys, whenever they ask you percentage chance of having a girl or a boy in the next phase is 50. Even if that couple had girls only, they can say that they have girls, 3 of them, and they ask you what is the percentage chance of having another girl. Still is gonna be what? 50. You understand? Hey. You have 50% of having a boy or a girl. So let me ask you a question. Do you think that you being a boy is because your father and your mother had a girl before? No. Do you understand what I'm saying? You being a girl or a boy is not connected to the kids your fathers and mothers had before. Your chances were your chances. You understand? Hey. Meaning that your sperm swim very fast, fit the egg, scored the goal. I don't want to talk about it. They ask you a question there in paper one that which part of the sperm has the enzyme. Yeah, people don't remember it. People wrote eggs there.
So what's this?
Acrozo. Someone said, I didn't see it. That was paper one. Please don't bring it here. Can we continue the next question? What is the percentage chance of the child to be to have an albino? John and Johanna. Yes. Here you have to cross. and then you find out. You understand? Meaning that you have to do a genetic cross, a simple cross, and then you find out. Meaning that if they are like this and this, then this is going to be 25, isn't it? Because if you have A, small a, wait, and then A, small a, you'll find out that this is going to be like this. Okay. And then this, you see, that the chance of this person having an albino is going to be 1 over 4 times 100, which gives me 25%. Is it clear? Guys, let me tell you something. Let me tell you something. Listen carefully. Sharon, listen carefully. Oh, Sharon, I'm still on you. Aha, stand up. Stand up, Sharon. Why now are you laughing? Stand up. Virakazi, stand up. What? Virakazi, stand up. Why are you laughing? Stand up. Happy birthday. Happy birthday. Happy birthday. When I want. I shall not be able to get it. Sorry. Okay, guys. Shh. 2 minutes. Let me finish. Let me wind up with this topic. Yeah? Eh? I don't worry about it when you come back. Alright, guys. Shh. Question. How many genes control blood group? Guys, don't give me wrong answers because I've been saying this over and over and over and over again. How many genes control blood group? 1. Which genes are those? Blood group. How many alleles control blood group? Which one are those? Allele for A, allele for B, and allele for? And allele for? If I have blood group A, ne, I marry a person of blood group A, can I produce a kid of blood group O?
Yes.
You again. Guys, don't do that to me. So guys, it depends on the situation of these blood groups. Because I can have blood group A, homozygous, or blood group A, heterozygous. So if I'm a blood group A, heterozygous, and also the wife is blood group A, heterozygous, the 2 recessive alleles can meet and produce blood group O. So don't just give a conclusion, you have to first identify. Is it clear? Yes. So, then the next question. Which of the blood group A or B, which of the blood group is regarded as a universal donor? Blood group O. Which of the blood group is regarded as a universal recipient? It means that it can receive from any blood group. Not O. O can donate, but it cannot receive from any blood group. And if you have a blood group O, I'm sorry. I'm telling you, Some of you think that, yeah, if you are blood group O, you are strong. Who told you?
So what is
Huh? All blood groups are the same. If you have blood group O, you get an accident, they will move hospital to hospital without your blood group. Sorry. Okay, blood group which is regarded as universal recipient is called blood group AB. It can receive from any blood group. Yes, this is what I am trying to mean, but I think, yeah. Then we continue the next question. What is okay? Tell me what is stem cells? What are stem cells? What are stem cells, guys? Wisdom, what's a stem cell? What are stem cells?
cells with unique
Cells with unique ability to develop into specialized cells in the body. From the book. And from your head.
cells which is specialized.
Okay, cells which have ability to develop into other cells. You understand? Yes. So it means that, look, I'm gonna give you a simple example. When there is no okay, when a man donates a sperm to a lady, Bineo, wabona, the sperm fuses to form a zygote. You think when you form a zygote, you form a ball of a cell which you call the morula, whatever. They asked you that question.
Yeah.
Do you think when you form the ball of cell, do you see where the head is?
No.
Do you see where the legs are? So those are stem cells. They have not yet specialized. But now, if you check your face cannot become the ass. Your legs cannot become hands. Do you understand? Your ears cannot become the mouth. Do you understand? Because they are already specialized cells.
I don't want to change the other things.
I don't know about that. I don't want to enter there. You understand? Just use good examples we see. So now it means that they have become specialized. You cannot use your hands to see because the hands are not specialized for seeing. It is the eyes which are specialized for what? However, some of you at night, when you wake up, no electricity, use your hands to see. You keep when the eyes are closed. You don't want to open the eyes because the sleep is too much. All right. We got the next question. What are some of the examples of stem cells? Stem cells. Hey, examples of stem cells. You don't know. You will know today.
Good barrel.
Hmm? Umbilical vein. Bone marrow. Umbilical vein or umbilical cord. Oh, cord blood. Okay. What are the advantages of what are Oscar? Give me the advantages of stem cells.
You can use it like to research. So can research. Maybe you can use this also like research like this. For development in let's see so for like no.
If you have a hole here, what do we do? A hole here. We can use the stem cells, ne? Huh? Yes. So we can use stem cells. So basically, stem cell can be used to replace. Yes. Also for research purposes, you understand? So we can use what? Stem cells. Dineo, is it clear? Yes. Peter, are you there? Okay. Samantha, are you there? All right. We continue. So we saw this one, we saw the stem cells. Now tell me the advantages of genetic engineering. Advantages of genetic engineering. Biotechnology. You can't tell me that you don't know the advantages of this biotechnology when every day in ShopRite you are buying things from here. ShopRite. Genetically modified. Producers required. Okay, you know what you need to do, ne? You just look at ShopRite. You understand? You see the chicken you eat. How many are coming from Limpopo? You know Limpopo have the regular, ne? Not the ShopRite life, ne? Yes. You see when you got, I'll say Limpopo because Limpopo, I know Limpopo too much. Ne? I'm a Solomon from Limpopo. You don't say. I'm a Zuluman from Limpopo who speaks Tswana and okay. You see when you're in Limpopo, ne? You have chicken. What you do, you just open for them in the morning, they go, they eat, in the evening they come back. Isn't it? You understand? But I want to ask you a question. How long do they take for them to grow? One chicken can take a full year. I'm telling you. In Limpopo, one chicken.
I don't know.
But look. Look. Shh. Do you think, guys, do you think the chicken you eat from those Pick and Pay, ShopRite, Checkers and what, they also take 2 years? 1 year. Maximum is 3 months. 3 is ready to be eaten. What causes that? GMO, biotechnology. You understand? This food, it has been genetically engineered so that you can have enough food. So it increases food security. I see. Yeah. But does God allow that?
What?
Does God allow that? No. So it interferes with the nature as a disadvantage.
Not written in the Bible.
It's not written in the Bible, but that's it. Whether it's there or not, that's it. Huh? All right. So simple. Hey, when you are not writing it. So if you see, we are saying that some of the advantages produce is very important. Production of medicine, pest control using this food increases food or crop yield. You understand? Crop yield means crop production. Are we together? Yes. Shh. Also, shh, increases the shelf life. What is shelf life? Inile. What is shelf life? Anguzwa, when you are busy talking, that's why I'm asking you. What is shelf life? Wisdom. 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 What is the opposite for wisdom? If you don't give me the answer, I'm going to call you the opposite for wisdom. I'm telling you. What's the opposite for wisdom? He doesn't know. You are like this, you always say that the opposite for pure is elite. You are mates, must The opposite for pure is what? All right, we continue to the next question. Shh. Shelf life means that they can stay in the shelf for long. You see these some tomatoes? Yes? Those ones which have this shape. Those tomatoes which look like that. Ne? They can stay for longer than these tomatoes which are like that. Now, it means that this one, they have a high shelf life. You understand? Yes. So, we can use biotechnology to increase all that. However, some of the disadvantages is expensive. This process is expensive to do. Number two, it interferes with nature. Hey, you find an orange as big as your head. Hey, you don't know them. Yeah. You see, even you're saying, how? But those oranges are there. It interferes with the nature. We don't know some of the health impacts. Is it clear? Then another thing is, we don't know the long-term effects. All those are disadvantages. Lastly, biotechnology. Genetic engineering. How do I know that this is genetic engineering? Sorry? They are there. How do I know that this is genetic engineering?
Expensive.
Yeah? How do I know that this is genetic engineering? They must talk about manipulation of genes. If they don't talk about, remember, engineering is manipulation. You understand? Yes. So you must talk about that. Lastly, on this, let's talk about this cloning. The cloning process. We can use this or we can use this. Okay, I'm gonna just talk about this, and then I fire you some questions on this also. Eh? Fire. Okay. Shh. 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 Can you look now? Onele. 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 Minat. All right. What is happening here? Production of insulin used by technology. Ne? So what happens? You get a bacteria. Is it clear? That bacteria has what we call a plasmid. A plus what? A plasmid. You remove this plasmid when you.
How did you get to bacteria?
Yes? How did you get? Yeah, we are in biotechnology. Yes. You remove that plasmid. Once you remove the plasmid, we use the restriction enzymes and we cut it. Sorry? Nakutanda. As long as I end with it, tender is correct. Yeah? Yeah? As long as I end with what? Tender. Yeah, so don't complain, eh? Yes, it's the same name. All right. So what we do? We use restriction enzymes, we cut it, then we want to produce insulin. Ne? Before, we used to use the pig. We get the liver, we get the what? The pig, we get the pancreas from the pig, we extract the insulin from there. You understand? Yeah. We extract the insulin from there. That's why I tell you that if you eat pork, you're eating brothers and sisters. Because yes, you almost have this. Yeah, you have almost the same system. Yeah. So what we do? But now we can manufacture it. So we get the DNA of this insulin, we insert it here. After that, we put this plasmid, we put it back, and then we trigger this bacteria to multiply. As it multiplies, it also multiplies with the what? This gene we have introduced in. After that, we remove them and then we make the medicine. Simple.
So what is a plus?
Plasmid. Plasmid. Plasmid is a plasmid. It's a structure which has which it contains DNA. You understand? Yes, in a bacteria. So we can remove it. Yes, we can remove it. We insert the DNA. We trigger the bacteria to multiply. Once it multiplies, it also multiplies with that DNA we have put in. Then after that, we extract the DNA from there, and then now we make the medicine. Okay, they are saying it's this thing. Hello, sir. Yes, sir. I was saying let's finish up. Let's finish up so that I can give you a short break, a long break, a long break, a long break. All right. So you have to listen. Shh, shh. Okay. Dolly sheep. Who is Dolly? Dolly Patra. You call it Dolly what? Whatever. That one from where? Yeah. Yeah. They name. All right. So what happens here, guys? How they can ask you questions about this? Guys, listen carefully. Shh. You are taking now your time for break. Okay. You have the sheep A. Sheep A. You understand? You have sheep 2. Are we together? So, yes, it's a sheep. So we want to make sheep, this sheep. Is it clear? So in sheep 1, what we do, we get a cell, a body cell. Ne? A body what? Cell. Remember, body cell has diploid number of chromosomes, 2n, isn't it? Then in sheep 2, we get a gamete. A gamete. Remember, a gamete has single set of chromosome, isn't it? Sharon. Isn't it?
Yes.
So what we do, we remove. We don't want this. Do you want this nucleus? Yes. We remove this nucleus and we maintain it. We throw this away. Are you seeing?
Yes.
Noctenda? Yeah. Then when we get this gamete, we don't want the nucleus because it has one end. We don't want its genetics. We only want the casing. So we remove the nucleus, we throw it away, we maintain the casing. I see. So the reason why I maintain the casing is because when I bring this nucleus, which is 2N, and I put it in the casing, you understand? The casing of the ovum, so it will look like a zygote. You understand? I always ask you a question that if you drink alcohol in the cup, no one will know that you're drinking alcohol. They will think you're drinking what? Tea. Because the casing is for the tea. You understand? But if you put tea in Johnny Walker's bottle, we shall say that you're drinking Johnny Walker. Isn't it? So, what does it mean? The casing also matters. You understand? Yes. So, that's why we put this in the casing of the ovum so that it looks like a zygote. But we don't just put, we use what we call mild electric shock, because they'll ask you which method, if they bring this, which method do they use? By the way, they have spent time without bring this question.
They won't bring.
You might be lucky and they bring it. Eh? Which method did you use to combine the nucleus with the shell? Yes. And then now, what we do, we put this zygote or this resulting cell, and then we make it to develop by mitosis. Mitosis. You understand? Yes. Yeah? It's not a zygote, really. Yeah, it's not a zygote. We can say resulting cell, whatever. But it looks to be a zygote because it is 2N. You understand? Yes. So now what we do, we put this embryo. We can cut these embryos into different sheep. And that sheep, because the DNA is the same, it will develop the same kind of what? Oscar. It will develop the same kind of Oscar. Oscar, don't deny. Oscar, don't deny. Oscar.
What did I?
You don't know what you did, you know? Is it clear? So now they ask you, why do they keep the nucleus of sheep 1? Give me the answer. Give me the answer. Why do they keep the nucleus of sheep 1? Yes. They wanted the characteristics of what? Sheep 1. Why do they throw away the nucleus of sheep 1? They did not want the characteristics. Which method did they use to combine the nucleus of sheep 1 with the shell of sheep 2? Which sheep between 1 and 2 will the resultant sheep resemble?
What?
They will ask you, give a reason for your answer. Remember, when they bring the question, they don't label everything you see here. You understand? You have to label. You understand? Yes. Give a reason for your answer. Why will sheep 1 resemble, the resultant sheep resemble sheep 1? Because they use the nucleus of what? Sheep 1. Karapita, are we together? Karapita, you're swapping. Karapita. Yeah, I swapping. Yeah, I was searching you everywhere. Then they can ask you another question. Look, guys, they can ask you another question. The question is gone. The question is gone. So, guys, ma'am, how long should we give them? A short break? A moderate break? Shh.
Exactly.
Eh,
Zero.
huh? Not many hours.
Hours. What do you have? One. It's fun.
Of life, whereby this topic, what you're supposed to look at is DNA structure. Describe the structure of DNA. That's, I go with the nucleus, and we say, our question is, I think I've exhausted that. Pedigree, let's just look at one question here which involves the pedigree, and then we can go to our, oh, there is one question here for blood group. There's one question here for blood group, but I think we exhausted the blood group. But you see that blood group also has 5 marks. Explain why paternity of the boy could not be established using the blood group. Why is paternity test is not able to be established, like to conclude that this is the father of the individual? It's because many people have the same type of blood group. So you can't say that, as I showed you, that is blood group A married blood group B, and then produce blood group O, still they produce blood group AB, still they produce blood group B. So whom are you going to say that is not the father? Who is, or whom are you going to say that is the father? So it means that you end up losing the what? The mark. All right. Why is it not conclusive? Why is it not conclusive? Because many people have the same type of blood group. But if they ask you, explain why paternity test for this situation, you will say that The mother is blood group O. You see, the mother is blood group O, while the boy is blood group AB, blood group B. So of which the boy can be can be B, heterozygous, or the boy can't be homozygous. Why? Because the mother is II. So gets one from the mother. So the genotype of the boy is heterozygous. Now, this one can give yes the B. Ne? And also this one can give the B. So whom are you going to say is the father? Whom are you going to say is not the father? So that's why in this case, blood group can only help you to eliminate those possibilities which cannot happen. For example, a person of blood group, the father can't be blood group A because this person is blood group B. You see? So if it's blood group A, there is no way how the boy is going to have blood group B. So if there is a man, another man who is blood group B, so blood group A, that one is wiped out. You understand? And then another man who is blood group O cannot be. So blood group A and blood group O, they are wiped out. Why? Because if it is small I, the boy is blood group B. So there is no way how this boy is going to get a B from the father who is blood group O, yet the mother is already known is blood group O. You see? So that's the meaning that you can't use blood group to conclude the father of the individual. Because you are blood group B, you can't say that this is my child because this child of mine is blood group B. There are so many people who have the same type of blood group, and the possibilities of having these are many. I hope you understand me better. Now, how do you write this? It means that this one is only blood group A and blood group O are going to only be wiped out. So the blood group A and blood group O, they cannot be the father. So you get those 2 marks already. And then this blood group, the male A can give this allele. So it can be the father. Or this one also can also give one allele for B and gets another one from here. Also can be the what? Can be the father. That's why we cannot conclude using blood group who is the father of this boy. As I repeat that, blood group can only be used to eliminate the possibilities which cannot happen in a situation. Who is the biological father of the boy using the DNA profile? Now you have to check and see. You will see that it is this and this. You will see that is male what? Male 2. Give a reason is because the remaining bands of DNA profile match with the male. The remaining, they must match. If there is one which is not matching, then automatically it's not the father. Might be a relative, but might not be the what? The father. Remember, all the genes that this boy or this baby is having is mother and the father. So if you take out all the mother's genes, because the mother is known, now the remaining bands of this boy must be those ones of the father. Let me show you what I'm trying to say. This mother, okay, this one is not for them. Yes, this one is corresponding. So this one is out. This one corresponds. This thing is now jamming. All right, corresponds. This one corresponds. Oh, this one corresponds. Yes. This one is not matching. So this one is not matching. This one is not matching. This one corresponds. Is out. This one is not matching. This one is not matching because this is not. This one is matching. So now all of those ones which have X must match with the male who is the father. That's why I'm saying that the remaining bands of the boy, after those ones of mother have been removed, they must match with the father. Okay, this one, yes, it matches. You see, this one, yes, it matches. You see, this one is matching with this. Use it to answer and also to score a mark. They are saying hemophilia is a that statement is enough. This statement, hemophilia is a sex-linked trait. Hemophilia is a sex-linked what? Trait. Meaning that if it is a sex-linked trait, what is it? It means what does it mean? It means that you're gonna use X and Y. Please, if they don't say is a sex linked in exam. Don't use X and Y. You're gonna get it wrong. Is a sex linked disorder. Don't use X and Y if it is not mentioned in the question. So here it is mentioned sex linked. Or sometimes they don't say sex linked. They say that hemophilia, the gene is carried on X chromosome. Still that is sex linked. If it is not mentioned, please don't even bother. Don't even waste your time. Don't even waste your time to let me show you here. One of the deafness in human is carried on a single allele. The diagram below shows, yeah, done. They are not talking about sex linked. However much they talk about Paul, and you know that Paul is a boy, Liz is a girl, Mary is a girl, John is a boy. However much you know that, please don't use X and Y. Please don't use X and Y. So if they don't mention sex linked, don't use X and Y. If they mention sex linked, then you talk about X and Y. How many generations are here? There are 3. 1, 2, 3. They are saying that and then number 2, it can be caused by something can be caused by a recessive allele, or it can be caused by a dominant allele. Anyway, let's continue with this question. They are saying, state what is represented by a circle or a diagram. I wanted to tell you that sometimes we don't bring that a circle is a male. No. A box is a male. A circle is a female. Sometimes don't give you that key. You have to go to the exam when you know that. All right. Now they're saying, what are they saying? Okay. Now they are saying, this is a boy. This is a. How many individuals? Let me brainstorm you. This is affected with hemophilia. Don't say he's sick. Is sick of what? Sick of what? Malaria, ne? Sick of what? Sick of hemophilia, or is hemophilic. All right. How many kids are produced by P and Q? They are how many? It's 1, 2, 3, 4, 5. I know many people are gonna say that. It's not. They are not 5. 1, 2, 3, 4, 5. No. This one is an intruder. This one intruder. This one got married this. This one got married this. So there are only 3 kids. How many generations are here? 1, 2, 3. There are 3 generations. You understand? What is the phenotype, the genotype, or the phenotype? They can ask you what is the genotype or phenotype of P or Q, whatever they can ask. But if it is shaded, it means that that individual is affected. Now, they're saying that state what is represented by a circle. We have said that the circle means a male. State the number of general A. We have talked about them. There are 3 generations. 1, 2, 3. Offsprings P and Q. State the number of offspring P. The same thing is this, and this. This one and this ones, they are only and only got married to that. So they are intruders. State, they're saying that let us give the letters of only male who are having hemophilia. Letters of the female. Female is T and the female is U. Those are the only 2 people who are having hemophilia. Why? Because they are shaded. And then lastly, the genotype of R. The genotype of R. Now, how do we find the genotype of R? Look at the babies or look at the parents. First, look at the babies. It can help you. If you see that the baby is one baby is hemophilic, then automatically the mother is gonna be heterozygous. So the mother is gonna remember that here you have X and Y. Y doesn't carry anything. Y doesn't carry anything. So it's X and Y. So Y does not carry anything at all. All right. I think now, okay, so since this one is sick, it means that, and is a boy, so it's gonna be like this. It's gonna be, this is a recessive allele. So if it's a recessive allele, obviously this can't come from the father, because the father already gave the what? The Y. Therefore, this came from the mother. So it means that the mother, since is normal, but a carrier. So the mother is gonna be, or R is gonna be capital A, capital X, capital X, capital H, small h. and that becomes the answer for that. Yes, let's continue now. Let's continue. So we are done with this. Sex determination and biotechnology. How do I explain sex determination? Sex determination is like describing a karyotype. When asked you to describe sex determination, it's like describing a karyotype. But here, if they ask you the genetic cross, you have to do the cross. Ne? So, for example, if it's a cross, you tell us that P1, F1, everything, ne? Male crossing with a female, yeah, and then you tell us that male is X. You tell us the phenotype here, ne? And then you tell us the genotype. Male is X, Y, and then female is XX. Then you cross. Then you tell us meiosis here. You tell us gametes. They come out here. And then I'm gonna show you how to write it if they say using a genetic cross, and then also show you how do you describe if they ask you to describe. Then these are gametes. These are gametes. Then they cross, then they cross, then this and this, cross Y, and then this and this, they cross like that. So this is the offsprings, meaning that you have 50% chance of having boys, and 50% chance of having girls. What about those who have girls, or boys? Yes, even if you have a boy here, the next time you have 50% of a boy again. Even if you have another boy, the next generation, you have the next reproductive cycle, you're gonna have still 50% chance of having a what? Having a boy. So it depends on how do you roll your dice, and how many chances do you have to have a boy. There are 50, but you are just lucky that you have what? Boys, or you are lucky to have what? To have girls. So in this case, we are saying that 50 boys, 50 girls. Yeah. If they ask you to describe, you tell us that individuals, humans are made up of 46 chromosomes. Ne? The 23rd pair is called gonosome. It's either X and X or X and Y. If X and X, if a male sperm with X fertilizes the female ovum, it will result in X and X. The individual will be a female. If a male sperm having Y fertilizes the ovum, remember ovum is always X, the individual will be X and Y, and therefore will be a male. There are 50% chance of having a male and 50% chance of having a female. That's how you can describe sexy linked. So let's finish up the cloning, and then we go to evolution. We finalize it with the evolution. All right. Biotech. Cloning. What is stem cells? What are stem cells? Stem cells, these are cells which have ability to divide to form other body structures. Imagine you are just a sperm. You are just a sperm, ne? Sperm fused with the ovum. You formed a zygote. The zygote, remember this paper one formed a ball of cells, ne? Which called the morula. You see, morula is a stem cell because there you guys back. We are back now, ne? Yeah, all right. Guys, yeah. Quiet, guys. Shh.
So today's like for
All right. Can we start? Can we start? Yeah, so that we can finish. All right. Now, evolution is part of paper 2 and it's out of 54 marks. It's 1/3 of the paper. Right, so there's part 1 evolution and part 2. Right, let's start. Evolution. First thing, what is evolution? Yeah, so evolution, it's change over time. It's change over time. So now there's an argument as to whether evolution is a theory or is a hypothesis. But now before we conclude whether evolution is a theory or a hypothesis, we need to understand what is a theory and also what is a hypothesis. All right, what is a theory? Right, so a theory, it's an explanation that, or it's an explanation for a phenomena that has been observed in nature that is accepted as the truth because it's supported by evidence and facts. But now, what is a hypothesis? A hypothesis is a testable statement. It's a testable statement. So a theory, it's an explanation for a phenomena that has been scientifically accepted as the truth because it is backed by evidence and facts. Whereas a hypothesis is a tentative explanation for a phenomena that can be tested and proven by further investigation. In other words, a hypothesis is a testable statement. Now, is evolution a theory or a hypothesis? Yeah, so evolution is a theory. Why are we saying evolution is a theory? It's because it is backed by evidence, right? Now the question is, what is the evidence of evolution? Right, number one, we have fossil evidence. Fossil evidence. Number two, we have genetic evidence. Number three, we have, okay, not yet. We have biogeography. Cultural evidence is the evidence for human evolution. We're still talking about evolution, right? And then the last one is, so we have fossil evidence, genetic evidence, biogeography, and we have modification by descent, sometimes called homologous structures.
structures. So if a question says to you, how does fossils serve as an evidence of evolution? How does fossils serve as an evidence of evolution? Right, you're going to say, right, so remember, fossils are remains of organisms that have lived in the past, right? And these remains of organisms, they are mineralized, which means they are found in rocks. Yeah? Are we together? So when they check the rocks, they find that the oldest rocks, so say this is a sedimentary rock. So the oldest rocks contain the simplest fossils. And the newest rocks contains the most complex fossils. Which means that this fossil has changed over time, right? So that is fossil evidence. The oldest rocks contain the simplest fossils, and the newest rocks contain the most complex fossils, right? Now, when we look at genetic evidence, we are saying that if different organisms have the same structure of genes, they have the same sequence of genes, and they have the same mutation, it means that they share a common ancestor. For example, when you look at the human beings and the chimpanzees, what is it that they share in common? They share what? A common ancestor, isn't it? Right. So why are we saying that they share a common ancestor? What is one evidence that tells you that they share a common ancestor? It's because their DNA is similar. Right. And then now we have biogeography. Biogeography means the study of the past and the present distribution of organisms. Right. When we talk of biogeography, we are speaking about speciation. Right. Yeah, there's a scientist which was called Alfred Wagner that pinned a theory which was called continental drift, where he said that the earth was one big landmass called Pangaea, and it's separated into two, isn't it? Into what? Gondwana land and Laurasia. So when the earth separated, the organisms were distributed into different environmental conditions. And we know that if we change the environmental condition, then natural selection take place, isn't it? Right. And then we have homologous structures. So homologous structures, those are structures which looks the same but perform different functions. Those are homologous structures. Different structures in different organisms, and they perform different functions, but they look the same. Those are homologous structures. So we're saying that if different organisms have the same anatomical plan, it means that they share a common ancestor. Right. So now evolution cannot take place until there's variation. Right? Who can tell me what is variation? What is variation? What is variation? Yes? Different of species. No, variation means differences in organisms of the same species. That's variation. Variation, it means differences in organisms of the same species. That's variation. And now you need to know the sources of variation. That yes, we are of the same species, we belong to Homo sapien, but we are not the same, we are varied. What causes variation? Right, we remember the sources of variation by saying C, R, R, M. So C for crossing over, random arrangement of chromosomes during metaphase, random fertilization, random mating, and then mutation. Right? So these are sources of variation. Right? It's crossing over, random arrangement of chromosomes, random fertilization, random mating, and then mutation. Then from there, you need to know two types of variation. There's what? Continuous variation and discontinuous variation. So these are types of variation. So you have continuous variation and discontinuous variation. Right? Continuous variation is a variation where there is a range of intermediate phenotypes for a particular characteristic. There's a range of intermediate phenotypes for a particular characteristic. For example, its height, its skin tone, its shoe size. Those are examples of a continuous variation, a variation that continues. Yeah, if you like, you have someone who's tall, taller, tallest, or you can have someone who's short, shorter, shortest. So this variation in terms of height, it continues, skin tone and shoe size. And then you have discontinuous variation. Right? Discontinuous variation is a variation where there is no range of different intermediate phenotypes for a particular characteristic. So this variation, it stops, it doesn't continue. An example of discontinuous variation would be blood groups. What else? Gender. Yeah, it's either you're a male or you're a female. It's either you belong to blood group A, B, AB, or blood group O. Right? So those are examples of discontinuous and continuous variation. Right? Let's just do a quick recap. We said evolution is change over time. Right? And then we said a theory, it's an explanation for a phenomena that is accepted as the truth because it's backed by evidence and facts. Whereas a hypothesis, we said it's a tentative explanation for a phenomena that can be tested and proven by further investigation. But a short explanation is that it's a testable statement. And now we said that evolution is a theory. The reason why we are saying it's a theory is because it is backed by evidence and facts. And there are four sources of evidence that we can draw from. It's fossil evidence, genetic evidence, biogeography, homologous structures. Right. And then we said that evolution cannot take place until there's variation. But now what is variation? Variation, it's the differences in organisms of the same species. And then we said that there's 5 sources of variation. It's crossing over, random arrangement of chromosomes, random fertilization, random mating, and then mutation. Right? So if a question asks you, name 2 sources of variation that we find in meiosis, it's crossing over and random arrangement of chromosomes. And then we spoke about the 2 types of variation. We have continuous variation and discontinuous variation. All right. Now let's go to evolution by Lamarck. Lamarck. Right. So Lamarck tried to explain how evolution takes place. In his quest, he came up with 2 laws. What is the first law? Law of use and disuse. And then what is the second law? Law of the inheritance. Law of the inheritance of acquired characteristics. Right. Now, what does the law of use and disuse state? Right. It states that, wait, so it states that if you use an organism, if you use an organ more often, if you use a structure or an organ more often, that structure will become better developed and elongated. And if an organism does not use a structure more often, that structure will shrink until it disappears. Right? So that's the law of use and disuse. If an organism uses a structure more often, that structure will become better developed and elongated. Right? And if an organism does not use a structure more often, that structure will shrink and disappear. Right? And then now we have the second law, which is the law of the inheritance of acquired characteristics. Right? So this law, it states that the characteristics that an organism has acquired in a lifetime can be passed on to the next generation. The characteristics that an organism has acquired in a lifetime can be passed on to the next generation. Right, let's explain these laws. So the law of use and disuse, it basically means that if you are going to use your hands more often, right, to do whatsoever you want to do, right, so it means that your hands will become better developed and elongated, and that characteristics of long hands will then be passed on to the next generation. In other words, your children will be born with long hands. All right. Now, if a question comes to you and shows you a diagram of a giraffe, right? Okay. So, if a question comes and says, use the laws of Lamarck to explain the development of long necks in giraffes. So now they are saying the development of long necks in giraffes, which means the giraffes originally they had short necks, isn't it? Right. Now, how do you go about answering this question? So you are going to say originally, number one, originally giraffes had short necks. Originally, giraffes had short necks. Point number two, they used their necks more often to reach the food on high trees. Point number three, all right, originally giraffes had short necks. Second point, they used their necks more often to reach the food on high trees. Number three, as a result, their necks became better developed and elongated. So originally giraffes had short necks. They used their necks more often to reach the food on high trees. As a result, their necks became better developed, and elongated. Point number 4. The characteristics of long necks was then passed on to the next generation. And then eventually, all giraffes had long necks, sorry. Right, so again, we are saying if the question says explain the development of long necks in giraffes using Lamarck. So we say originally giraffes had short necks. They used their necks more often to reach the food on high trees. As a result, their necks became better developed and elongated. Then the characteristics of long necks was then passed on to the next generation. Eventually, all giraffes had long necks. Right. But now if a question comes again and say use Lamarck, the laws of Lamarck, to explain why is it that the snakes do not have legs. Use the laws of Lamarck. It's a snake now. Use the laws of Lamarck. Use the laws of Lamarck to explain why is it that the snakes do not have legs. So you're going to say originally snakes had legs. Originally snakes had legs. That's point number 1. Point number 2, they did not use their legs more often. Point number 3, as a result, their legs shrinked until they disappeared. And then point number 4, the characteristic of no leg was then passed on to the next generation. And then eventually, all the snakes did not have legs. So we have explained the laws of use using the giraffe, and we've explained the law of disuse using the snake. Right, this is the laws of Lamarck. Right, but now these laws of Lamarck, they were rejected. And the reason why they were rejected is because, number 1, it's very important. They will ask you, why is it that the laws of Lamarck were rejected? They were rejected because acquired characteristics cannot be passed on to the next generation. Did you get that? Why was it rejected? Acquired characteristics cannot be passed on to the next generation. Number 1, acquired characteristics cannot be passed on to the next generation. The second reason why they rejected these laws is because organisms, they do not evolve because they want to. Right. So since these laws were rejected, then there must be another theory, accepted theory for evolution. And that theory is called natural selection by Charles Darwin. So Charles Darwin explained how evolution takes place by the process called natural selection. Right. Now we had Lamarck, now it's time for us to hear Darwin. So Charles Darwin, when he explains natural selection, he starts by saying, all right, number 1, there's a great deal of variation amongst offsprings. There's a great deal of variation amongst the offsprings. Great deal of variation amongst the offsprings. Right. In the second point, he explains the variation. He says some organisms had favorable characteristics and some organisms had unfavorable characteristics. So he first says in point number 1, there's a great deal of variation amongst the offsprings. The second point, he explains the variation. He says some offsprings had favorable characteristics and some offsprings had unfavorable characteristics. And then point number 3, he says now when there's a change in the environment that sparks competition for survival, when there's a change in the environment that sparks competition for survival, then point number 4, obviously organisms with favorable characteristics will survive and organisms with unfavorable characteristics will die. So first point, there's a great deal of variation amongst offsprings. Second point, he explains the variation. Some offsprings have favorable characteristics and some offsprings have unfavorable characteristics. Point number 3, now when there's a change in the environment, there's now a competition for survival. Point number 4, organisms with favorable characteristics survive and organisms with unfavorable characteristics, they die. And then point number 5, the organisms that survive, they reproduce. These organisms that survive, they reproduce and they pass on the allele of a favorable characteristic to their offspring or to the next generation. And then point number 6, eventually there's a great proportion of organisms with favorable characteristics. That's natural selection, right? And then point number 6, eventually there's a great proportion of organisms with favorable characteristics, right? So that is Charles Darwin explaining evolution and how it takes place. Says there's a great deal of variation among offsprings. Says some offsprings have favorable characteristics and some offsprings have unfavorable characteristics. Then he says when there's a change in the environment, those organisms with favorable characteristics will survive and the ones with unfavorable characteristics will die. And the ones that survive will reproduce and pass on the allele of a favorable characteristics to the next generation. And eventually, we have a great proportion of organisms with favorable characteristics. All right. Now, if we go back to this diagram, this structure again, and then now they are saying using the theory of natural selection, explain why is it, or explain the development of long necks in giraffes. So we are going to say there was a great deal of variation amongst giraffes. You mentioned the species. There's a great deal of variation amongst giraffes. That's point number 1. Point number 2, you explain the variation. Some giraffes had long necks and some giraffes had short necks. And then point number 3, when food ran out on lower trees, that's a change in the environment. When food ran out on lower trees, the giraffes with long necks, point number 4, giraffes with long necks survived because they could reach the food on high trees. And giraffes with short necks, they died because they could not reach the food on high trees. Then point number 5, the giraffes that survived reproduced and passed on the allele of long necks to their offsprings. And then eventually, there's a great proportion of giraffes with long necks. Are we fine? Right. Now let's go to artificial selection. Artificial selection. Artificial selection, it means that it's the selection and the breeding of organisms with desirable characteristics by humans. Artificial selection, it means it's the selection and breeding of organisms with desirable characteristics by humans. That is artificial selection. Is the selection and the breeding of organisms with desirable characteristics by humans. That's artificial selection. Right. So how does it take place? Human beings, they are going to select organisms with desirable characteristics, interbreed them, then they are going to produce offsprings. And then from the offsprings, they are going to select organisms with desirable characteristics and breed them. And repeat that process until they reach a desired outcome. Right? If I was you, I was going to write that down. So humans, they select organisms with desirable characteristics and breed them, and they'll produce offsprings. Then from the offsprings, they'll select organisms with desirable characteristics and breed them, and repeat that process until they reach a desired outcome. So if they say to you, for example, a farmer, a cattle farmer, wants to start a business where he sells milk, but there are few cows that produce a lot of milk from his farm. Explain how he can use artificial selection to increase the number of cows that produce a lot of milk. So you are going to say the farmer will select the cows that produce a lot of milk, breed them together, and then they will produce offsprings. And then from the offsprings, he will select the cows that produce a lot of milk and breed them together, and repeat that process until he reach a desired outcome. All right. Then they'll also ask you to tabulate the differences between artificial selection and natural selection. The first thing you do is that you draw a table. After drawing a table, you write natural selection, Don't write NS, write natural selection. And then here you write artificial selection. Then you are going to get 1 mark for a table. Right. Now, what is the difference between natural selection and artificial selection? Right. It's mostly 5 marks, which is 1 mark for a table, then 1 mark for the differences. Different number 1 is that natural selection, the environment represent a selective force. The environment is a selective force. In natural selection, it's the environment that decides which organisms survive. So the environment is the selective force. Whereas in artificial selection, humans represent a selective force. Humans represent a selective force. Right. Number 2. Right. Number 2. In natural selection, evolution occurs within a species. It takes place within a species. It takes place within a species. Natural selection takes place within a species. That's why we are going to say a population. I mean, there was a great deal of variation amongst giraffes. Amongst giraffes. So it happens within a species. Whereas in artificial selection, it can happen across different species. It can happen across different species. Can happen across different species. All right. We'll just do a quick recap before we move on. Right. We spoke about Lamarck. We said Lamarck came up with 2 laws of use and disuse, right? And the second law is the law of the inheritance of acquired characteristics, right? So the law of use and disuse, we said it states that if an organism uses a structure more often, that structure will become better, developed, and elongated. And if an organism does not use a structure more often, that structure will shrink until it disappears. Nah? Then the second law, it's the law of the inheritance of acquired characteristics, which states that the characteristics that an organism has acquired in a lifetime can be passed on to the next generation. Right. Then we said that if a question comes and says, explain the development of long necks in giraffes using Lamarck. So you're going to say, originally, all giraffes had short necks. Hey, what happened? They used their necks more often to reach the food on high trees. As a result, their necks became better, developed, and elongated. Then the characteristics of long necks was then passed on to the next generation, right? Then eventually all giraffes had long necks. So there's no variation when it comes to Lamarck. And then if they say to you, explain why is it that the snakes do not have legs, you are going to say no, originally snakes had legs. The problem is that they did not use their legs more often. And as a result, their legs shrinked until they disappeared. Then the characteristics of no legs was then passed on to the next generation. Eventually, all the snakes did not have legs. Right? But then we said now these laws of Lamarck were rejected. The reason why they rejected number one is because acquired characteristics cannot be passed on to the next generation. And then number two, organisms do not evolve because they want to. Right, and then we spoke about natural selection. We said that there's a great deal of variation among offsprings, eh? And then number two, we explained the variation. Some have favorable characteristics and some have unfavorable characteristics. And then number three, there's a problem. When there's a change in the environment, those with favorable characteristics, they survive, and those with unfavorable characteristics, they die. And the ones that survive reproduce and pass on the allele of favorable characteristics to the next generation. Right, and then now we spoke about artificial selection. We said it's the selection and the breeding of organisms with desirable characteristics. Nah. And then we spoke about the differences. Right, now question is, what is punctuated equilibrium? What is punctuated equilibrium? Yes, sharp. Right, punctuated equilibrium, it's a hypothesis that explains the speed at which evolution takes place. That is punctuated equilibrium. It's a hypothesis that explains the speed at which evolution takes place. So a scientist that came up with punctuated equilibrium is called Neil Eldridge. It's Eldridge and Wood. And Coult, sorry. Right, it's Eldridge it's Neil Eldridge and Coult. Right, now what does punctuated equilibrium state? Right, so now we have time and then we have change. Right, punctuated equilibrium, it states that evolution involves a long period of time where there's no change, and this alternates with a short period of time where there's rapid change through natural selection. If I was you, I was gonna write that down because I'll tell you why Monday. Right, so punctuated equilibrium, it states that evolution involves a long period of time where there is no change, and this alternates with a short period of time where there's rapid change through natural selection. That is punctuated equilibrium. Right, are we fine? Can we move now? Right, now there's what we call speciation. Speciation. The word speciation, it means the formation of new species. The formation of new species. That is speciation. It means the formation of new species. Right, so if a question comes to you and say explain speciation using geographic isolation, then you must say, number one, if a population of a single species becomes separated by a geographical barrier. Right? If a population of a single species becomes separated by a geographical barrier, point number 2, obviously the population will split, isn't it? Yeah, it will split into two, provided that that geographical barrier is separating the population into two. Right? So if a population of a single species becomes separated by a geographical barrier, the population will split into two. Point number 3, there is now no gene flow between the two populations. Point number 4, since the two populations are now exposed to different environmental conditions, natural selection will take place independently, such that they become different from each other genotypically and phenotypically. And even if they were to mix again, they would not interbreed and produce fertile offsprings. All right. Okay, let's go back. Right, so we are saying that if they say to you, explain speciation using geographic isolation, so you are going to say, now if a population of a single species becomes separated by a geographical barrier, number 1, if a population of a single species becomes separated by a geographical barrier, number 2, the population will split into two. Number 3, there is no gene flow between the two populations. Number 4, since the two populations are now exposed to different environmental conditions, natural selection will take place independently in each population, such that the two populations become different from each other genotypically and phenotypically. And even if these two populations were to mix again, they would not interbreed and produce fertile offsprings. All right. Now let's explain it using the application of theory. All right. So let's say now we have a pond or a dam. Yeah? Right. So in this dam, there's species of fish, isn't it? And then there's variation there because we know that there's always variation, isn't it? Right? There are fishes which are blue. And there are ones that are black. Look at the fishes there. Right, so we have two types of fish. We have blue fish and then we have black fish. Right, so if this population becomes separated by a strip of land, right, so this is the original population. So this population is separated by a strip of land. Separated by a strip of land. of water or of land. Ne? Right. Now we have these fishes here. All right. So now let's say this one is a dark reef, maybe dark pond, or here it's a blue. Right. So now, so if they say to you, use the diagram above to explain the formation. Let's give these fishes names. What name can we give this one? Yeah? Naled. And then this one, Sihle. Alright. So now they are asking you explain using speciation the formation of a species called Sihle and another one called Naledi in the fishes. Right. So can we pay attention? Right. So we are going to say, right, the population of fishes was separated by a strip of land. The population split into two. There was no gene flow between the two populations. Since the two populations were now exposed to different environmental conditions, then you explain that different environmental condition. Some they were found in a blue dam and others they were found in a dark dam. Right? Since the two populations are exposed to different environmental conditions, some are found in a blue dam and some are found in a dark dam. Natural selection took place independently in each population such that they became different. The fish became different from each other genotypically and phenotypically. And even if they were to mix again, they would not interbreed and produce fertile offsprings. Hey, right. So you can see that here in a dark dam, the ones that are dark colored, they survive, right? Why? Because they are camouflaged. And the ones that are blue, they die because they are easily exposed. Can you see it? Right? Did you get that one? Or should we do another example? The last example. Right. So if they show you, right, now let's say this is Africa, and then here we are going to have Australia, right? And then we have dogs. Here we have what you call jackals, and then here we have coyote, right? And they ask you explain the formation of jackals and the coyote in the dog family, right? Using speciation. Right, explain the formation of the jackal and the coyote in the dog family using speciation. So you are going to say the population of a dog family was separated by the ocean. The population split into two. There was no gene flow between the two populations. Since the two populations were exposed to different environmental conditions, natural selection took place independently in each population, such that the two populations became different from each other genotypically and phenotypically. And even if they were to mix again, they would not interbreed and produce fertile offsprings. Right. Now let's do mechanisms of reproductive isolation. Mechanisms of reproductive isolation. Oh yeah. All right. So we have 5 mechanisms of reproductive isolation. What are those? Those are mechanisms that keep organisms which are not of the same species separate. Right. Mechanisms. If I was you, I was gonna write notes instead of checking the book. It helps. Of reproductive. Mechanisms of reproductive isolation. Those are mechanisms that keep species separate or keeps organisms of different species separate. Right, we have 5. The first one is species-specific courtship behavior. Species-specific courtship behavior. Species-specific courtship behavior. Yeah, species-specific courtship behavior. That if different organisms have different courtship behaviors, then they won't be able to interbreed. Right. Number 2, it's being adapted to different pollinating agents. Being adapted to different pollinating agents. Right. So flowers reproduce by a process called pollination, which is a transfer of pollen from an anther of one plant to the stigma of another plant. That's called cross-pollination. Right. But this pollination is made possible by the agents which are called pollinating agent. It can be bees, it can be wind, it can be any flies. So if different flowers are adapted to different pollinating agents, then they won't be able to interbreed. Are we together? Right. And then number 3. Hey, there's noise there. That disturbs me. Right. Number 1, species specific courtship behavior being adapted to different pollinating agent. Number 3, infertile offsprings. Infertile offsprings. That during artificial selection, if you interbreed organisms of different species, then the offspring will be infertile, which means the generation ends there. It won't reproduce any further. Right. Number 4, it's breeding at different times of the year. Breeding at different times of the year. Breeding at different times of the year. Right. And then the last one, it's prevention of fertilization. Prevention of fertilization. Prevention of fertilization. Right. What causes that one? Right. Who can tell me what causes prevention of fertilization? You can't have a huge animal interbreeding with a small animal because a huge animal has big genitals. Yeah, so those genitals really that they cannot. In other words, let me just put it blunt: the penis cannot deposit sperms inside the vagina. So we call that prevention of fertilization because it's too big and the vagina is small. Yeah. All right. Now we go to human evolution. Right. Human evolution. Human evolution. Right. Hey, shh. We are doing human evolution now. Right. But before we go to human evolution, please just know present day evolution. When we are talking about present day evolution, that's when we are talking about uh, resistance, having resistance. So they are going to ask you, maybe a question will come and say, right, explain the development of resistance of bacteria to antibiotics. Explain the development of resistance of bacteria to antibiotics. So you're going to say, now there's a great deal of variation among bacteria. Some bacteria are resistant to antibiotics, and some bacteria are not resistant to antibiotics. When the antibiotics are introduced to their environment, the bacteria that are resistant to antibiotics, they survive. And the ones that are not resistant to antibiotics, they die. And the bacteria that survive, they reproduce and pass on the allele of resistance to their next generation. And eventually, there's a great proportion of bacteria which are resistant to antibiotics. Hmm? Right. The question is, explain the development of resistance of bacteria to antibiotics. Explain the development of resistance of bacteria to antibiotics. So we are going to say there's a great deal of variation amongst bacteria. Some bacteria are resistant to antibiotics and some bacteria are not resistant to antibiotics. When the antibiotics is introduced to their environment, the bacteria that are resistant, they survive. And the bacteria that are not resistant, they die. And the bacteria that survive reproduce and pass on the allele of resistance to their offsprings or to the next generation. Then eventually, there's a great proportion of bacteria that are resistant to antibiotics. Now we are doing human evolution. Human evolution. Right, human evolution. Human evolution. All right. Are you ready for human evolution? Yes. All right. Um, right. Human evolution. Human evolution. All right. So for you to master human evolution, number one, you must know the classification of humans. Classification of humans, number one. Right. Classification of humans. Right. Number one, just know that humans belong to kingdom Animalia. Classification. Number one, kingdom Animalia. Number two, phylum Chordata. Number three, class Mammalia. Number four, order primates. Number five, to what family do human beings belong to? Hominidae. Right, so it's kingdom Animalia, phylum Chordata, class Mammalia, order primates, family Hominidae. Right, and then number six, we have genus. The genus for human beings, it's Homo. And then number seven, it's species. To what species do we belong? We belong to Homo sapien. Underlined. So when you write Homo sapien, you must ensure that you underline it. Right, it's kingdom Animalia, phylum Chordata, class Mammalia, order primates, family Hominidae, genus Homo, and then species Homo sapien, underlined. Right, then from there you need to know the similarities that human beings share with African apes. Right, you underline the entire thing. Right, everything. Right, now we need to know the similarities that human beings share with African apes. Okay, I'm trying to draw a human being. Right, so similarities that human beings share with African apes. Number one, we have large brain size. Number two, we have two front eyes. Number three, we have freely rotating arm. Number four, we have long upper arm. Number five, we have rotation at the elbow joint. Number six, we have opposable thumb. Opposable thumb. And then number seven, we have flat nails instead of claws. Right. So these are similarities that human beings share with African apes. So we have large brain size, two front eyes, freely rotating arm, long upper arm, rotation at the elbow joint, opposable thumb, and then flat nails instead of claws. So this is the upper limb. The hand is the upper limb. So if a question says, name the similarities between human beings and African apes in their upper limbs. So obviously it's freely rotating arm, long upper arm, rotation at the elbow joint, opposable thumb, flat nails instead of claws. Right. Those are similarities between human beings and African apes. Similarities between human beings and African apes. Right. Now let's go to differences between human beings and African apes. Are we still together? Are we still together now? Yes. All right. Now the differences between human beings and African apes. So we'll have a human, and then you're going to have an African ape. Nice drawing, eh? All right. So this is a human, and then this one is an African ape. Right. Now they ask you what is the difference between the human beings and the African ape, or they say tabulate visible differences between human beings and African apes. Right. Number one, African apes have a small cranium size. Humans have a big cranium size. That's number one difference. So there's a small cranium size compared to this one. African apes have a small cranium size. Humans have a large cranium size. Number one. Number two is that African apes have pronounced eyebrow ridges. African apes have pronounced eyebrow ridges. Humans have less pronounced eyebrow ridges. Number three is that African apes have a sloping face. And then humans have a flat face. And then number four, African apes are prognanthous. African apes are prognanthous. And then humans are non-prognanthous, not less. They are non-prognanthous. The giraffe is appearing there. Lying again corn. Right. So just a review of visible differences. African apes have small cranium size. Human beings have a large cranium size. African apes have pronounced eyebrow ridges. Humans have less pronounced eyebrow ridges. African apes have a sloping face, whereas humans have a flat face. And then number four, African apes are prognanthous, whereas humans are non-prognanthous. And then number five, African apes have large canines, whereas humans have small canines. Then number six, African apes have a rectangular shaped palate and humans have a C-shaped palate. Then number seven, oh wait, right, number seven, the foramen magnum of African apes is pushed to a more backward position, whereas the foramen magnum of humans is pushed to a more forward position. Right, so those are differences between human beings and African apes. Right, then now we go to bipedalism. 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 Right. Bipedalism is the ability to walk on two lower limbs. Right. Your hands, they are called your upper limbs. Your legs are called your lower limbs. So bipedalism is the ability to walk on two lower limbs. That is bipedalism. The ability to walk on two lower limbs. Right. There are three factors that affect bipedalism. There are three factors that affect bipedalism. In other words, there are three things that makes organisms who are bipedal to be bipedal. Right. Number one is that the foramen magnum is pushed to a more forward position. So foramen magnum is pushed to a more forward position so that the spinal cord can join in vertically. The foramen magnum is pushed to a more forward position so that the spinal cord can join in vertically. That's number one. Number two, the spine is S-shaped. The spine, so we don't say the spinal cord. We say the spine is S-shaped to allow for flexibility. The spine is S-shaped to allow for flexibility. And then number three, the pelvis is short and wide to support the upper body mass. The pelvis is short and wide to support the upper body mass. So those are three factors that affect bipedalism. Foramen magnum is pushed to a more forward position. The spine is S-shaped. The pelvis is short and wide. Right. Now, we need to know the differences between quadrupedalism and bipedalism. When they say tabulate the differences between quadrupedalism and bipedalism, right? Quadrupedalism is the ability to walk on four lower limbs. The ability to walk on four limbs. That is quadrupedalism. Right, so we have bipedalism here, and then we have quadrupedalism. Right, so in bipedalism, the foramen magnum is pushed to a more forward position, whereas in quadrupedalism, the foramen magnum is pushed to a more backward position. And then in bipedalism, the spine is S-shaped, and in quadrupedalism, the spine is C-shaped. And then last one, in quadrupedalism, the pelvis is short and wide, whereas in quadrupedalism, the pelvis is long and narrow. It's long and narrow. So those are differences between bipedalism and quadrupedalism. Right. Then they are going to ask you about, they might ask you about the advantages of bipedalism. What are advantages? What are advantages? Number one is that you have a better field of view. Number one, you have a better field of view. You can see far. You can spot predators from afar. That's number one. And then number two is that movement is easy and efficient. You can move easily and efficiently. Right, number three is that your hands are free to carry tools. You can use your hands. Your hands are free to carry tools. Right, they won't ask you more than three advantages. Right, now let's come to the evidence of human evolution. If human evolution is really a theory, then there should be evidence of human evolution. Right, evidence of human evolution. Evidence of human evolution. Right, so we have three evidence of human evolution. Number one, we have fossil evidence. Fossil evidence. When we are talking of fossil evidence, we are talking about the transitional fossils. Transitional fossils. Fossils that have characteristics which are common to the ancestor species and the species that follows after. Right. And then number two, we talk of cultural evidence. When we speak about cultural evidence, we are talking about the use of tools. Then the last one is genetic evidence. The last one is genetic evidence. Genetic evidence. So here we are talking about mitochondrial DNA. So these are three evidence of human evolution. It's fossil evidence. We are talking about the transitional species or fossils. And remember, who was this one? Karab. Right? Australopithecus sediba. But we'll go there, it's fine. Right, and then cultural evidence, we are talking about the use of tools. Here we are talking about Homo habilis, handyman, the first one to use tools. And then we speak of mitochondrial DNA when we are talking about genetic evidence. Right, let's explain this evidence further, and then we do out of Africa hypothesis. We look at the phylogenetic trees, and then we are done. All right. Now when we talk of fossil evidence, we are looking at the fossil record. We are looking at the fossil record. All right. Do you remember the fossil record?
Yes.
Let's see if you can remember it. See, people, you must close your books.
Sure. Sure.
Can we close distinction material? Thank you. Hey, these ones, they want monetary value now. They want man. There is, it's fine. Yeah.
Oh.
All right. So remember, on the fossil record, you must know the scientific name. Nah? I, you must know the scientific name. Right. You must know the scientific name. And then you must know the common name. And then you must know who discovered it. Who discovered it. And then you must know where. Where did they discover it? Right. What was the oldest fossil? Right. So the first one was Ardipithecus. Ardipithecus ramadas. Right. So what was the common name for Ardipithecus ramadas? It was Ardi. Right. Who discovered Ardipithecus ramadas? It was Tim White. Where? In Ethiopia. Right. So the oldest fossil, it was Ardipithecus ramadas. Common name was Adi. And then Adi was discovered by Tim White. And then in Ethiopia. Right. After Ardipithecus ramadas, we had who? Australopithecus afarensis. Right. Australopithecus afarensis. The common name was Lucy. Who discovered Lucy? Johansson. Where? In Ethiopia. Right. After Australopithecus afarensis, we had what? Australopithecus africanus. So it's Australopithecus africanus. Ne? Africanus. Australopithecus africanus. Africanus. So there are three, isn't it? Right. The first one is Daung child. Right. So who discovered Daung child? It was discovered by Raymond Dutz. Right. Where? In Daung. Right. And then the second one was okay, Mrs. Bless. Right. Mrs. Bless was discovered by who? Roberts. Who? Where? In the Steekfontein, right? And then the last one was Littlefoot. So Littlefoot was discovered by who? Ron Clark. Where? In the Steekfontein. Steekfontein. Is it jealous or something? Right. Now after these species, Australopithecus africanus, we had a transitional fossil. Right. Who was a transitional fossil? Australopithecus sediba. Right. What was the common name for Australopithecus sediba? Karabo. Hey, now, why is it doing this? Karabo. Right. Who discovered Karabo? It's Prof. Libega. Ne? Right. Where? Malapa. Cradle of humankind. Yeah, it's a fossil site. Right. And then after Australopithecus sediba, we had who? Homo habilis. So we had Homo habilis. Right. What was the common name for Homo habilis? So this one was handyman. Why was he called handyman? Yeah, so the first one to use tools, eh? Right, who discovered Homo habilis? Yeah, it's Mary and Louis Leakey. Right, where? In Tanzania. So you need to know all of this. All of it, you need to know it. And I'm glad you do. Right. And then we had Homo erectus. Homo erectus. Right. What was the common name for Homo erectus? Upright man. Right. Who discovered upright man? It was Eugene Du Bois. Right. Where did Eugene Du Bois discover this fossil? In Indonesia. Right. And then the last one was Homo sapien. Right. Common name for Homo sapien? It was wise man. Right. Where did they discover? Oh, who discovered wise men? Tim White. Where? In Limpopo. Right. That is fossil evidence. So now we are done with fossil evidence. Actually, we are done with paper 2. We've moved. Ngiyadlala. Hayi. Sebashaya izandla bafuna ukulala. All right. Now we come to cultural evidence. But cultural evidence and genetic evidence is explained better in the out of Africa hypothesis. So let's just go straight to out of Africa hypothesis. Right, out of Africa hypothesis. All right, you can tell me, what does the out of Africa hypothesis state? All present day. All modern life forms originated from Africa and migrated to other parts of the world. That is the out of Africa hypothesis. You're free 2 marks. All modern humans originated from Africa and migrated to other parts of the world. Out of Africa. Hypothesis. Right. It states that all modern humans, all the keyword, it's all modern humans originated from Africa. And migrated to other parts of the world. That's out of Africa hypothesis. All modern humans originated from Africa and migrated. All modern humans originated from Africa and migrated to other parts of the world. That is the out of Africa hypothesis. Right. Now, what is the evidence for out of Africa hypothesis? People, this one is very important. It will be there. I feel it. Okay. They will ask you, explain how fossil evidence. GPS signal lost. Okay, sorry. All right. Explain how fossil evidence shows the evidence of out of Africa hypothesis. Right. Number 1. Fossil evidence. Fossil. You are going to say fossils of Ardipithecus were found in Africa only. Ardipithecus found in Africa. Only. Right, that is 2 marks. Fossils of Ardipithecus were found in Africa only. Right, number 2. Fossils of Australopithecus and Homo habilis. Australopithecus and Homo habilis were found in Africa only. 4 marks. Number 3, you are going to say the oldest fossil of Homo sapien. Homo sapien was found in Africa. So this is fossil evidence. Fossils of Ardipithecus were found in Africa only. Fossils of Australopithecus and Homo habilis were found in Africa only. And then the oldest fossil of Homo sapien was found in Africa. So you need to know this fossil evidence by heart. Right, and then now we come to cultural evidence. We are trying to explain that all modern humans originated from Africa and migrated to other parts of the world. Right, so now we have cultural evidence. All right, cultural evidence. Cultural evidence. Right, so now in cultural evidence, we are going to explain how does cultural evidence serve or supports the out of Africa hypothesis. Right, now the use of tools improved human nutrition. And when you look at evolution, the development of tools became more sophisticated as hominids evolve. Right, so the first fossil to ever use tools was found in Africa, and his name was Homo habilis. Right, when we talk about culture, we are talking about the way of life. Right, you are talking about the way of life. Right, so the first man to ever use tools was found in Africa. That is cultural evidence, which means all modern humans really originated from Africa because now tools are used everywhere. Right, then the last one is genetic evidence. Genetic evidence. Right, when we are talking about genetic evidence, we are talking about mitochondrial Eve. Right, so here you are going to say the analysis of the mitochondrial DNA shows that the oldest female was found in Africa, and her name was mitochondrial Eve. Right. So mitochondrial DNA comes from the maternal lineage, right? Right. So when they were analyzing the mutation on the mitochondrial DNA, they found out that the oldest female was found in Africa, and her name was mitochondrial Eve. Right. That is genetic evidence. Right. And so we are done. We are done. Oh, phylogenetic tree. Aye, aye, aye. We must finish now. Right. Shh. Now let's look at few questions and then, yeah. Remember, the warmer the blankets, the colder the future. Yadlala personal. All right. All right. Now let's look at evolution. Question number 1. Question number 1. Tabulate 3 differences between Lamarckism and Darwinism. Right. Let's look at the differences between Lamarckism and Darwinism. People, are we together?
Yes.
Right. So we have Lamarck and then we have Darwin. Right. So the first difference is that when we look at Lamarck, in Lamarck there is no variation. Whereas in Darwin, there is variation. That's number 1. What is the second difference? Or should I give you 5 minutes to do it and then we do corrections? Aye, nah. What's another difference between Lamarck and Darwin? Yeah, so in Lamarck, no, in Lamarck, not all offsprings are affected there. It's only those ones that are determined to change that change. Yeah, so in Lamarck, individual changes, whereas in Darwin, the whole population changes.
So can you also say Lamarck says that um, organisms evolve because they want to, and then Darwin says the environment needs to evolve.
Not necessarily, but what we can say is that in Lamarck, characteristics are acquired, whereas in Darwin, characteristics are inherited. Right, very important. Right, so this is a difference between Lamarckism and Darwinism. Right, again, I'm recapping. The first difference is that in Lamarck there's no variation, in Darwin there's variation. And then in Lamarck, individuals change, whereas in Darwin the whole population changes. And then in Lamarck, characteristics are acquired, and then in Darwin, characteristics are inherited. All right. So we move on to another question. Sorry. Where is this food? All right. Shh. Now the question is, um, are we reading the question? Right. Ancestors of duck did not have webbed feet. Are you seeing it? So the ancestors did not have webbed feet. In terms of natural selection, explain how the webbed feet could have evolved.
Yes.
Explain how the ancestors of ducks did not have webbed feet. In terms of natural selection, explain how the webbed feet could have evolved. All right, let's do it. There's a great deal of variation amongst the ancestors of ducks. Some ducks had webbed feet, and some ducks did not have webbed feet. When there was a change in the environment, the ducks that had webbed feet survived, and the ones that did not have webbed feet, they died. Ne? Right? The ones that survived reproduced and passed on the allele of having webbed feet to their offsprings. Ne? Then eventually, there's a great proportion of ducks that has webbed feet. Yes, yes. Yeah. So here the competition is not stated, ne? But you can state it. You can say maybe the ducks had to swim because these webbed feet they are used to swim. But since here it's not mentioned, I don't think it's compulsory. Right, and then question number 3. Describe how speciation occurs when a population becomes separated by a geographical barrier. So we've done this one, isn't it? Right, if a population of a single species becomes separated by a geographical barrier, the population will split into two. There is now no gene flow between the two populations. Since the two populations are exposed to different environmental conditions, natural selection will take place independently in each population, such that the two populations become different from each other genotypically and phenotypically. Even if they were to mix again, they would not interbreed and produce fertile offspring. All right, question number 4. Study 3 diagrams, diagram A, B, C, and C, below that shows the mechanism used to explain evolution. Right. Okay. Okay, we've seen the diagram. Let's just memorize the diagram. Here's a diagram. Right, here's a diagram. So we have roots. These are roots. Some they are long and some they are short. And then in diagram B, we can see that the short roots, all of them, The trees with short roots or plants with short roots, what happened to them? They died. Are you seeing it? Yeah, they died. All of them, they died. And then in diagram number C, we can see that the new offsprings are developing what? Long roots. This is obviously natural selection, right? So you can see that there's a great deal of variation among the roots of the plants, right? Some plants have long roots and some plants have short roots, right? When there's a change in the environment, can be droughts, the plants with long roots survive because they could reach groundwater or water table, and the plants with short roots, they die because they cannot reach groundwater. They are no longer there. These plants with long roots, they reproduce and pass on the allele of long roots to their offsprings. And then eventually, there's a great proportion of plants that has long roots. They are called cutty plants. All right. Okay, so it says from a comparison, picture A and B, describe the feature of the cats that have enabled them to survive long periods of hot and dry weathers. The answer there is long roots. Right. And then 4.2, name the mechanism put forward by Darwin to explain his theory of evolution that illustrates in the diagram. What is that? Natural selection. Then they say use the three diagrams to explain the mechanism that you've mentioned. Right, I've already explained it, isn't it? Yes. Right, now let's come to question number 5. People, are you following? Or right, they are saying study the diagram below that shows the change in the number of black and white mice living in an environment with light sand. Okay. Shh. Right, study the diagram below that shows the change in the number of black and white mice living in an environment with light colored sand. So the land is light colored. The mice are white, others are black. Obviously, the white ones are going to be camouflaged, isn't it? Yes. The black ones will be exposed. Right. 5.1. What phenomena is illustrated in the diagram? Natural selection. Describe how the sequence of events A, B, and C represent the phenomenon. Right. Let's go back. Right, Stini, there was a great deal of variation amongst the mice. Okay? Some mice were white and some mice were black. Are we together? Right, when the predators came, the white mice survived because they were camouflaged by the light colored sand, and the black mice died because they were exposed by the light colored sand. The mice that survived reproduced and passed on the allele of being white to their next generation. Then eventually, all the mice, oh no, there was a great proportion of mice that was white, isn't it? Yeah. Right. 5.3. Will the next generation of mice population more likely be all white or all black? The answer is all white. Right, question number 6. Study the diagram below showing a process of evolution. Diagram 1, 2, and 3 shows the sequence of events that occurred in the rabbit population. Right. Okay. Okay, so now there's a river here. We know a river is a geographical barrier, isn't it? Right. This river was found on the site. People A. And then there was gene flow, which means these rabbits were able to interbreed, isn't it? And pass on their genes. Right, in diagram 2, I don't know whether that is called river capture or what. Active. Right, or stream pirates. Alright, now listen game. This river started bending downwards. Are you seeing it? Yeah, so it started bending downward and it separated the populations. Now we have population A and population B. Right, in diagram number 3. Right, in diagram number 3, there is now no gene flow. Right, and since there's no gene flow, obviously they cannot interbreed anymore. And they are exposed to different environmental conditions. Natural selection took place independently and they became different from each other. Right, so we ended up with species X and species Y. Right, 6.1. Name the evolutionary process represented by the sequence of events shown in diagram 1, 2, and 3. It's speciation. Speciation. Right, describe the process stated using the diagrams above. So we are going to say right, there was once one population of rabbits. Right, this population of rabbits was separated by the river. The population split into two. We had population A and population B. Since these two populations are exposed to different environmental conditions, did you mention gene flow? No. All right, let's start afresh. Right, we said that there was once one population of rabbits, then this population was separated by the river. The population split into two. We had population A and population B. There was no gene flow between the two populations. And since the two populations were exposed to different environmental conditions, natural selection took place independently in each population such that the two populations became different from each other genotypically and phenotypically. Hence the formation of species X and species Y. Even if these species can mix again, they would not interbreed and produce a fertile offspring. Right, question number 7. Question number 7. Right, the diagram below represent the evolution of horse compiled from a fossil record. Right, so we have a modern horse. And now let's look at their legs and their toes. Now 7.1. How many times is the modern horse taller than the earliest ancestor? Now this is an earliest ancestor. They are saying how many times is the modern horse taller than the earliest ancestor. So you are going to say 1.6 divided by 0.4. Then you're going to get an answer. What is the answer there?
You say it's 1.6 minus 0.4.
Yeah. No, it's 1.6 divided by 0.4.
It's 4.
It's
No, it's 4. So it's 4.
it's 4. Okay, which means the answer is 4 times. So we said 1.6 divided by 0.4. Then you see the answer is 4 times. Right. And then 7.2. What happened to the appearance of the feet of the horses over time? What happened to the appearance of the feet? Right. So now we can see there's feet here. Toes. Eh? And then over time, they started doing what? Shrinking. Isn't it? Right. Paleontologists suggest that the ancestor horses lived in forest, but modern horses lived in open grass. Explain one way in which the modern horse is adapted to open grassland. One way in which the modern horse is adapted to live in a grassland. Here's a sorry. It is used. Eh? What is she saying? Yes, good. It has wolves. Right. Now, question number 8. The last one. Say amen. All right. So the red-bellied snakes and the green tree snakes are predators that sometimes feed on the cantods, no totsmos, frogs, that contain a toxin that may kill them. So these snakes, they feed on the toads that has toxin which may kill them. Right? So the snakes consume the toads by swallowing them whole. So they just eat them whole. A decrease in the average jaw size of the snake has been observed over a period of 70 years. Right? Which means these snakes, the red-bellied ones and the green ones, they're a larger jaw size. So, or rather, maybe there was variation. There were some with a larger jaw size and there were some with a smaller jaw size. Right? The ones with a larger jaw size, they are the ones that swallowed the toads that had toxin. So all those that had a larger jaw size, they did what? They died. All right, let's look at the questions. Okay, the question is define punctuated equilibrium. Can you see how many marks? 3 marks. Define punctuated equilibrium. Right. So we are going to say punctuated equilibrium. It's a hypothesis that explains the speed at which evolution takes place. Then you further explain that it states that evolution involves a long period of time where there is no change and a short period of time where there's rapid change through natural selection. Right. And then 8.2. What characteristic of toad species protect it from the predators? Toxin. Contains toxin. Right. Now 8.3. Explain how the change in jaw size helped the snakes to survive. So they are saying that the snakes consumed the thoughts by swallowing them whole, a decrease. So there was a decrease in the average jaw size of snakes. So how did that save them? The smaller the jaw size. So as the jaw size decreased, it meant that the snakes could no longer swallow the thoughts, isn't it?
Yes.
And that made them, or that protected them from the toxin that is produced by the thoughts. Yeah, because they were swallowing the thoughts as wholes. They were killed by the poison. This is sport. Right now, 8.4. How would Lamarck have explained the development of small jaws in the snakes? All right. So I think Lamarck could have said, okay, so the snakes saw that the thoughts are toxic or they have toxin. Ne? So they did not use, or rather, originally, snakes had a larger or a bigger jaw size. They did not use their jaw size more often to swallow the thoughts. As a result, their jaw size decreased and became smaller. And the characteristics of small jaws was then passed on to the next generation. I just want something that has to do with human evolution, then we are done. Okay, let's do this one. 29, the last one. Last one, ne? Last one. All right. Question 29. It says, Professor Liebega and his colleagues studied the fossils they found in the cave at the cradle of humankind. The skeletons from the cave are ranked among the most complete finds to date. The adult female Australopithecus sediba was remarkably well preserved, and some of the following characteristics were identified. Right, so Australopithecus sediba had a small brain size, was bipedal, smaller canines, projecting nose, and a smaller body size. Right. Question 29.1. Write down 3 characteristics from the list above that also applies to Homo sapien. Right. Which one applies to Homo sapien? 3. Number 1, bipedalism. Number 2, smaller canines. Number 3, no, not small body size, a projecting nose. Our nose, yeah? Yeah, but we have projecting nose. Yeah. Right. Now, state 2 advantages of Ardipithecus sediba. I mean, Australopithecus sediba being bipedal. What are advantages of bipedalism? Can see far. Hands are free to carry tools.
Working is efficient.
But the sexual dimorphology is readily exposed. And movement is easy and efficient. Yeah? The sexual dimorphology. Your genitals. Yes. Right, Professor Becker called Australopithecus sediba a transitional fossil. Using the information in the list, explain why he was called Australopithecus sediba, a species in transition. Want to try? Yes.
So I think because the fossil was the only one that had the characteristics of the African apes and the modern humans.
Yeah, because yet characteristics of the ancestor species and modern humans. Yeah. Are you right here? Yeah. It's your kanjani le nto le?
Said, Baba isang na mag-YouTube sa channel. I can do it if
Search. Okay, ngiyabonga now.
There's no space there. Yeah, there's no space to break it. I know. How much are you getting?
M.Saidi.
Yeah. Thank you so much, guys, for the who have been online. And we thank all the teachers who did a great job today. Yeah. So we'll meet again in another discussion. Don't forget to watch the predictions. All right. See you again. Of life, whereby this topic, what you're supposed to look at is DNA structure of every blood group. Well, blood group O donates. So blood group O is a universal donor. Some people, when they are blood group O, they think their blood is strong, they feel good. No, it's worse. Why? Because if you lose blood, if you need blood, we have to look for only one person with blood group O. But if your blood group AB, you are lucky. Anybody who comes, you can donate that blood to you. So blood group AB becomes a universal recipient. It receives from everybody. While blood group O is a universal donor, only donates to anyone. Someone is asking me that why these crossings are not allowed. The crossings are not allowed here because they are not crossing. They're already gametes. After that, you can draw your crosses here. Ne? So the crossing here, the cross here, no, it's not allowed there. It's not allowed. Crossing here is fine. If you cross them, it's fine. You can either use the Punnett square or you can just do the crossing. All right. Let's go to. We talked about the blood group. I'm coming back with the cloning. Cloning. Or I'll give you one tip of an example of. Okay, let me give you the question, because I just want to go just for 2 or 3 minutes. Yes, 3 minutes. Let me give you just one question for you to practice. Practice for me. Is 6 marks, ne? Yes, this one. It's a simple question. There is something behind it, most especially on this question. I need to see so that you don't lose that mark. Please do that for me, ne? Do that for me. Then after that, I'm gonna do it again for you. What's the time? Is 39. I'm giving you 4 minutes. 4 minutes to finish, ne? I will be back. I will be back. I'm saying I will be back. I hope you are done, eh? Hope you are done. Hope you are done. Okay. Oh, come on, saying yeah, it's done. All right. Anyway, ne? All right. Okay. That's nice. Okay, now let's go through it. They are saying diagram below, okay, controlled by R and R. Check here. The first point is R and W. These are capital letters. The moment they are capital letters, it means that both of them are dominant. All right. Now they're saying red cross with white produce pink. You see, they have 3 phenotypes. The moment you have 3 phenotypes, it means that is incomplete. Dominancy. It is incomplete dominancy. A. The question is here. State the type of dominancy shown in the Snapdragon plant. Here is the incomplete dominancy. Give a reason for your answer, because a third phenotype is given or an intermediate is given. You see, a third phenotype is given or an intermediate is given. Now, if it is complete, if it was complete, ne? I would have been with red and white. I either produce red or white. Which one is dominant? Depending on which one is dominant. And then if I have codominance, red and white, I would have produced a mixture of red and white. But because this one is pink, a third phenotype, therefore, or an intermediate, therefore it is incomplete dominancy. They're saying Gardener crossed 2 pink flowered snapdragon plants using the genetic cross. Show the ratio of the expressed phenotypes in the offsprings. Now, remember I told you that the first thing you need to do is to write P1 F1. But in this case, they're saying they crossed 2 pink. Pink is found on the second generation. Therefore, it won't be P1 F1. It will be P2 F2. In most cases, students lose that mark. Ne? If it is the second generation, then you say P2, F2. If the first generation, then you say P1, F1. Ne? Yes. So now, there you talk about phenotype, which is pink. I told you that this is obtained from the question. Check. They are saying, what are they saying? Check what are they saying. They're saying genetic cross showed a gardener crossed two pink flowered snapdragon plants is always obtained from there. Are you seeing? Then now, so it means that I've got a tick here. I've got a tick there, and also I've got a tick there. So now 2 marks. Now they are saying that pink, remember they say that white is capital W and red is capital W, capital R, white is capital W. Therefore pink is going to be capital W, capital R, capital W, whatever, capital W, capital R, or capital R, capital W. It's the same thing. So it's going to be capital R, capital W crossing with capital R, capital W. But this is the genotype. See, now you get another tick. Then they are saying that meiosis will take place, and then you have capital R, capital W, capital R, capital W. I said don't leave these so-called gametes to be as if they're at the beach. No beach, ne? At the beach, ne? You live naked, ne? You enjoy yourself. Ne? Don't leave them naked. Try to dress them up. Try to dress them what? Try to dress them up. At the beach? So try to dress them up so that, ne? Yeah, they are not there at the waters there when they are enjoying their life. Ne? Try to dress them up so that you separate them. Even if they are too close to one another, you won't lose a mark for that. You won't. Then after that, you do the crossing, or you do a Punnett square. So cross with R, capital R, capital R, capital W, capital R, capital W. Because I started with R, I will start with R, capital W, and then capital W, capital W. Whereby these are offsprings. Those are offsprings. Now, if those are offsprings, what is it? R, this is red, this is pink, and then this is white. Meaning that I will have one red, 1, 2 pink, and then 1, 1 white. There is a mistake where the student do, whereby they ask you the ratio. In this case, you write 1 to 2 to 1. Ne? Some people, when they ask them the ratio, they write expression, and sometimes when they ask you the expression, they write the percentage. So when they ask you the ratio, please write the ratio. If they ask you how many were red, or what is the percentage of individual plants which were red, then you have to say it's 1 out of what is the total. The total is 4 times 100. Then you give us the answer. You give us the answer as 25%. Please don't forget that. All right. Let's continue. And we say our question is, I think I've exhausted that. Pedigree. Let's just look at one question here, which involves the pedigree, and then we can go to our. Oh, there is one question here for blood group. There's one question here for blood group, but I think we exhausted the blood group, but you see that blood group also has 5 marks. Explain why paternity of the boy could not be established using the blood group. Why is paternity test is not able to be established, like to conclude that this is the father of the individual? It's because many people have the same type of blood group. So you can't say that, as I showed you, that is blood group A, married blood group B, and then they produce blood group O, still they produce blood group AB, still they produce blood group B. So whom are you going to say that is not the father, who is, or whom are you going to say that is the father? So it means that you end up losing the what? The mark. All right. Why is it not conclusive? Why is it not conclusive? Because many people have the same type of blood group. But if they ask you, explain why paternity test for this situation, you will say that the mother is blood group O. You see, the mother is blood group O, while the boy is blood group AB, blood group B. So of which the boy can be can be B, heterozygous, or the boy can't be homozygous. Why? Because the mother is II, so gets 1 from the mother. So the genotype of the boy is heterozygous. Now this one can give yes the B, ne? And also this one can give the B. So whom are you going to say is the father? Whom are you going to say is not the father? So that's why in this case blood group can only help you to eliminate those possibilities which cannot happen. For example, a person of blood group, the father can't be blood group A because this person is blood group B. You see? So if it's blood group A, there is no way how the boy is going to have blood group B. So if there is a man, another man who is blood group B, so blood group A, that one is wiped out. You understand? And then another man who is blood group O cannot be. So blood group A and blood group O, they are wiped out. Why? Because if it is small I, the boy is blood group B. So there is no way how this boy is gonna get a B from the father who is blood group O, yet the mother is already known is blood group O. You see? So that's the meaning that you can't use blood group to conclude the father of the individual. Because you are blood group B, you can't say that this is my child because this child of mine is blood group B. There are so many people who have the same type of blood group, and the possibilities of having these are many. I hope you understand me better. Now, how do you write this? It means that this one is only blood group A and blood group O are going to only be wiped out. So the blood group A and blood group O, they cannot be the father. So you get those 2 marks already. And then this blood group, the male A can give this allele for B to the boy. So it can be the father. Or this one also can also give 1 allele for B and gets another one from here. Also can be the what? Can be the father. That's why we cannot conclude using blood group who is the father of this boy. As I repeat that, blood group can only be used to eliminate the possibilities which cannot happen in a situation. Who is the biological father of the boy using the DNA profile? Now you have to check and see. You will see that it is this and this. You will see that is male what? Male 2. Give a reason is because the remaining bands of DNA profile match with the male. The remaining, they must match. If there is one which is not matching, then automatically it's not the father. Might be a relative, but might not be the what? The father. Remember, all the genes that this boy or this baby is having is mother and the father. So if you take out all the mother's genes, because the mother is known, now the remaining bands of this boy must be those ones of the father. Let me show you what I'm trying to say. This mother, okay, this one is not for them. Yes, this one is corresponding. So this one is out. This one corresponds. This thing is now jamming. All right, corresponds. This one corresponds. Oh, this one corresponds. Yes. This one is not matching. So this one is not matching. This one is not matching. This one corresponds, is out. This one is not matching. This one is not matching because this is not. This one is matching. So now all of those ones which have X must match with the male who is the father. That's why I'm saying that the remaining bands of the boy, after those ones of mother have been removed, they must match with the father. Okay, this one, yes, it matches. You see, this one, yes, it matches. You see, this one is matching with this. This one is matching this. That's why we say that individual 2 is the father. Male 2 is the father of the boy, because the remaining bands of the boy have matched with the male, with male 2. All right. Now, let's go to just one example of a pedigree diagram. Yeah, pedigree, where are you? Pedigree, and then I show you how you can use it to answer and also to score a mark. They are saying hemophilia is a that statement is enough. This statement, hemophilia is a sex-linked trait. Hemophilia is a sex-linked what? Trait. Meaning that if it is a sex-linked trait, what is it? It means what does it mean? It means that you're gonna use X and Y. Please, if they don't say is a sex-linked in exam, don't use X and Y. You're gonna get it wrong. Is a sex-linked disorder. Don't use X and Y if it is not mentioned in the question. So here it is mentioned sex-linked. Or sometimes they don't say sex-linked. They say that hemophilia, the gene is carried on X chromosome. Still that is sex-linked. If it's not mentioned, please don't even bother. Don't even waste your time. Don't even waste your time to let me show you here. One of the deafness in human is carried on a single allele. The diagram below shows, done. They are not talking about sex-linked. However much they talk about Paul, and you know that Paul is a boy, Liz is a girl, Mary is a girl, John is a boy. However much you know that, please don't use X and Y. Please don't use X and Y. So if they don't mention is sex-linked, don't use X and Y. If they mention sex-linked, then you talk about X and Y. How many generations are here? There are 3. 1, 2, 3. They are saying that, and then number 2, it can be caused by, something can be caused by a recessive allele, or it can be caused by a dominant allele. Anyway, let's continue with this question. They are saying, state what is represented by a circle or a diagram. I wanted to tell you that sometimes we don't bring that a circle is a male, a box is a male, a circle is a female. Sometimes don't give you that key. You have to go to the exam when you know that. All right. Now they're saying, what are they saying? Okay. Now they are saying, this is a boy, this is a... How many individuals? Let me brainstorm you. This is affected with hemophilia. Don't say he's sick. Is sick of what? Sick of what? Malaria, ne? Sick of what? Sick of hemophilia, or is hemophilic. All right. How many kids are produced by P and Q? They are how many? It's 1, 2, 3, 4, 5. I know many people are gonna say that. It's not. They are not 5. 1, 2, 3, 4, 5. No. This one is an intruder. This one intruder. This one got married this. This one got married this. So there are only 3 kids. How many generations are here? 1, 2, 3. There are 3 generations. You understand? What is the phenotype, the genotype, or the phenotype? They can ask you what is the genotype or phenotype of P or Q, whatever they can ask. But if it is shaded, it means that that individual is affected. Now, they're saying that state what is represented by a circle. We have said that the circle means a male. State the number of general A. We have talked about them. There are 3 generations. 1, 2, 3. Offsprings P and Q. State the number of offspring P. The same thing is this, and this. This one and this one's they are only and only got married to that. So they are intruders. They're saying that let us give the letters of only male who are having hemophilia. Letters of the female. Female is T and the female is U. Those are the only two people who are having hemophilia. Why? Because they are shaded. And then lastly, the genotype of R. The genotype of R. Now, how do we find the genotype of R? Look at the babies or look at the parents. First, look at the babies. It can help you. If you see that the one baby is hemophilic, then automatically the mother is gonna be heterozygous. So the mother is gonna, remember that here you have X and Y. Y doesn't carry anything. Y doesn't carry anything. So it's X and Y. So Y does not carry anything at all. All right. I think now, okay, so since this one is sick, it means that, and is a boy, so it's going to be like this. It's going to be, this is a recessive allele. So if it's a recessive allele, obviously this can't come from the father because the father already gave the what? The Y. Therefore, this came from the mother. So it means that the mother, since is normal but a carrier. So the mother is going to be, or R is going to be capital A, capital X, capital X, capital H, small h. And that becomes the answer for that. Yes. Let's continue now. Let's continue. So we are done with this. Sex determination and biotechnology. How do I explain sex determination? Sex determination is like describing a karyotype. When asked you to describe sex determination, it's like describing a karyotype. But here, if they ask you the genetic cross, you have to do the cross. Ne? So, for example, if it's a cross, you tell us that P1, F1, everything, ne? Male crossing with a female, yeah, and then you tell us that male is X. You tell us the phenotype here, ne? And then you tell us the genotype. Male is X, Y, and then female is XX. Then you cross. Then you tell us meiosis here. You tell us gametes. They come out here. And then I'm gonna explain. I'm gonna show you how to write it if they say using a genetic cross, and then also show you how do you describe if they ask you to describe. Then these are gametes. These are gametes. Then they cross, then this and this, cross Y, and then this and this, they cross like that. So this is the offsprings, meaning that you have 50% chance of having boys, and 50% chance of having girls. What about those who have girls, or boys? Yes, even if you have a boy here, the next time you have 50% of a boy again. Even if you have another boy, the next generation, you have the next reproductive cycle, you're gonna have still 50% chance of having a what? Having a boy. So it depends on how do you roll your dice and how many chances do you have to have a boy. There are 50, but you are just lucky that you have what? Boys, or you are lucky to have what? To have girls. So in this case, we are saying that 50 boys, 50 girls. Yeah. If they ask you to describe, you tell us that individuals, humans are made up of 46 chromosomes. Ne? The 23rd pair is called gonosome. It's either X and X or X and Y. If X and X, if a male sperm with X fertilizes the female ovum, it will result in X and X. The individual will be a female. If a male sperm having Y fertilizes the ovum, remember ovum is always X, the individual will be X and Y, and therefore will be a male. There are 50% chance of having a male and 50% chance of having a female. That's how you can describe sexy linked. So let's finish up the cloning, and then we go to evolution. We finalize it with the evolution. All right. Biotech. Cloning. What is stem cells? What are stem cells? Stem cells, these are cells which have ability to divide to form other body structures. Imagine you are just a sperm. You are just a sperm, ne? Sperm fused with the ovum. You formed a zygote. The zygote, remember this paper one formed a ball of cells, ne? Which called a morula. You see, a morula is a stem cell. Because here, if I ask you, show me the eye, show me the legs, show me the chest, show me the, you can't show me. It's just a ball of cells. So these cells, they have ability to divide again, to form other body structures. You understand? Yeah, you form other body what? Structures. And then, yes, you become, yes, you have the eyes, you have the ears, you have everything. So we call them stem cells. Those are cells which have the ability to divide to form other body structures. All right. Give me 2 examples of stem cells. Umbilical, adipose tissue, bone marrows. So you have to know those stem cells. Sometimes we bring this as an extract. Let me check and see. If I have any question regarding 2 stem cells. This is gene. Calculate the percentage of, okay. This is gene. I skipped something which is very important. That is the dihybrid cross. Yes. Let me leave it there so that I don't forget again. Dihybrid. I want to show you how do you identify the genotype. How do you identify the genotypes? Using the gametes, because that is a very problematic question. Okay. So in this case, I can't find the question there. All right. Now, let's go to cloning. I only want to discuss about cloning here. Ne? Yes? About the what? IA. Cloning. Yes? I have sheep A. I have sheep B. Ne? I get a cell of sheep A from the skin. Let me say somatic. And then this one I get it from ovum. Ne? What I do, I remove the nucleus of this. I go with the nucleus. Ne? I throw away this shell. Well, here, I go with the shell, but I throw away the nucleus. I fuse them using electrical method, and then I form a cell which looks like a zygote. It looks like a zygote. Now, this zygote, I put it into another animal. A foster mother. Animal. Yes? Yes. And then what I do, I produce a lot of these animals. And these animals are identical. Question, why are all these animals identical? They're identical because when this forms a zygote, it forms a ball of cells. Now this ball of cells, now I divide these balls. Now I'll have small balls, small balls, small balls, and then the small balls, I keep on going and then insert them in different other shape. Ne? So it means that because I use the same genetic material, obviously these ones, they're going to be identical. It's because it's the same genetic material which was used. Now, a question. Will these babies look like B or will look like A? They will look like B. Why? Because I use the nucleus of B. I didn't use the nucleus of A. Why did I use the nucleus, the shell or the casing of B? It's because I wanted this to look like a zygote, because what is outside is an ovum. You understand? It's like drinking alcohol in a cup. I won't say that you're drinking alcohol. I will say that you're drinking tea. So because the outside is a casing of the nucleus, therefore this will look like a zygote. And because this one is diploid, 2N, they can ask you how many chromosomes are found in this. Because it's a somatic cell. Somatic cells are always diploid. You see? Somatic cells are always diploid. Someone is asking me to do the part of evolution. I'm coming there. I'm finalizing genetics here. And then I'm coming to evolution. So because this one is what? Is a somatic cell. Therefore, automatically, this is going to be a diploid. And then, because it's brought in the a casing of the ovum, so it's going to look like a what? A zygote. And now I split them in 2 small pieces, and then I insert them in different shape, and each shape will produce identical what? Identical shape. Yes, I think I'm done there. Sorry. I'm done there. All right. Now, lastly on genetics is this question of the dihybrid cross. Identify what is Z. You just have to know. Cross this, and then you write what is Z. What's the genotype of the parents? Now, how do I identify the genotypes of the parent? Genotype of the parent. Look at the gametes. You'll find the genotypes from there. Start with F. For example, you write the F. If it is repeated, leave it. Is there any other small f? Yes, the small f is there. Then go to capital H. Yes. Is there any other small h? Yes, is there. Done. Look at them. If there was no, if all of them were capital H, ne? All of them were capital H, ne? For example, capital H, even here capital H. I would have said, do I have F? Yes. Do I have small f? Yes. Do I have capital H? Yes. Do I have small h? No. They are all capital H. Done. That's how you can identify. Simple, no stress. They're saying that number of the genotype that could result in offspring that is short finger, and short is dominant, ne? And then this one is recessive. Because it's recessive, I need to have with the small h. As long as I have capital H, is 1, is 2. Is 1, is 2, yours, and then yeah, is 3. So there are 3. And then allele that for continuous hairline, allele, not genotype. So it's gonna be small h. The genotype of the child that is heterozygous recessive for both alleles. So is this one, which is small f. Yes. So this dihybrid cross, simple. You should not go to the exam when you don't know anything. Welcome to evolution. Now, evolution. Okay, evolution. Let's look at evolution by natural selection. This one does not have too many questions, which I'm gonna speak about, but it has many questions in human evolution. However, these days we are setting evolution by natural selection than human evolution. I think Mutasi, now we are on you now. Ne? Share the video so that now people can join for evolution. Ne? Because they find a lot of challenges with the evolution. Evolution, what is a hypothesis? What is a theory? You have to go when you know the difference. Ne? Yes. Hypothesis basically is a tentative statement which is subjected to be tested. It can be proven correct or wrong. And then a theory, basically what you need to look at is a statement which has been proven by facts. You understand? Situation which is being proven by facts. So it means that it must have evidences to support it. Then it becomes a what? It becomes a theory. Now, before I go there, I think this one is a little bit clear. The 6-mark questions or 7-mark questions, they are going to come from here. Number 1, theories of evolution. Number 2, that is Lamarckism or Darwinism. Number 2, speciation. Number 3, present day evolution. Now this difference between Lamarck and Darwin, yes, but present day evolution. You can't go to the paper without, because these are six marks. 6 marks each. Yeah, I told you, you go back, you come back and then you tell me what you found there. Ne? Yeah. All right, let's continue. Theories of evolution. Lamarck's theory, Darwin's theory. Lamarck, what you need to know is how to apply it, but I'm just going to talk about it. Ne? I'm not going to take too much time on it. I will spend the last few minutes on evolution, human evolution, but I'm going to explain everything so that you don't lose anything. Lamarck's theory. State 2 laws of Lamarck's theory. It's law of use and disuse. That's number 1. Number 2 is law of inheritance of acquired characteristics. So it means that Lamarck has 2 laws. Number 1, law of use and disuse of a structure. Number 2, law of inheritance of acquired characteristics. Why is Lamarck's theory not accepted scientifically or by scientists in the world? Number 1, there is not enough evidence to support his theory. Number 2, organisms don't change because they want to change. Organisms change not because they want to change. You understand? Yes. For him, he says that if you want to be tall, you just become tall. If you want to be short, you just become short. No, organisms don't change because they want to change. Number 3, what is the difference? You have to know how to use how Lamarck's theory is applied. For example, how did the big elephants with the big trunks came up with the big trunks? Using Lamarck's theory. So meaning that the more you use the structure, the more. At the beginning, all elephants had short trunks. Then you tell us that these elephants, they use their trunks frequently, maybe to defend themselves or to reach the trees, and then their trunks became bigger and longer. They passed their genes. Yes, they produced babies with big, long trunks. But if you are using natural selection, let me just use this as for natural selection. You have to start with, there is variation among the elephants. Those with long trunks and those with short trunks. Those with long trunks, they could survive. Yes? Why? Because they are able to defend themselves. They could reach the tall tree plants. While those with short trunks, they could not survive. They died in large numbers. You understand? This is called natural selection. This is called natural selection. I think. Now, those with big trunks, they pass their genes to the next generation. After many generations, all elephants had big trunks. Let me show you. Let me see if we can get any question here. But you can't go to the exam without knowing that. using sex, show gender in human, how it is. I see, I explained this. I explained this. You see, 6 marks. 6 marks. Okay, when I find it, I'll try to show you that example. Speciation. Now, students confuse speciation with natural selection. What is speciation? Speciation is the formation of new species. Please don't confuse those two. Look, speciation is the formation of new species. What's a species? It's a group of organisms that can interbreed, meaning that they have similar characteristics. Group of organisms with same characteristics that can interbreed to produce a fertile offspring. What is a population? The population is the group of organisms of the same species that can interbreed to produce a fertile offspring. That's it. Now, speciation is the formation of new species. When they talk about speciation, they will bring about, for example, one area, and then this area is divided into 2. You understand? And then after that, if you try to mix them, they will form different organisms. They will form different organisms. They will form different organisms. So now, let me explain to you. You have a population of A, so you have to tell us that there was one there was one population. Yes. Now the population is divided into 2 by a geographical barrier. You have to tell us the geographical barrier. Let me show you, because I saw a question here. Yup. So what is the geographical barrier here? The geographical barrier describes speciation of poto and lemurs. How did they become different species? There was one population of these organisms. There was one population of these animals. Did they talk about the ancestor? If they talked about the ancestor, then you have to speak about it. Scientists believe that poto and lemur share a common ancestor. They didn't mention the ancestor. So you have to tell us that. Don't say that there was one population of poto and lemurs. They have not yet become poto and lemurs. No. So they have not yet become those new species. You have to start with what they started with. There was one population of these animals which shared a common ancestor. Yes. And then what happened? The population was divided into 2 by 2 subpopulation by a geographical barrier. What is the geographical barrier here? Ocean. So you don't need to tell us by geographical barrier. Tell us by the ocean. If it is a river, tell us by the river. Unless the question is just open. And then now, you formed 2 subpopulations. That is east coast of Africa and Madagascar. And then what happens? There is no gene flow. There is no, this one can't come this side. Gene flow means that there is no sex between them. There is no gene flow between these populations. Whereby these 2, they experience different environmental conditions. Are we together? So because they experience different environmental conditions, natural selection occurred differently. Oh, someone is asking me about punctuated equilibrium. I will come there. They formed, I'm going to explain it. Different environmental conditions, therefore natural selection occurred differently. Whereby they became genotypically and phenotypically different. And if you remove the river, if you remove the ocean, they cannot mix to interbreed. Therefore, they formed 2 new species of poto and lemurs. They formed 2 new species of poto and lemurs. You can only talk about these 2 new species. Don't talk about 2 species before you talk about genotypically and phenotypically different. If you talk about it, you're going to lose marks. You're going to lose marks. Even if someone is marking, it will pain, but nothing we can do. You will lose the marks. So what do? Is you only talk about different species after talking about genotypically and phenotypically different. If you don't do that, and you talk about you form 2 different species, that the population was divided into two different species by the ocean, you are gone. You have lost the marks. The paper has killed you. You didn't kill it. I think you understand what I'm saying. Yes. So now, I repeat, let me repeat it for you. There was one population of a common ancestor. One population of common ancestor. The population was divided into two subpopulations of Eastern Coast Africa and Madagascar by what? By the ocean. There is no gene flow between the two subpopulations. You see, they are still populations, not yet new species. Remember, a population is a group of organisms of the same species. And then now, they experience different environmental conditions. Natural selection occurred independently or differently. And then now, they became genotypically and phenotypically different. If you remove the ocean and you let them mix, they cannot interbreed to produce a fertile offspring. Therefore, two new species of Poto and Lemma have formed. They can bring when they are more than two or more than three. So in this case, you tell us three new species have formed. So don't stack on two new species. Don't stack on one new species. So two new species. So I'm coming for punctuated equilibrium. Charlotte, yes, you are saying hi. Are you still here? All right. Now, let's go to punctuated equilibrium before I go to present day evolution. Oh, let me just finish the present day evolution and then I'll talk about the punctuated equilibrium. If I forget, you remind me. Present day evolution, it deals with the bacteria, virus, black peppery moth. We are talking about present day evolution. Evolution which is taking place, which we see. You understand? DDT on mosquitoes. Yes. So, you know, if they are bacteria, it means that you're going to use antibiotics. Use the same principle of natural selection. Please don't confuse natural selection with the speciation. So in this case, you will say that there was one population of bacteria. These bacteria are separated by the variation which is there. So now when you're explaining, you say that there was one population, you say that there was variation among these bacteria. Those which are resistant to antibiotics and those which are not resistant to antibiotics. Then now tell us that the bacteria which are resistant to antibiotics, they survive on the application of the antibiotics. While those which are not resistant to antibiotics, they die. Those which are this is called natural selection. Those which are resistant, they pass their genes to the next generation. So the next generation will have more bacteria which are resistant. And after many generations, the antibiotics will become ineffective to bacteria. Let me repeat. There is variation among the bacteria. Those you can use for mosquitoes about DDT. You can use for virus about the antiviral or ARVs. There was one. There is variation among the bacteria. Those which are resistant, you have to tell us the variation. If you don't tell us the variation, you lose the mark. There was variation among the bacteria. Those which are resistant and those which are not resistant. Those which are resistant and those which are not resistant to antibiotics. Those which are resistant to antibiotics, they survive on the application of the antibiotics. While those which are not resistant, they die in large number on application of antibiotics. Those which are resistant, they pass their genes to the next generation. After many generations, the bacteria are going to become ineffective to antibiotics. Simple. There is no way how you're gonna skip. Oh, they're gonna bring a lot of things which you don't know here. Never. What is the difference between artificial selection and natural selection? And what is the difference between Darwinism and Lamarckism? Difference between artificial selection and natural selection. You know that artificial selection, what is basically on artificial selection is what is the selective force? First ask yourself, what is the selective force? Selective force in artificial selection is human, while in natural selection, the selective force is nature, is the selective force. You understand? Yes. And then Lamarckism and Darwinism, the major difference is Lamarckism, organisms change because they want to change. But in Darwinism, organisms change due to the environment. Yes. Then number two, in Lamarckism, individual in the population change. Like if me, I want to be big, I become big. If I want to be small, I become small. Not the whole population. But in Darwinism, only the whole population change. Remember, when Corona came, if you want or not, they say that you have to put on a mask. Natural selection. You don't put on a mask, you die. Whether you want or not, you have to do so. You understand? Therefore, that is natural selection. Is it clear? So, in Lamarckism, individual in a population change. While the Darwinism, only the whole population will change. So you can also talk about other differences between the two. Let me talk about punctuated equilibrium. And then after that, I go to human evolution. Someone is asking me, how do I draw a pie chart? How do I draw a pie chart? I'll go to the last question. That is the last question, question 3. The pie chart, how do you draw this at the end of human evolution? So that just I show you how do I identify independent variable, dependent variable. How do I get it from the question? How do I know the control variable? How do I know the conditions which are supposed to be put in place before you start? All right. Punctuated equilibrium. It is very simple. Gradualism versus punctuated equilibrium is gradualism versus punctuated equilibrium is saying like this. You have two scenarios. Punctuate, gradual, when you say gradual, it means that it's slow. The change is slow. While punctuated equilibrium, the change is fast. You have periods of no change and periods of change. The change is rapid. Periods of no change, periods of change, periods of no change, periods of change. This is punctuated. Punctuated. What this is gradualism. Here you require a lot of time. A lot of time. Here it occurs in short periods. So punctuated equilibrium, you have periods of no change, period of change, period of change, period of change. And in most cases, the punctuated equilibrium is caused as a result of mutation. While this one is changed, is caused as a result of natural selection. Yes. Natural selection here, yes. Well, here mutation. Why? Because you just wake up in the morning and then you have something different which you didn't have before. Mutation maybe occurred. So that is punctuated equilibrium, or the other side is gradualism. This marks the beginning of the end of evolution by natural selection. Let's kill the human evolution now. Human evolution. Human evolution, guys, is the most important part where students sometimes lose marks is a phylogenetic tree. I don't know why, but we'll talk about it. Someone is asking, do you do calculation for pie chart? Yes, you do the calculation for pie chart before anything else, because you can't do percentage in a pie chart because the circle is not in percentage. All right, let me just see if I can find here a question. The first question in human evolution can be, to which family do humans belong? To which family do humans belong? They belong to family Hominidae. Hominidae. Differences between, let me tell you what are the short questions, guys, in human evolution. The short questions which you're gonna find, and then you have to give the answers. 6 marks. Number 1, it is out of Africa hypothesis. That's number 1. Number 2 is, so here you have to tell us the evidences. Evidences of that is fossil. You have to tell us the mitochondrial, DNA. Sometimes you call it genetics. Then you have to tell us cultural evidence. And then they can also talk about the differences between humans and apes. Yes. And then also they can talk about similarities between apes and humans. There is no 6 question mark they can bring here. Nothing. I repeat, nothing. Unless it's gonna come from heaven. Okay, now let me explain. There are some things which we changed a bit, and you have to know. That's why if you check my book of distinction material, you'll find out that it's always updated each and every year. It's not static. It's always a version for that year. There will be a version for 2023. All right. Now let's continue. They're saying that, now look at this. Position of foramen magnum. They're saying, which diagram represents the skull? There is a difference between a skull and a cranium. A skull is everything which is encloses the head. That's a skull. But a cranium is only the part which encloses the brain. Only the part which encloses the brain is what you call cranium. But a skull is everything which encloses the what? The brain. I think I'm clear there. Number 2. Number 2, they're saying that, which diagram represents the skull of a bipedal? Now we have 3 factors which 3 factors which affect or which do the bipedalism, of which they can ask you the advantages of bipedalism. You can cipher, you can hold the organisms, you can hold the objects. Sexual dimorphism is exposed. You can see the sex organs. They can be shown because you are walking, yes. And then the body is lifted. You lift the body out of the ground, yes. So it means that less heat is gonna be reflected to your body. There is small surface area for heating of your body from the upper side. So those are some of the advantages of bipedalism. But now, what are the factors which contribute to bipedalism? There are 3. The first one. Why is this thing doesn't want me to write? I will write even if doesn't want me to write. Okay. Number 1 is shape. You look at the shape of the spine. Shape of the spine is what? Is shape. And then you look at the pelvis. Pelvis is short and wide. And then number 3, these are factors which affect bipedalism, short and wide. is wide, ne? And short. So if you look at it, is wide and short. And then the third one is the third one is the position of foramen magnum. It is in forward position. Ne? Yes. Now before I go to explain this, what is bipedalism? We changed this. You don't just say that bipedalism is walking on two limbs. Which limbs? Hind limb or forward limbs? You have to tell us walking on two hind limbs, or walking on two lower limbs, or walking on two feet. But we prefer walking on two hind limbs. If you're using limbs, it must have the word hind limbs. So how is the shape of the spine able to for bipedalism? Shape here is S shape to allow flexibility. That's number one. And then pelvic girdle is short and wider to support the upper weight. Position of foramen magnum is in forward position to allow the spine connect vertically. So basically, that's what you are supposed to tell us. Then they are saying that the next question is tabulate visible. They are saying visible. Don't bring about anything else. Visible difference between the upper jaw and the lower jaw of A and B. Check upper jaw, lower jaw. They're only looking for this. You see, in most cases now when we bring these, you have to know the differences. So in this case, you talk about the canines here are well developed. They are not well developed. There is one student who is saying that this one has canines, this one has no canines. No, the canines are there, but these are well developed, then this one are less developed. This one is rectangular shape, the jaw is rectangular shape, while this one, the jaw is what? Is C shape or U shape, C shape. Then if you look at here, you have spaces between the teeth. There are spaces between the teeth, large spaces between the teeth. Here, no space between the teeth, or small space between the what? Between the teeth. Now, they can ask you, now I'm gonna start firing you questions. How is the brain size related to intelligence? How is the brain size related to intelligence? The brain size related to intelligence, or how is the skull related to intelligence? How is the brain size related to intelligence? If the skull is big, it is going to accommodate more brain cells. The higher the brain cells, the higher the intelligence. If the brain size is big, you have more brain cells, and then this is linked to intelligence. That's why the previous organisms, for example, Ardipithecus, they had a small skull, therefore they had a small brain capacity, therefore the intelligence was also small compared to us. And when you are explaining this, when you're explaining the brain, there is something which we are looking for. If you don't mention it, you don't get the mark. The brain of human is not bigger than the brain of the elephant. It's only bigger than the brain of the elephant if you are comparing it with the body size. If we have a big brain compared to the body size, compared to the elephant. So when you are comparing, when you're talking about the brain or the cranium, you have to talk about compared to the body size. Please, when you talk about cranium, comparing them, then you compare it with the body size. I think I'm clear there. Who discovered Homo sapiens? You have to know who discovered it. I think I should drop. We have a short video which is talking about it. Who discovered Homo sapiens? Tim White. Oh, who discovered Ardipithecus? Tim White. Tim White is Australopithecus. Sorry, is the Homo sapiens. Now, who discovered humans? Is Tim White. Yet is also Homo sapiens. He discovered himself. No, he discovered the oldest skull of Homo sapiens. Is it clear? Yes. So you need to know, don't go to the paper without knowing 3 species discovered in South Africa. South Africa, at least 3. Don't go without knowing the 3 species which belong to Australopithecus africanus. Please don't. Which is Mrs. Place, Taum, Child, and then Little Foot. These are the 3 Australopithecus africanus discovered in Africa. What is the transitional fossil? A transitional fossil is that fossil which has the characteristics of the ancestor and the next generation, or the children. It means that it has not got extinct. It's still in transition. You understand? Which one of the following is a transitional species? In most cases, we look at Karabo. Australopithecus sediba is the transitional species between Australopithecus africanus and Homo habilis. So that is the only transitional species or fossil we talk about. However, even Homo naledi is also a transitional what? A transitional fossil or species. Now, question. Give the difference between the skull of humans and other primates. There is something which I want to talk about prognanthus, because when you are marking that one, humans, prognanthus means protruding jaws. So the other primates, they are prognanthus. Humans are not prognanthus. Don't say they are less prognanthus. No, if you have it in your book, yes, I know it's there, but we will not give you a tick. Humans are not prognanthus. Protruding, ne? Yes. While the other ones are prognanthus. Flat face, sloping face. Those ones are fine. Foramen magnum in forward position. We don't want anything else. Forward position. Foramen magnum in backward position. Don't tell us that foramen magnum is in the center. Center where? Foramen magnum is in forward position. Foramen magnum is in a backward position. Those are the things which changed, and then you needed to know this before you go for your paper. Because if you make a mistake and you mess them up, you're gonna lose a mark. Yet you knew the answer. It's just to rectify those few small mistakes. Lastly, describe, state the out of Africa hypothesis. State the out of Africa hypothesis. We say that modern humans, the word modern must be there. Modern humans originated from Africa. Modern humans originated from Africa and migrated the rest of the world. Originated from Africa and migrated. If you don't write the word modern, it's gone. The mark is gone. Give 3 evidences to support the out of Africa hypothesis. That is fossil evidence, mitochondrial DNA, and cultural evidence. Now, remember I said that this can come in this 6 mark. Yes? So how do I use cultural evidence? How do I use the mitochondrial DNA? How do I use fossil evidence to do this? Now, fossil evidence, you need to talk about few aspects. You have to talk about Adipithecus. You have to talk about Australopithecus. You have to talk about Homo habilis. You have to talk about Homo erectus. You have to talk about Homo sapiens. This is what you're supposed to talk about. Tell us that Adipithecus and then Australopithecus and Homo habilis only found in Africa. I think. Then when you reach here, you say that oldest, and also when you reach here, you say oldest. fossil of Homo erectus and Homo sapiens were found in or discovered in Africa. I repeat, the fossil of Ardipithecus, Australopithecus, and Homo habilis were only discovered in Africa. Only in Africa. Fossil of Homo erectus and Homo sapiens, the oldest fossils were discovered in Africa. That's how you're supposed to write the answer. Now, what about mitochondrial DNA? Here in mitochondrial DNA, we use the mutations which occurs in a mitochondria. Why do we use mitochondrial DNA? Because it has a small DNA, so it is easy to focus on it. But it's rare to find mutations there. So if the mutation occurs, we can use it to trace the lines of what? Descent. And if we trace them, it brings back to Africa. That's why it's called the mitochondrial Eve. And then cultural evidence, you talk about the oldest tools were discovered in Africa. And those are called the Odowan tools. Odowan tools. Yes. As I'm winding up with the today's class, so that you go and get your distinction, please, if it requires you to repeat this, to rewatch it, I'm telling you, I've tried to discuss each and everything which you're gonna find in the paper. You understand? If I've forgotten something, it might be only few, but you have 90%. I'm saying 90%, meaning that, what does it mean? It means that over 130 marks, you have the marks. All right. Let me conclude with the pedigree. Then I will just talk about few of the experimental question, and then lastly I'll go to the terminologies. Just only talk about the terminologies, and then we conclude for this class. I thank everybody who is attending, who is in my class, and I wish you the best. You must get a distinction. Let's try to finish up. Now, pedigree. Not pedigree. Phylogenetic tree. Before I go to phylogenetic tree, how do you write the scientific name? I think phylogenetic tree I need to use a diagram here. Let me just put out the diagram here, which I can use. Okay. All right. Let me use this. It's fine. Let me check if I have any phylogenetic tree here. This is the thing I was trying to explain before. Describe one other structure difference by pedo and quadrupedal. You can talk about. Oh, okay. I gave the difference, ne? Short and wide, the factors which affect that. Okay. You have this now because I tried to explain it. Okay, let me use this. I'm gonna tell you all the questions which can come here. How many genera are indicated here? How many genera? Genera comes from the word genus. How many genera? Here is it's getting to 6 o'clock. To 6 o'clock. I don't know whether the electricity is going this side. Ne? Just in case that it has gone, at least you have everything. Ne? Yes. When you see me offline without saying bye bye, it means that it has gone because I didn't check anything. Describe the process of natural selection. Now you see this one. You just explain. You just explain. You just explain. Now I explained it. You don't use any example, ne? Yes. Okay. Now, name. How many genera are indicated here? Genera is genus if it's one. Genera when there are many. You count the first name, but you don't repeat them. For example, 1 Australopithecus, Homo 2. Do I have any other? Nothing. So it means that I have only 2 genera there. To which family do humans belong? To which family do all, to name the family to which all the represented organisms belong? To which family do we belong? We belong to Hominidae. Yes. Then they are saying describe the cultural evidence used as a tool. You see, I explained it. Yeah, oldest tools are old ones. Ne? So it means that how does it support the out of Africa hypothesis? It supports supports the theory of human evolution. Simple tools were discovered with the simple skulls, and then which means more advanced tools with the more or larger skulls, which means that they had the ability to modify these tools and they were found in the upper layers. How long did okay, now let me start. How long ago did most of the how long ago did the most recent common ancestor of this and this existed on earth? Now when they say how long ago, ne? For example, did the Homo ergaster existed? You come here. For example, if this is Homo ergaster, I come and draw where it appeared and where it got extinct. So it means that I will say 2 minus 1, which means how long ago? No. How long ago? How long ago did they appear? This is how they appeared. It means that it's gonna be 2 million years. How long ago did they extinct? It's gonna be here. So it's gonna be 1 million years. How long did they exist? How long did they exist? That's a question. It means that I'm gonna say where they became appeared and where they got extinct, and then I say 2 minus 1, I get 1. But here is gonna be million years. It's not going to be million years ago. It's gonna be million years. I think I'm clear there. Here you say million years ago, here you say million years. Sometimes we always mark this unit. Then now they are saying which species first used the tools. When they ask you which species, if it is not indicated here, it is Homo habilis. That's why it's called handyman. Which species first walked on 2 limbs? If they ask you species which first walked on 2 limbs, if it's not indicated here, it's gonna be Homo erectus. I repeat, Homo erectus. Then, which species are competed for resources? In this case, you'll find out that if this species and this species, they exist at the same time. If you draw a line like this, you see that this one and this one, they touch each other. It means that they exist at the same time. It means that they competed for resources. So maybe you'll say that Homo ergaster and Homo habilis. So it depends on the species which existed at the same time. But it must be having a line where they touch each other. Not exactly, but they must have a line where they touch each other. Then now, they are saying, what are they saying? They are saying, explain the possible reason why Homo ergaster was placed between Australopithecus afarensis and the Homo. It means that this is a transitional fossil. Ne? Yeah, it's a transitional. Is a transitional fossil, meaning that it has the ability, has the characteristics of this organism, and then it has also characteristics for this. All right, explain the possible reason why Homo ergaster. Where is Homo ergaster? Homo ergaster is here, ne? And then Australopithecus afarensis is here, and then Neander is here. So it means that this is a transitional fossil. Yes, it has this one. Homo ergaster has some characteristics of this and also some characteristics of that. Because it didn't get extinct, therefore it's still going. Therefore it is a transitional fossil. How is the brain size compared to intelligence? I explained that. There's no questions they're gonna bring about this and then you can't answer. Yes. Lastly, how do I identify independent variable and dependent variable? Those who attended my paper 1 class, I think you know exactly. Just write ID. ID. Ne? And then after that, X, Y. I think. After that, you tell us what is ID. Independent variable, not identity card. Independent variable, independent variable on x-axis. So when you're drawing, independent is is x-axis is here. So independent variable must be here. It must have units, please. Then this one, dependent variable, is this y-axis. So dependent variable is here. And then now, please put the title. It must have both the independent and the dependent. Put units here, please. Don't forget about that. And then now, because if you forget, you lose the marks. Scale, please. Don't say 2, 4, 7, 8, 10. No, the range is not the same. Here is 2, here is 3, here. You have to use the same scale. 2, 4, 6, 8, 10. You understand? If you miss them, you lose this and this. And then, now, the problem, now you have got to know that independent is x, dependent on y. But how do I identify them from the table? In most cases, the first column, however much, how many columns you have, is I, D, still. The first column in most cases is independent. And the second column is dependent. Please don't mess up. Yes? The second and the third is always dependent. So now, how do I plot? How do I plot and how do I draw a pie chart here? If you are drawing a pie chart, please convert this into degrees. For example, if I have maybe 10, 20, 20-ish. Not technology. 20, 30. For example, this is Africa, Asia, maybe Europe. Then they say, so to put this on a pie chart, you have to say 10 plus 20 plus 30. So it gives me, this is 50, 60, 60. That's the total. So to do this, I'll have to say 10 out of 60 times 360, because this is the pie chart, pie chart, which is 360. So it give what you get here, you do that for 20, you do that for 30, then you plot. Protractor measure for this, then you change the protractor, start from here now, measure, then the remaining one will be for that. Then you put the title, then you shade whatever you shade, you put a key here, you get the answer, get the marks. So please don't mess it up. And then now, that is a pie chart. But now there is one for line graph. If it is on the same axis, and then the bar graph, it is if it is a bar graph, yes. For example, you have 20, 30, 40. Ne? Bar graph, you plot this and this, you get that and that. Then plot this and this. Then after that, give a space. Ne? Plot this and this, maybe 20. And then plot this and this, maybe 50. Then a space. Now, these bars must be the same. If this bar is not the same as this, you lose a mark. If this bar is not the size must be the same. These spaces must be the same. Ne? If those spaces are not the same, you lose a mark. That's how kids lose mark with these graphs. Then, if it is a line graph, it is a little bit different. Because a line graph, you have to first plot one by one. Line graph, look. Line graph, this and this. But in most cases, to get a line graph, you have to be with the figures. Maybe 10, 30, 40. So that this and this, you plot, you get a tick there. This and this, you plot, you get a tick or a spot there. This and this, you plot, you join. Then if it is two graphs at the same time, now this and this now. So you first finish one column and another column, but independent must be kept. Now this and this, then you plot. This and this, then you plot. This and this, then you plot. Yes. Now, how do I know that my graph is wrong? We don't have a graph which runs like that. Some people have, it means that you swapped the axis. If you still have time, redraw it. We don't have a graph which runs like that. No, this is not math. Yes. So the graph must either be straight, going, if it's going up, it must form a wave, or it's going down, but this so-called parabola, what? No. We don't have them. Once you see that, you just know that you have swapped the axis. It means that what was X must be Y, what was Y must be what? Must be X. Please take note of that. Last of life, whereby this topic, what you're supposed to look at is DNA structure. Describe the structure of DNA. That's a question. Describe the structure of DNA. In this case, what are you supposed to look at? I'm going to tell you. Number 2, you know that DNA replication, yes. Transcription, translation, yes. These ones, you know them. It's easy to get them. But now, the most important part here is the structure of DNA, because sometimes people neglect that. You know how to describe DNA replication. Structure of DNA, how do you describe? Just in case it has come. First of all, you know that DNA is made up of a simple structure called nucleotides. If they ask you what is the monomer of DNA, you have to know that monomer of DNA is a nucleotide. And these nucleotides, yes, they have different parts, whereby this is a phosphate, this is a sugar, and this is a nitrogenous base. This is a phosphate, this is a sugar, is a nitrogenous base. Remember, this side you also have the same kind of structure. Yes? If you have the same structure this side, it means that if they ask you to describe, you have to tell us that DNA is made up of a nucleotide. Yes? These nucleotides are made up of phosphate. Nucleotide is a mark. Nucleotide is made up of phosphate, sugar, that is deoxyribose sugar, and nitrogenous bases is also another mark. Then you mention the nitrogenous bases. What are those nitrogenous bases? Then you tell us is adenine, is thymine, is guanine, and then is cytosine. You mentioning this is another mark. Then you tell us that it is made up of 2 strands. Yes, these strands, you telling us strands is another mark. These strands are joined together by weak hydrogen bonds. These weak hydrogen bonds is also another mark, weak hydrogen bonds. And then it twists around to form a double helix. Double helix is also another mark. There is only one mark I've not spoken about of you mentioning the bases, but also saying that whereby adenine goes with the thymine, guanine goes with the cytosine is also another mark. You mentioning that adenine, thymine, guanine, cytosine is a tick. Adenine with thymine, guanine with cytosine is also another tick. So that is DNA. So all these, if you count the marks which are there are 8 marks, but we only need a few of them. And those few of them, what is it? We only need 6 of them. So don't go to the paper without knowing to describe the structure of DNA, because it belongs to 6 marks. DNA replication. Here you have to describe the structure of DNA. DNA replication, whereby you tell us that DNA double helix is unwinds, weak hydrogen bond, break, yes. Unzip and the breaking of weak hydrogen bond is 1 mark. We don't give you 2 marks there, is 1 mark. Then to form 2 separate, let me repeat it. The word separate must be there. To form 2 separate strands. Each strand is acting as a template. It's Zoleka, I think you are hearing me. Ne? Yeah, I'm just looking at the people who are live, so that if you have any challenge, let me know in the comment section. I will answer it immediately. All right. Saying 2 separate strands is another mark. The 2 strands, they will act as a template. They will act as template. And then using free floating DNA nucleotide. We use DNA nucleotide to form a complementary strand whereby adenine goes with thymine, guanine goes with cytosine. I see. Then you form this DNA with one original strand and one new strand, which are genetically identical. That's how you can describe DNA replication. But we have some few questions. I'm gonna show you these other questions because this is a 6-mark question. Just in case, remember your paper, it has almost 45 marks full of this short question. In each section, we bring at least one, of which this, at least one will come. All right. Then, transcription. Transcription must also know it, that DNA double helix unwind, weak hydrogen bond, break to form 2 separate strands. Now, one strand acts as a template. You have to know that. One strand acts as a template to form the messenger RNA using free floating messenger RNA nucleotide. In this case, we are using free floating messenger. For the case of DNA replication here, here we are talking about transcription. But when you're talking about DNA replication, 2 strands act as a template, while in transcription, 1 strand is acting as a template. Then, whereby adenine goes with uracil, adenine goes with uracil, and then guanine goes with cytosine. Yes? Then this messenger RNA moves out of the nucleus via the nuclear pore to the ribosome. So, that's once you write it like that, obviously in these questions we always have questions or marks which are excess. So we are looking for only 5 to 6. Transcription is done. What about translation? In translation, depending on what you have memorized, but I'm going to show you the marking point in each. If you skip, you have lost a mark. According to messenger RNA codons, codon is a mark. The transfer RNA with the complementary anticodon, anticodon is a mark. Complementary anticodon is also another mark. Brings the required amino acid. It brings the required amino acid to the ribosome. The required amino acid is also another mark to the ribosome. Ribosome is another mark. To form, the amino acids are joined together. The amino acids are joined together by peptide bond to form a required protein. That's it. Nothing else. According to the messenger RNA codons, the transfer RNA with the complementary anticodons bring the required amino acid to the ribosome. The amino acids are joined together by peptide bond to form a required protein. DNA code of life is done in terms of the 6 marks. So it means that you have at least one question which is most likely to come. Now listen, I'm gonna show you now these other questions which might come, or which you're gonna find in your paper. Listen. Before I go there, students make a mistake and they call peptide bond, they call them polypeptide. This is dumb. This is wrong. Polypeptide bond. We don't have this. We call it peptide bonds. We don't have polypeptide bond. Please don't call them polypeptide bond. We call them peptide bond. And then I'm going to show you also another mistake where students make. They call a polypeptide chain polypeptide chain a protein. It is only a polypeptide chain if it is less than 50 amino acids. 50 and above amino acids, we call them protein. It's only a protein when it's 50 amino acid and above. I hope you are getting it right. Okay. What we are saying here.
Recording in progress.
Sorry about that. Ne? All right. We are saying that. Look at the questions where other questions. DNA replication. Why does it occur? To increase the chromosome number. Why does it occur? To increase the chromosome number. Where does it occur? It occurs in the nucleus. When does it occur? It occurs during interphase. I think. How does it occur? Is the process which you have described. So DNA replication is done. Who discovered the structure of DNA? James Watson and Francis Crick. James Watson, Francis Crick. Another question. What is the difference between DNA nucleotide and RNA nucleotide? What is the difference between DNA nucleotide and RNA nucleotide? They are not asking what is the difference between DNA and RNA. They are looking for only the nucleotide of DNA and the nucleotide of RNA. So in this case, you talk about DNA has deoxyribose sugar, RNA has ribose sugar. DNA has DNA nucleotide has thymine, RNA has uracil. We don't have any other. That's it. Ne? Someone is asking me that I should repeat the structure of DNA. Describe the structure of DNA. Yes, I'm going to go to even genetics. I'm going to go even to evolution. I'm going to show you the whole paper, what you expect, and within these 2 hours, you'll be able to write the paper. Ne? You write the paper within 1 hour. So the discussion we're going to do within these 2 hours, you'll be able to write this paper within 1 hour. Meaning that nothing we're going to miss. Okay. So now as I'm winding up, what is the difference between the difference between DNA and RNA? Those ones you know. But there is one question they can ask you. What is the difference between transcription and transcription and DNA replication? Transcription, here you form one strand will act as a template, while DNA replication, the 2 strands will act as a template. I think. In transcription, yes, we use uracil, adenine, thymine, guanine, uracil. While in translation we use adenine, in transcription we use adenine, guanine, cytosine, uracil for the new strand which is formed, meaning that we form a messenger RNA. While on DNA replication we form DNA. Then in transcription, yes, you'll find out that in transcription and DNA replication, we have also some few similarities, whereby DNA double helix unwind, also in transcription, DNA double helix unwind. Weak hydrogen bond break, also decide weak hydrogen bond break. If just in case they are looking for the similarities there, you are able to answer that. Now, as I'm winding up this DNA, there is a question which is always a challenge to students. What is that question? That question is about the genes. The gene mutation. Gene mutation. Now, let me give you an example. If you have A, G, C, and this is codes for leucine, and you have A, C, G here, and this codes for tryptophan, yes, And then you have A, C, G, and this also codes for leucine. Then they ask you, how will the protein be affected if this is changed to this? Because you form a different amino acid, the protein functioning or the functioning of the protein will change. So it means that you have to tell us that when AGC is changed to ACG, tryptophan will be added to the protein instead of leucine or the polypeptide chain. This leads to a different protein or the different functioning of a protein. You mentioning this and mentioning this is a tick, and also mentioning different protein is also another tick, because this question is always 3 marks. What about if this with this? This G changes this and this changes to this, even if it is 1. But it is the same amino acid. This means that the functioning of the protein will not change. However much the base sequence has changed, but what is formed codes for the same amino acid. Therefore, it means that you will find out that it is exactly the same protein. The functioning will not change. It's like someone saying that, for example, let me just use an example here. Someone can call you Sibusiso. Someone, you will say yes, sir. Or can call you Ntuli. Still you say yes, sir. I think. Meaning that is the same kind of person. But if this, because this is leucine and this is leucine. What if someone calls Sibusiso, and then the next time the next caller calls you Dobe? Obviously, Sibusiso will not reply. No, Sibusiso will not reply. It's Dube who will say that, yes, sir. Because now it is different protein, the functioning will change. But it's the same protein, the function will remain the same. So, I repeat, I've concluded with DNA. We will only look at it when you're talking about the questions. So here you're saying that DNA, structure, replication, transcription, translation. Please don't go without knowing it. And this gene mutation of DNA. When you talk about DNA profiling, that one is a little bit easy. Please just read the advantages, disadvantages, and sometimes the trick which is there is they tell you that other than that what is mentioned in the question, give another, and you find the student giving the same answer in that question. So it means that you lose a mark. Let's check and see here. I'm not going to go through objectives, but I'll go through the terminologies because I love the terminologies. Here is the question of DNA. Let me just reduce it a bit. Now, fast. Now, this is just a simple question. What is X? In most cases, people say that X is a cell membrane. X is not a cell membrane. X is a nuclear membrane because the cell membrane is out. Nuclear membrane, this is a nucleus, but they can ask you the process. What is this? This is translation. Once you see something like this, it is transferrin A, this is a amino acid. But if they put dots there, this is not a protein, it's a polypeptide chain, because I said that polypeptide chain is less than 50 amino acid. Above 50 amino acid, it is a protein. Since this is just 1, 2, 3, 4, 5, only 5 amino acid, therefore that is a polypeptide chain. And then what is this? This is messenger RNA, and then which organelle is this? Is a ribosome. Now, question can come. How many bases, how many nucleotides, how many codes code for amino acid? So, let's answer that. How many bases code for an amino acid? Amino acid, remember, an amino acid, yes. An amino acid, yes. it's always having 3 bases because of this 1, 2, 3. So if how many bases is 1, 2, 3. But they're saying that if there are 3 amino acids, how many bases are there? So it's going to be 1, 2, 3 times 3, which gives you 9. If I have 9 bases, how many codons are there? Remember, each 1, 2, 3 is one codon. So how many codons are in 9 bases? So I will say that divide by 3. Therefore, how many codons? Codons will be also 3. I think you have to know when to divide and when to multiply. Yeah. Lastly, on this, they can also give you a question that they can ask you a question that look. They can ask you a question that 20% is adenine. What is the percentage of guanine? So you have to know this. Ne? If 20%, you have to know that A goes with T, C goes with G. Ne? C goes with what? C goes with G. So if A is 20, this one is also what? 20. So 20 plus 20 is equal to 40. Ne? So now, because this percentage is out of 100, therefore I'm going to say 100 minus 40. So it gives me 60. I think if it is 60, what is really happening there? 60 is going to be for these two. So divide by 2. So it's going to be 30 is going to be 30. So it means that the percentage of guanine is going to be 30%. Don't go to the paper without knowing how to calculate the base. Ne? Yeah. I'm using these as examples. So we are saying that which organelle is X? We have seen organelle X is nucleus. We have seen it. Let me do like this. Is nucleus. And then which molecule is Z? Which molecule is Z? Where is Z? Where Z is messenger RNA. Don't just say RNA. It's messenger RNA. State 2 locations in the locations of DNA in the cell other than the nucleus in the cell. So it means that you have to talk about number 1, mitochondria and chloroplast. Yes, mitochondria and chloroplast. Describe process W. W is we don't have any other process occurring in the nucleus except transcription. So you have to describe transcription. I've exhausted most of the questions you're gonna find in DNA Code of Life. It's only one last question. Let me yes, this is the question, how to interpret this. And however, I've tried to explain something here. The trick behind here is one. How? The trick which is here, please check and see what is there. If they say this is DNA and they gave you DNA at the beginning, then you don't need to change. If they say they give you DNA and what is here is transfer RNA, you have to change it to messenger RNA and you have to change it to transfer RNA. Are you seeing? So, in this case, they are saying that name the DNA-based triplet code for serine. Now this is transfer RNA. Serine is here. So I'm going to say is UCG. This is transfer RNA. Convert it to messenger, which is going to be, yes, AGC. Then convert it to DNA because they want DNA. How many times do you convert? It depends on what they have given you. So it's going to be, yes, it's going to be A, yes, G, and then C. So that's going to be, are they saying serine? Yes, that's what is going to be the answer. First 2 amino acids coded for Z in the diagram. So they are saying they want to identify. Now, this is transfer RNA, but Z, what is Z? Z is messenger. Therefore, you have to just change this into transfer RNA, one conversion, because it is messenger to transfer RNA, and then you come to the table, you find it. What is the change in sequence of nitrogenous bases? I've talked about it here, that these changes, gene mutation, sudden change in the sequence of DNA. So that's what you call gene, or the nitrogenous bases. When they change the sequence, then automatically you're going to have a different protein structure if it codes for a different amino acid. So what is a change in a sequence of nitrogenous base in DNA code? Gene mutation or mutation. They're saying that code CUU changes to C. Now they're saying codons. It means that they are not saying anticodons, codons. So it means that I have to convert this into transfer RNA. Sibosiso, are you hearing? All right. We are saying that change this is going to be GAA. And then this is going to be GA. So GAA is going to be what? So now you saying this is going to be leucine. Like the same example I gave. CCU is going to be GGA. GGA is going to be what? Proline. Proline leucine. Now you writing to us that this is going to code for leucine, this is code for proline. Those are 2 marks. Leucine, a mark. Proline, a mark. I see. Now you tell us, because of a different amino acid is coded for, it will result in a different protein. But if it was coding for the same amino acid, then the protein would have remained the same. So it would have said that this code is for the same amino acid, for example, leucine, and also this code is for the same amino acid, which is leucine. This will not change the functioning or the structure of the protein. But because now it is changing, this is leucine and then this is proline, you're mentioning leucine a mark, proline a mark, this will result in a different protein or functioning of the protein. All right, let's go to meiosis. Yes, meiosis, unless you have any question. Someone is asking me, where do you get my booklet? There is a link in the description below. Ne? Yes, that link, it will take you to the website. You go to grade 7 to 12. Then it's going to bring you another page which has past paper 2022. It's going to bring you notes. You click on notes. Then it's going to give you the different subjects for grade 12 and grade 11 and stuff. You click grade 12 and then it's going to give you the subjects now. Now the subject, when you click life science, is going to show you the different topics. Then you download the topic per topic. Yes. That's how you can get that booklet. Let's look at meiosis. Meiosis. What do you expect to find in meiosis in your exam? Meiosis. What do you expect to? I'm now talking about the 6-mark question. I showed you the 6-mark question for DNA. So now I'm showing you the 6-mark question for meiosis. You cannot miss. You will come back and then comment that, sir, thank you for giving us the scope. Yeah. If you follow what I'm telling you, I guarantee you a distinction. Share the video to others so that the others can also. Someone is saying that it's not clear. The clear is as a result of your the internet and your internet connection. You can increase the maybe from 128 to maybe 480, something like that. All right, let's continue. Meiosis, number 1, you need to know how to describe the karyotype. Karyotype. I think you know the karyotype. If you don't know, I'll show you how to describe it. You need to know how to describe crossing over. You need to know how to describe non-disjunction. Ne? Now, this one is a star, is a drawing. Phase before, phase after, phase at this side. So you have to know that. How do you describe a karyotype? Let me see if I can find a question here which has a karyotype. And then I'm gonna describe for you so that when I'm describing, you see ne? You see what I'm trying to describe. Sorry for, yeah, here is an example of a karyotype. Describe the karyotype of individual 2. Ne? But when the question comes, might not, the karyotype might not be there. You have to be knowing it by head. For example, the karyotype is individuals are made up of 46 chromosomes or 23 pairs of chromosomes. Yes? From you mentioning that 46, ne? 46 is a mark. Ne? 40, sorry. You know, technology. What's happening? Okay, let me check and see. 46 or 23 pairs. Don't say 23, because if you just say 23 without writing the word pair is wrong. It's made up of 23 pairs of chromosome. From pair number 1, yes, to pair number 22 is called orosome. You see, now you're describing a karyotype. It's called what? Orosome. Yes, the 23rd pair is gonosome or sex chromosome. Yes, the 23rd pair is made up of X and Y. If X and X, they're together, the individual is a female. All those are marks. If X and Y come together, the individual is a male. You see, you are describing the karyotype. You have already exited the 6 marks. Each chromosome pair, they have different sizes. So that's how you can describe the karyotype. It's made up of 23 pairs. Pair number 1 to pair number 22 is called orosome. The 23rd pair is called a gonosome. These pairs, yes, each pair of chromosome have different size. The 23rd pair, if X is made up of X and Y, if X and X meet, is the individual is a female. Is X and Y meet, the individual is a male. That's how you describe. Now, what about if they bring a karyotype like this? Yes, like this, and then ask you which one is a male, which one is a female. Check. This is short. This is, 2 of them are long. Therefore, this one is a male, this one is a female. Give a reason for your answer. The chromosomes here, they are not of the same size. I see here, same size and shape. While here, they are same size and shape. So it means that this is X and X, while this is X and Y. Remember, we said is longer than Y. That's why you find that to happen like that. Now, someone is asking me, all right, I think it's sorted. Now, we are saying that let's go to another question of karyotype here, just in case, because I was describing the 6-mark question, but there can bring some few questions here. What are somatic cells? Somatic cells, these are body cells. These are just body cells. Name the specific type of chromosome number 1 to, I said it is the orosome. Each pair shown in the homologous chromosome, each pair states the origin of each pair of chromosome. Definitely, one came from the nucleus of the ovum, another one came from the nucleus of the sperm. Meaning that one chromosome, for example, this chromosome came from the mother, and this chromosome came from the father. So came from the nucleus of the ovum, this one came from the nucleus of the sperm, and then fertilization took place, and then a zygote was formed. That's what they are looking for. They are saying that state 3 characteristics of chromosomes which are homologous. Homologous chromosomes, these are chromosomes with the same shape, size, same location of genes. Not same genes, no. Same location of genes. If they are same genes, then it would have looked alike. But the chromosome where they are located, the locus of these genes in these chromosomes are the same. I think now I'm clear on that. So please, you must not miss out any question. Explain one observable reason why these two individuals are not identical twins. Why are these ones not identical twins using this karyotype? These are not identical twins. Why? Because this is a male and this is a female. Obviously, you know that if it is a male, what happens? What happens? If it is a male, the sperm is gonna be Y from the father. Ne? And if it is a female, the sperm is gonna be, if it is a female, the sperm is gonna be X. You understand? We only have one ovum. So I cannot use one sperm and then I form X and Y. Never. Now, why are they not identical? Because of the different sex. Observable, the 23rd pair, this individual one has X and Y, while individual 2 has X and X. So, identical twins are as a result of one sperm fuses with the ovum and then the ovum splits into 2. Non-identical twins are as a result of 2 sperms fusing with the 2 ova. So, meaning that you have different individuals. That's why is the non-identical, you can have a boy and a girl, but the identical twins is either boys or is girls, because we used one sperm. If the one sperm of X fuses with the ovum, then it's gonna be identical twins girls. If one sperm of Y fuses, it's gonna become identical twins male. So that's exactly happening. Yes, I think yes, I'm done with the karyotype. What about crossing over? Crossing over. Describe the process of crossing over. I think I organized for you a question which you're gonna see in crossing over. Oh, I think it's not there. It's here. Crossing over. Yalla, it's here. When you talk about crossing over, you talk about 2 homologous chromosomes. These chromosomes, they come together. I think these chromosomes, they come together. They cross over at a point called chiasma. They must be homologous. The word homologous chromosomes align together. That's the key point. Homologous chromosome. They come together and the chromatids of the homologous chromosome cross over at a point called chiasma. I think at a point called chiasma. You understand? And then these chromosomes, these chromatids, they exchange the genetic material. When they exchange the genetic material, yes, they split and then they form genetically different chromosomes. That's why I told you these are genetically different chromosomes. But these homologous chromosomes, they are not the same. They don't have the same chromosome number. They have the same location of the genes in their chromosomes. That is the meaning. All right. Now, what is P? What is this P? We have talked about. We said that easy. Some people, they call this centromere. That is not a centromere. A centromere is Q. Please don't confuse centromere and chiasma. C, chias, H, this pen, chiasma. If swan is called chiasma, if the men we call them chiasmara. This is a centromere. Centromere. And then this is the chromatid. It's not a chromosome, it's a chromatid. What about this? These are homologous chromosomes. Sometimes they can ask you, how do we call the homologous chromosomes under crossing over? It's called a bivalent. It's called a what? A bivalent. Chromosomes under, homologous chromosomes under crossing over. We call it a bivalent. Yes, I've not checked if because the schedule changed. If just in case that electricity goes at 4 o'clock, ne, I will come back at 6 o'clock live. But if it doesn't go, we are still continuing until a distinction is scored. Yes. All right, let's continue. Now, we are done with the crossing over. Let's talk about non-disjunction. What is non-disjunction? Are you there, guys? Non-disjunction. If you have any challenge, if I'm going to another point, please let me know. I'll read your comment there and then I'll come back to you. All right. Non-disjunction. What is non-disjunction? Before I go to non-disjunction, where does crossover take place? It takes place during prophase 1. Don't just say prophase. We will get it wrong. Prophase 1. Phase 1. What are some of the causes of mutations? No. What are some of causes of variation? We'll talk about them. Causes of variation. I'll see how do people lose marks here. You think that you have written the paper, can't the paper has written you? I'll show you. There is no way the paper is going to write you tomorrow. You're going to have to write it. All right, let's continue. God willing. We are saying that non-disjunction occurs during anaphase. Non-disjunction occurs during anaphase. It can be anaphase 1 or it can be anaphase 2. Anaphase 1 or it can be anaphase 2. So what is this non-disjunction? Check here. I'm going to show you how. Someone is asking me that, can I please repeat bivalent or bivalent? Bivalent is when homologous chromosomes are under crossing over. Chromosomes under crossing over is called bivalent or bivalent. Yes, that's what I said. All right, now come back here. You have chromosomes in metaphase. Sometimes students fail to understand the chromosomes under metaphase. The chromosomes must be in the equator. Equator is an imaginary line. An imaginary line. We just imagine a line which is happening. Yes? It's called what? Equator. But we say that is the center of the cell. So these chromosomes must be at the center of the cell. This thing must be on the sides. Unfortunately, I have not used the camera. I would have showed you. You'd have seen me when I'm trying to say sides. But you have to know that. Make sure that the centrioles are inside, so that sometimes we twist it so that we confuse you, so that you go out, so that we know that you if you just cram, then gone. So they must be on sides. So they can ask you, draw a cell after cell before, or crossing non-disjunction. How is this non-disjunction? How this cell gonna lead to non-disjunction? Now, if these spindles contract and they pull this chromosome, now this whole set is pulled to this side, and then this one comes this side, this one comes this side. You result in a cell whereby this cell has 1 plus this, and then this one, because the whole thing went to the because this one went this side and the whole thing of this went to this side. Let me show you. Let me do like this. Let me do like this. let me do like that so that, yeah, so it means that this part came to this side, this one came this side, while this one came to this side. It means that now this cell is going to have 1, 2, 3 chromosomes. What about this side? It's going to have only 1. I think now this one is 3 instead of 2, and then this one is 1 instead of 2. I think so, because it has 1, therefore it's going to call it monosome. Well, because this one has 3 instead of 2, it's going to be call it trisome. I think, how does this lead to Down syndrome? How does this lead to Down's syndrome? I think so. It means that now, if this fertilizes the ovum, then it's gonna lead to an individual less of 1 chromosome. If this fertilizes the ovum, it's gonna lead to an individual with an extra chromosome, and this is called Down syndrome. But it's not called Down syndrome except it has occurred on chromosome number 21. So which chromosome number is chromosome number 21? Sometimes when we bring these karyotypes, okay, I think I used it. No, karyotype. What is karyotype? Sometimes, okay, I wanted to show you. Sometimes we bring it and then we confuse you on the karyotype, ne? And then we ask you, how did yes, check here, ne? On this chromosome number, they put another one there, and they ask you, is this chromosome individual fine? How many chromosomes? Yeah, how many chromosomes are there? Because you know that this an individual is supposed to have 23 pairs. When I just say 23 pairs or 46 chromosomes, wrong. Check before you conclude anything. Please check the chromosome number 21. I'm done with that. Let's go to this. They're saying that a drawing, we have shown it, but a drawing, it must have a title, ne? Must have a title. When they ask you to shade, for example, if the chromosome is like this and this one is shaded, ne? Please, when you draw, still draw it like that and it's shaded. If you shade it down, shade it down. If you shade it up, shade it up. After that, label. Have some 2 labels. I want to give you one thing students always mess up with is metaphase. Listen carefully, don't go away. Anaphase, yes, then telophase. I'm gonna say this: metaphase 2, anaphase 2, telophase 2. When these chromosomes, let me show you here down. When these chromosomes, look, I have it here. When they're under metaphase, we call them chromosome. When they're under, they're being pulled now, ne? They're being pulled to opposite poles, ne? Like this. These are not chromosomes. These are now chromatids. But the moment they reach the poles, we don't call them chromatids. We call them single-stranded chromosomes. Unreplicated chromosomes or single-stranded chromosomes. We only call them chromatids if they are at the under anaphase. They are being pulled. Here, they are chromosome. These are replicated chromosomes or double-stranded chromosomes. These ones are called chromatids. The moment they reach the pole in telophase, it becomes what? It becomes unreplicated chromosome. Don't call them chromatid. Yes. Let's go back to this last question of meiosis. Then we conclude it. Yes. Okay. Now, they are saying they are saying that drawing variation. What is variation? Variation is the difference among organisms of the same species. Variation is the difference among organisms of the same species. Don't compare mangoes and oranges. Don't compare dogs and pigs. That's not variation. Variation must be comparing dogs. Variation is the difference among organisms of the same species. It must be the same species. Yes. Now, what are some of the causes of variation? If you're comparing humans, you must compare humans. So what are some of the causes of variation? The causes of variation is, now, the major mistake students do in this exam, if they ask you 3 causes of variation, the first thing the student is going to say that the first cause of variation or genetic variation is going to say that meiosis. Yeah, we shall say correct. And then the next point is going to say crossing over. We're going to say thank you. Then it's going to say random arrangement of chromosomes during metaphase. We're going to say yes, but no. Why? Why are we crossing it when it's correct? It's because you mentioned here meiosis, and meiosis involves crossing over and random arrangement of chromosomes. So if they ask you this question, please remove meiosis. Don't think about meiosis. Then you talk about crossing over, random arrangement of chromosomes. There are 4. We will not put you this. We will give you a tick here and we'll give you a tick here. So you see, you have got 2 marks. Then plus you can talk about mutation, random fertilization, unless they have said that give 3 causes of variation during meiosis or during the process of meiosis. Then now you talk about crossing over, talk about random arrangement of chromosome, and then you can talk about mutation. Then you don't talk about random fertilization, don't talk about random fertilization or random mating. You don't talk about it. Yes. Yeah, I think that is the meiosis. Can we check and see? There is one question you usually lose marks. How many number of chromosomes? That question is always killing students. Is always killing students. Is always killing students. All right. Let's go through this question paper. This one. Then they're saying that what is A? A is a cell membrane, not nuclear membrane. You can't see nuclear membrane when a phase is meiosis, is metaphase. Spindle fibers, then this is a chromosome. Yes, identify the phase of meiosis above. Because they are single chromosomes, therefore it's gonna become what? It's gonna become metaphase, because they are single metaphase 2. If they are double, then it becomes metaphase 1. But here single metaphase 2. All right. Let's go to another question. They are saying that what is A? We have seen it. What is B? We have seen it. Give one observable reason for your answer. Chromosomes align along the equator singly, individually. That's, if you don't say singly, 0. Or individually, 0. It must have that word, individually or singly. Chromosomes align along the equator, 1 mark, singly. What about describe the role of B in the movement of these chromosomes during meiosis. B is spindle fibers. They pull the chromosome, the chromatids, in this case, the chromatids to opposite poles. They try to shorten. When they shorten, they pull the chromatids to opposite poles. Charlotte, are you still here? All right, let's continue. Draw it. This is the question I was talking about. You can't go without. Put the title. Good. After putting the title, what next? What next? What next? You talk about, you draw, because they're saying that draw a diagram of C, structure C. They're saying structure C, ne? As it would appear in the final phase of this meiotic division. They are saying only C. So if they're saying structure C, they're not saying draw the cell, how it will appear. Since they're saying structure C, then automatically you're gonna draw this. Okay, let's see. Structure C. You have two options. Why two options? Then this chromosome, you see, it's 1. So you draw that chromosome with the centromere there. While you can draw this one also, draw the same thing with the centromere there. Are you seeing? But now this one is shaded. This one is not shaded. Why? Because it is colorless. See? Nothing shaded. This one is shaded. Therefore, you shade it up. I see. When you shade it up, what next? So it's gonna be like this. But now, where do I get? Where am I going to get the marks? Remember, they're saying final stage. Final stage has a nucleus. So this chromosome must be in the nucleus. Don't forget that at the final stage you have the centrioles. You also put the centrioles in there. Centrioles are outside the nucleus. Then after that, please give a title and then you label cell membrane, nuclear membrane. Now, okay, you can talk about centromere, and then now this is not a chromatid. This is a chromosome. The chromosome I talked about, that these are called unreplicated chromosomes. These are unreplicated chromosomes. Yes. So I think you understand it very well. I think you really understand it very well. No, I think you got it right now. Ne? If you have not got it right, please let me know. Let me know. All right. Now, let's go to another sub-topic. Hey, there is one which stresses you. How many chromosomes would be there at the beginning of this cell? How many chromosomes are here? Oh, how many chromosomes are in this cell? So now, people always confuse. Please, if they don't mention this is a human cell, don't think about 46 chromosomes. Unless they have said this is a human cell. If they have not said this is a human cell, no, you are not supposed to draw both. You only have to draw one, because they say draw a diagram. Label the diagram, not diagrams. Ne? Someone is asking, should we draw two? No, we only draw one. Yes. Or follow the instruction. What is it saying? But in most cases, they want one. But when you're marking, we draw the two, and then we see which one to give you. Ne? Basically, depending on which one you have drawn on the memorandum, the answer is there, then we will do that. Yes. All right. Now, and when you are marking, we don't just mark. We just have to mark the title. We mark the title. We mark the shading. The shading is correct, maybe. Then we mark maybe 2 labels, or the correct diagram, 1 label, and then the correct diagram, then here. All right. They're saying that how many chromosomes are found here in this cell? Someone is going to say 23. I ask you a question. 23 from where? This is not 23. I see 2 chromosomes. Why do you say 23? So it's going to be 2 chromosomes. People are always talking about 23. Why? How many chromosomes will there be at the end of this cell in telophase 2? There will be 2, because this is meiosis 2. The meiosis 2 maintains the number of chromosomes. Another question. Sorry. Then they're asking you, How many chromosomes were there? How many chromosomes were there in the same phase of meiosis 1? There were 4, because meiosis 1 halves the chromosome number. So if meiosis 1 is 5, then meiosis 2 is gonna be if meiosis 1 is 10, meiosis 1 is if meiosis 1 is 10, meiosis 1 is gonna be meiosis 2 is gonna be 5. So there is half the chromosome number. So please, you only if they say this is a human cell, sometimes they ask you that how many chromosomes will there be present in the same phase as a human cell. Now here, because it's meiosis 2, then you talk about 23. But if they don't talk about human, please and please, I beg you, don't talk about 23. All right. Then they're saying that, lastly, about meiosis, before I go to genetics, I show you the 6 marks which are going to come from genetics, and then I show you the simple questions, the way we are trying to define them. Share the video to others so that they don't miss out. Don't forget to subscribe, like, and also drop the question which you really want me to sort out. All right. Question: what is the importance of meiosis? What is meiosis? What is the importance of meiosis? The reason why we have meiosis is to have half the chromosome number, so that this doubling effect, because you remember sperm, let me show you sperm. The sperm, we'll call it sperm, ne? All right. The sperm, what happens? This technology, eyeball. Okay, I hope it's right now. The sperm will bring 23, and then the ovum will also bring 23. So when they fuse during my fertilization, you form 46. Yes. So meiosis must divide them again so that they form 23. So that's why we are saying that it reduces the chromosome number, or it halves the chromosome number, or it solves the problem of the doubling effect of fertilization. Number 1, that is it. Number 2, the major importance of meiosis is to produce gametes. Gametes, these are the sperms and ova or ovum. Number 3, where does meiosis take place? Meiosis takes place in testis, in human, and then in ovary, that is female. Anthers in plants, that is male, and the ovary, that is female for the case of plants. You have to know those things. I think I've exhausted the questions of meiosis, unless someone is saying that load shedding is coming. Oh, sorry. And then okay, can I make it downloadable? I cannot make it downloadable until it is I'm offline. The moment I'm done, I'm completely done, then it will be able to this one is asking me that how do I know the number I must count? The number is shown there. If look at this, these are 1, 2, 3. Let me check here. Okay, let me look for another question here. You will see the numbers. Sorry, when I'm looking for the question. Okay, that one. This one we are done with it. What about this? Let me check here. How do I know the number? Oh, in this case we don't have, but this is the crossing over. This is the thing I was talking about, the process of crossing over. Centromere, chromosome, chiasma, this is a bivalent still, it's also a bivalent still, also this is a bivalent, but depends. You see now the crossing over here, there are two. So when they are drawing, for example, you have to draw them. In this case, you draw them up. You draw them up here, you draw them 2 times. So I think I've exhausted. But what you see, if the cell, if the cell is showing you, chromosome 1, chromosome 2, chromosome 3 like this, and then they ask you how many chromosomes are there. These are the number of cells, number of chromosomes, 1, 2, 3. So tell us 3. How many will be there in meiosis 1 of the same phase? There will be, because meiosis 2 is single strand, chromosomes are 1, 1, therefore it's gonna be 6 in meiosis 2. Unless they say that how many will be there in the same phase of meiosis, but for human. I say that if they don't talk about human, forget about. You see now? Here is the question. Oh, where in the human female body would this type of cell division? This is where does meiosis take place in female? I've answered this, and I say that ovary. Give a letter name of the structure that attached to the spindle fiber, which is C, and then this case is the centromere. How many chromosomes will be found in each letter cell at the end of meiosis? Now come back here and see. The diagram represents the chromosome in the cell that is undergoing normal cell division. You see, it's undergoing normal cell division. So now they're asking, how many chromosomes will there be found in each daughter cell at the end of this cell division? How many chromosomes? How many chromosomes are you seeing here? This is chromosome 1, chromosome 2, chromosome 3, chromosome 4, chromosome 5, chromosome 6. Because they are still under meiosis 1, so at the end of this cell division, it will be half the chromosome number, whereby 1, 2, 3, 4, 5, 6. So it means that the answer is gonna be 3. I think Chantel, I think I've answered your answer correctly. Yes. Please share the video so that others can also benefit. Sometimes I do these lives, and then after that they look at it and they say, I didn't see this live. So share it so that even other people can benefit from this live. All right. Let's go to our favorite topic called genetics. Called what? Genetics. Share the video so that everyone can get all the marks in genetics. Genetics. I'm not explaining too much of this, but I'm explaining only what you expect in exam. I'm not explaining the basics. 6-mark questions. 6-mark questions. 6-mark question. When you talk about 6-mark questions, yes, in genetics, what are you supposed to look at? Number 1, genetic cross. In this genetic cross, you need to know, when do I say sex-linked and when do I say non-sex-linked? Yes, sex-linked and non-sexy-linked. I think load shading is not there. I think so. On my side, ne? I think it's not there. Ne? I think they have changed the schedule. All right. Non-sexy linked. Sexy linked. Ne? All right. Number 3 is pedigree. You have to know the pedigree, but still here, you have to know whether it's sex or none. Another thing, disaster, blood group. And then lastly, cloning. Here we are going to talk about biotech. All these questions are 6 marks. 6 marks. I'm not saying this is what I'm going to talk about only. These are questions which come with the 6 marks. With the 6 marks. So if you mess them up, you lose. As I said that each section you're going to have one which has 5 marks, 6 marks, 7 marks. Latest. Oh, the worst is 6 marks. This, please don't lose them. Don't lose these marks. Distinction. Call your name Mr. and Mrs. Distinction. Tomorrow we have to crash the paper. All right, let's continue. Genetic cross. But before I start genetic cross, I've not talked about terminologies. I've not talked about them. But I want to know this. What is a locus? What is a locus? Or a position of a gene along a chromosome is called locus. Now, when do I use sex-linked and when don't I use sex-linked? I'm trying to go through this. After that, I go back. I'll show you. When you're doing the genetic cross, guys, even if you don't know anything, just write P1 space F1. We shall come and give you a tick. So you can't go to the paper without having this. So you have gone to the paper when you know. When you say genetic cross, before even thinking P1 F1, you get a tick. Okay, this is a phenotype. Phenotype is always given in the question. They will say that blue eyes, married, black eyes. You see, you come here and say blue eyes crossed with black eyes. That's a mark. Another mark you have got. After that, you say genotype. This is the only disaster which is there. We're going to look at it. And then now, what about here? Now we say phenotype. We are done. Genotype, we are done. Then now you go to meiosis. Meiosis. They are saying that, okay, P1, F1. I'm going to show you when do you write P2. Because sometimes, yeah, but you saying P1, F1, mark. They say phenotype, a big head. You see? I don't know. Are you a big head or a small head? I don't know. All right. Then they are saying that big head crossed with small head is given in the paper. So you don't need to stress. So you get a mark for this. You get a mark for this. Now, after that, you go to genotype. This is the only disaster I've talked about. We're going to look at it. You have to identify it. I'll show you. For example, T, small t, T, small t. Now you go to meiosis. Yes. They're saying meiosis. They're saying T, small t, T, small t. Which are these are gametes. You see? Now, gametes, in most cases, we love it when you circle them. You know why? Because sometimes people try to bring this close together. And once you bring them together like this, we cross. We don't give you a tick. So make sure that I prefer, always I tell my students, since you are my student now, circle them. Circle them. The moment you circle them, at least we shall know that, we shall know that nothing, even if they are too close, since you have circled them, still we will give you a mark. Now, then after that, you talk about fertilization, where you have the crosses. Now, you mentioning meiosis and fertilization is another mark. In most cases, this question is 6 marks. So you have got 1 mark, 2 marks, 3 marks. So you have got 50 out. 50 out of how much? Out of 6. 50% you have got it. Before whether stress have started or not, whether you got these things correct or wrong, as long as you have that, you have the 3 marks. All right. Now, you continue. You cross this. Okay. I'm going to show you another way how you lose marks and how to get marks. This T with the T, and then this with this, T with the T, this with this, T with the T, then this with this, small t with the small t. Now, these are offsprings. Listen. In the question, sometimes we put what you call compulsory mark. We put what you call compulsory mark. When you're talking about compulsory mark, someone is asking me if I'm going to do cloning. I'm coming there to cloning. Compulsory mark, for example, if this is a compulsory mark, even if you get 1, 2, 3, so it's 1, 2, 3, 4, 5, 6. And if this question is 5 marks, and they say this is a compulsory mark, even if you say 1, 2, 3, 4, 5, 6, you got 6, but you lost the compulsory mark. We won't give you 6, no, we'll give you 4. We'll give you 4. But if you write this, the 3, the other 3, they are obvious whether the compulsory mark is there, whether the compulsory mark is not there. Those 3 are secured. You go to the paper when you have secured them. Then the other ones, the compulsory mark depends on the question they have asked you. In most cases, they ask you, show this. Then you do what? You show. That showing will lead to the compulsory mark. I hope I'm clear on that. Then they are saying, I'm gonna fire you a few questions here. So the crossing, all of them, they do the same. Oh, I have not spoken about determination of sex. Sex. Don't forget to stay virgin. Determination of sex. Determination. Someone is asking. All right. Sex determination. Someone is talking about meiosis. Meiosis, we finished it. Sex determination. Okay. But you'll be able to watch it when the live is done. All right. We are saying that question can come, and this always kills the student. How many genes control blood group? How many genes control blood group? Let me know in the comments. Just now. Just type how many genes control blood group. I'm waiting before I continue. How many genes control blood group? Let me see. Who is the first one to answer? How many genes control blood group? I'm waiting. Define mutations. Yes, I'll talk about the mutations. How many genes control blood group? Yes, I'll cover every topic. Don't worry. I'll cover every topic. How many genes? Guys, type. I want to see the answer. How many genes control blood group? I'm not seeing the answer. 4. Someone saying 4. Thank you. How many genes control blood group? I'm still waiting for the answer. Type. Another one saying, I think 4. All right, thank you. How many genes control blood group? Tell me, Chantel. Yeah, okay, I think 4. Thank you. I want to know. How many genes control blood group? 3. How many genes? Okay, guys. Thank you very much. That's what I wanted to know. Yeah. Now, since we are looking for the distinction, they are not 4. It's only 1 gene. The gene, it is 1 gene polyallele. 1 gene. Which gene is that? Blood type. Blood group is only 1 gene. Are we together? How many alleles control blood group? There are 3. 1 gene. Remember, which gene is that? Let me show you here. Yeah, Terence said what? After giving. Okay. Okay. Blood group. Blood group. The gene is called. It's like saying height. You're either tall or short. Is 1 gene which is height. You see? Height. You're either tall height, you're either tall or short. You understand? Since it's one gene of height, but two alleles of tall and short. So blood group, the gene is blood group or type. Yes, how many alleles? There are 3. So say genes for blood group is 1. Alleles, there are 3. That is allele for A, allele for B, and allele for O. Sorry, allele for O. Like that. Are we together? Yes. Thank you for those people who tried to participate to give the answers. Since we are looking for the distinction, we can't go without knowing this. Gene, one gene. How many alleles? 3 alleles. Ne? 3 alleles. How many genotypes? Genotypes, there are 6. Yes? Genotypes, there are 6. You have this. Remember, A is either homozygous or heterozygous. That's 2. B is either homozygous or heterozygous. Then you have, that's 1, 2, 3, 4. Then you have A, B, and then you have that for O. That's it. So 3 genes, 1 gene, 3 alleles, 3 genotypes, and then phenotypes, there are 4, which is A, B, and O. So basically that is it. Oh, AB, yes, AB is codominant. Yeah, O is this is homozygous. Ne? This is the codominant or heterozygous. No, codominant. This is heterozygous, is homozygous, heterozygous, homozygous. So there are 6 of them. 1 gene, 3 alleles, 6 genotypes, 4 what? 4 phenotypes. Now, a tricky question can come. A man who is blood group A married a lady who is blood group B, and they produced a kid of blood group O. Show how this is possible. A man of blood group A married a woman of blood group B, and they produced a woman of blood group O. How is this possible? It means that once you find that question, for you to cross, it means that a man, because they are saying they produce a kid of blood group O. O is small I. Before I go there, let me show you the mistake here you do. And we don't mark it. However much you love it, we don't mark it. Some people, they write blood group like this. A. What is this? This is not I. Don't write I like this. And you put a dot like that. I is like this. Don't write I like this. This is 1 in Roman figures. Check. You have sometimes, you have watches where it shows 1, 2, 3. Yes, they are like that. So this is not I. This is 1, this is 2, this is 3 in Roman numerals or Roman figures. So don't write A like this. We won't give you a mark. However much some computers, yes, they do. You are not a computer. Write the way
Use
correctly.
my distinction
1
material.
A.
Yes. Those people who use my distinction material,
make
I
sure
know
that
you got
all these
this.
are things which might lead you to lose the distinction. For those people who have been using my book, Distinction Material, you know exactly what I spoke about it, and those people who have been watching the video. So it doesn't matter now. What matters here is for tomorrow's paper, for you to crush it. All right. Now, we say that how is a man of blood group A marrying a lady of blood group B and they produce a kid of blood group O? One, know that blood group O, it has two eyes. Yes? Yes. If they have two eyes, how about this? Remember, A can be homozygous or it can be heterozygous. B can be homozygous and heterozygous. Now, if I have O, which is II, yes, it means that I used this and this. Because if I make this and this, there is no way I'm going to produce these two. Even if I say this and this, there is no way. I'm going to only have one I, small I. Therefore, it's going to be heterozygous blood group A. So when you talk about the genotype, is going to be the phenotype. So I'm going to say P1, F1. Genotype, phenotype, phenotype. I'm going to say A crossed with B. And then I say genotype, I'm going to say A heterozygous crossing with B heterozygous. Then meiosis will take place. Some people, they then say A, I, B, this is B, ne? I. And then I circle them. Please don't put a cross here. Please don't. You'll get it. You'll get it 0. You'll get 0 there. Please don't put a cross there. Ne? Yes. Someone is asking, why didn't you find the channel at the beginning? That's the answer for God. All right. Now, let's continue. Let's continue, guys. But you can tell your grade 11 or your sisters, your brothers, subscribe the channel. Look for M.Saidi. Yeah. You always find the content there. All right. Now, look. Now, this is gametes. Gametes. So if these are gametes, now what happens here? You can use a Punnett square method, or you can just cross. Whatever you want, still it is fine. For example, let me use the Punnett square because I've been using the cross. So you draw your Punnett square here, and then you say, okay, you have A, you have I here. You have B, you have I here, small I. And then now you cross, you're going to say this is A, B. Ne? This is going to be B. And then this is going to be A. And then this is going to be O. You see? You see? These are offsprings. Offsprings. You see? Now this one produced a person who is blood group O. That's how it was possible. It can even come in the 20 in the 7-mark question. That's why I showed you those crosses. Those crosses. So please, blood group should not be a problem. Which blood group is now I'm done with the long question of blood group. Few questions now. Which blood group is a universal donor and which blood group is a universal recipient? Blood group AB receives from every blood group, while blood group O donates. So blood group O is a universal donor. Some people, when they are blood group O, yeah, they think their blood is strong, they feel good. No, it's worse. Why? Because if you lose blood, if you need blood, we have to look for only one person with blood group O. But if your blood group AB, you are lucky. Anybody who comes, you can donate that blood to you. So blood group AB becomes a universal recipient. It receives from everybody. While blood group O is a universal donor, only donates to anyone. Someone is asking me that why these crossings are not allowed. The crossings are not allowed here because they are not crossing. They're already gametes. After that, you can draw your crosses here. Ne? So the crossing here, the cross here, no, it's not allowed there. It's not allowed. Crossing here is fine. If you cross them, it's fine. You can either use the Punnett square or you can just do the crossing. All right. Let's go to we talked about the blood group. I'm coming back with the cloning, cloning. Or I'll give you one tip of an example of. Okay, let me give you the question, because I just want to go just for 2 or 3 minutes. Yes, 3 minutes. Let me give you just one question for you to practice. Practice for me is 6 marks. Yes, this one. It's a simple question. There is something, most especially on this question. I need to see so that you don't lose that mark. Please do that for me. Do that for me. Then after that, I'm gonna do it again for you. What's the time? Is 39. I'm giving you 4 minutes. 4 minutes to finish. I will be back. I'm saying I will be back. I hope you are done. Hope you are done. Okay. Oh, come on, saying yeah, it's done. All right. Anyway, all right. Okay. That's nice. Okay, now let's go through it. They are saying diagram below, okay, controlled by R and R. Check here. The first point is R and W. These are capital letters. The moment they are capital letters, it means that both of them are dominant. All right. Now they're saying red cross with white produce pink. You see, they have 3 phenotypes. The moment you have 3 phenotypes, it means that is incomplete. Dominancy. It is incomplete dominancy. A. The question is here. State the type of dominancy shown in the Snapdragon plant. Here is incomplete dominancy. Give a reason for your answer, because a third phenotype is given or an intermediate is given. You see, a third phenotype is given or an intermediate is given. Now, if it is complete, if it was complete, I would have been with red and white. I either produce red or white. Which one is dominant? Depending on which one is dominant. And then if I have codominance, red and white, I would have produced a mixture of red and white. But because this one is pink, a third phenotype, therefore, or an intermediate, therefore it is incomplete dominancy. They're saying Gardner crossed 2 pink flowered snapdragon plants using the genetic cross. Show the ratio of expressed phenotypes in the offsprings. Now, remember I told you that the first thing you need to do is to write P1 F1. But in this case, they're saying they crossed 2 pink. Pink is found on the second generation. Therefore, it won't be P1 F1. It will be P2 F. Most cases, students lose that mark. Ne? If it is the second generation, then you say P2, F2. If the first generation, then you say P1, F1. Ne? Yes. So now, there you talk about phenotype, which is pink. I told you that this is obtained from the question. Check. They are saying, what are they saying? Check what are they saying. They're saying genetic cross showed a gardener crossed two pink flowered snapdragon plants is always obtained from there. Are you seeing? Then now, so it means that I've got a tick here. I've got a tick there, and also I've got a tick there. So now 2 marks. Now they are saying that pink. Remember, they say that white is capital W, and red is capital W, capital R. White is capital W. Therefore, pink is gonna be capital W, capital R, capital W, whatever. Capital W, capital R, or capital R, capital W. It's the same thing. So it's gonna be capital R, capital W, crossing with capital R, capital W. But this is the genotype. See, now you get another tick. Then they are saying that meiosis will take place, and then you have capital R, capital W, capital R, capital W. I said, don't leave these so-called gametes to be as if they're at the beach. No beach, né? At the beach, né? You live naked, né? You enjoy yourself. Né? Don't leave them naked. Try to dress them up. Try to dress them what? Try to dress them up. At the beach? So try to dress them up so that, né? Yeah, they are not there at the waters there when they are enjoying their life. Né? Try to dress them up so that you separate them. Even if they are too close to one another, you won't lose a mark for that. You won't. Then after that, you do the crossing, or you do a Punnett square. So cross with R, capital R, capital R, capital W, capital R, capital W. Because I started with R, I will start with R, capital W, and then capital W, capital W. Whereby these are offsprings. Those are offsprings. Now, if those are offsprings, what is it? R, this is red, this is pink, and then this is white. Meaning that I will have one red, 1, 2 pink, and then 1, 1 white. There is a mistake where the student do, whereby they ask you the ratio. In this case, you write 1 to 2 to 1. Ne? Some people, when they ask them the ratio, they write expression, and sometimes when they ask you the expression, they write the percentage. So when they ask you the ratio, please write the ratio. If they ask you how many were red, or what is the percentage of individual plants which were red, then you have to say it's 1 out of what is the total. The total is 4 times 100. Then you give us the answer. You give us the answer as 25%. Please don't forget that. All right. Let's continue. And we say our question is, I think I've exhausted that. Pedigree. Let's just look at one question here, which involves the pedigree, and then we can go to our. Oh, there is one question here for blood group. There's one question here for blood group, but I think we exhausted the blood group, but you see that blood group also has 5 marks. Explain why paternity of the boy could not be established using the blood group. Why is paternity test is not able to be established, like to conclude that this is the father of the individual? It's because many people have the same type of blood group. So you can't say that, as I showed you, that is blood group A married blood group B, and then they produce blood group O, still they produce blood group AB, still they produce blood group B. So whom are you going to say that is not the father, who is, or whom are you going to say that is the father? So it means that you end up losing the what? The mark. All right. Why is it not conclusive? Why is it not conclusive? Because many people have the same type of blood group. But if they ask you, explain why paternity test for this situation, you will say that the mother is blood group O. You see, the mother is blood group O, while the boy is blood group AB, blood group B. So of which the boy can be can be B, heterozygous, or the boy can't be homozygous. Why? Because the mother is II, so gets one from the mother. So the genotype of the boy is heterozygous. Now this one can give, yes, the B, ne? And also this one can give the B. So whom are you going to say is the father? Whom are you going to say is not the father? So that's why in this case, blood group can only help you to eliminate those possibilities which cannot happen. For example, a person of blood group, this father, the father can't be blood group A because this person is blood group B. You see? So if it's blood group A, there is no way how the boy is going to have blood group B. So if a man, there is a man, another man who is blood group B, so blood group A, that one is wiped out. You understand? And then another man who is blood group O cannot be. So blood group A and blood group O, they are wiped out. Why? Because if it is small I, the boy is blood group B. So there is no way how this boy is gonna get a B from the father who is blood group O, yet the mother is already known is blood group O. You see? So that's the meaning that you can't use blood group to conclude the father of the individual. Because you are blood group B, you can't say that this is my child because this child of mine is blood group B. There are so many people who have the same type of blood group, and the possibilities of having these are many. I hope you understand me better. Now, how do you write this? It means that this one is only blood group A and blood group O are going to only be wiped out. So the blood group A and blood group O, they cannot be the father. So you get those 2 marks already. And then this blood group, the male A can give this allele for B to the boy. So it can be the father. Or blood, this one also can also give one allele for B and gets another one from here. Also can be the what? Can be the father. That's why we cannot conclude using blood group who is the father of this boy. As I repeat that, blood group can only be used to eliminate the possibilities which cannot happen in a situation. Who is the biological father of the boy using the DNA profile? Now you have to check and see. You will see that it is this and this. You will see that is male what? Male 2. Give the reason is because the remaining bands of DNA profile match with the male. The remaining, they must match. If there is one which is not matching, then automatically it's not the father. Might be a relative, but might not be the what? The father. Remember, all the genes that this boy or this baby is having is mother and the father. So if you take out all the mother's genes, because the mother is known, now the remaining bands of this boy must be those ones of the father. Let me show you what I'm trying to say. This mother, okay, this one is not for them. Yes, this one is corresponding. So this one is out. This one corresponds. This thing is now jamming. All right, corresponds. This one corresponds. Oh, this one corresponds. Yes. This one is not matching. So this one is not matching. This one is not matching. This one corresponds. Is out. This one is not matching. This one is not matching because this is not. This one is matching. So now all of those ones which have X must match with the male who is the father. That's why I'm saying that the remaining bands of the boy, after those ones of mother have been removed, they must match with the father. Okay, this one, yes, it matches. You see, this one, yes, it matches. You see, this one is matching with this. This one is matching this. That's why you say that individual 2 is the father. Male 2 is the father of the boy, because the remaining bands of the boy have matched with the male, with male 2. All right. Now, let's go to just one example of a pedigree diagram. Yeah, pedigree, where are you? Pedigree, and then I show you how you can use it to answer and also to score a mark. They are saying hemophilia is a that statement is enough. This statement, hemophilia is a sex-linked trait. Hemophilia is a sex-linked what? Trait. Meaning that if it is a sex-linked trait, what is it? It means what does it mean? It means that you're gonna use X and Y. Please, if they don't say is a sex-linked in exam, don't use X and Y. You're gonna get it wrong. Is a sex-linked disorder. Don't use X and Y if it is not mentioned in the question. So here it is mentioned sex-linked. Or sometimes they don't say sex-linked. They say that hemophilia, the gene is carried on X chromosome. Still that is sex-linked. If it's not mentioned, please don't even bother. Don't even waste your time. Don't even waste your time to let me show you here. One of the deafness in human is carried on a single allele. The diagram below shows, yeah, done. They are not talking about sex-linked. However much they talk about Paul, and you know that Paul is a boy, Liz is a girl, Mary is a girl, John is a boy. However much you know that, please don't use X and Y. Please don't use X and Y. So if they don't mention is sex-linked, don't use X and Y. If they mention sex-linked, then you talk about X and Y. How many generations are here? There are 3. 1, 2, 3. They are saying that, and then number 2, it can be caused by, something can be caused by a recessive allele, or it can be caused by a dominant allele. Anyway, let's continue with this question. They are saying, state what is represented by a circle or a diagram. I wanted to tell you that sometimes we don't bring that a circle is a male. No. A box is a male. A circle is a female. Sometimes don't give you that key. You have to go to the exam when you know that. All right. Now they're saying. What are they saying? Okay. Now they are saying. This is a boy. This is a. How many individuals? Let me brainstorm you. This is affected with hemophilia. Don't say he's sick. Is sick of what? Sick of what? Malaria, ne? Sick of what? Sick of hemophilia, or is hemophilic. All right. How many kids are produced by P and Q? They are how many? It's 1, 2, 3, 4, 5. I know many people are gonna say that. It's not. They are not 5. 1, 2, 3, 4, 5. No. This one is an intruder. This one intruder. This one got married. This one got married. This. So there are only 3 kids. How many generations are here? 1, 2, 3. There are 3 generations. You understand? What is the phenotype, the genotype, or the phenotype? They can ask you what is the genotype or phenotype of P or Q, whatever they can ask. But if it is shaded, it means that that individual is affected. Now, they're saying that state what is represented by a circle. We have said that the circle means a male. State the number of A. We have talked about them. There are 3 generations. 1, 2, 3. Offsprings P and Q. State the number of offspring P. The same thing is this, and this. This one and this ones, they are only and only got married to that. So they are intruders. They're saying that let us give the letters of only male who are having hemophilia. Letters of the female. Female is T and the female is U. Those are the only two people who are having hemophilia. Why? Because they are shaded. And then lastly, the genotype of R. The genotype of R. Now, how do we find the genotype of R? Look at the babies or look at the parents. First, look at the babies. It can help you. If you see that the baby is hemophilic, then automatically the mother is gonna be heterozygous. So the mother is gonna, remember that here you have X and Y. Y doesn't carry anything. Y doesn't carry anything. So it's X and Y. So Y does not carry anything at all. All right. I think now, okay, so since this one is sick, it means that, and is a boy, so it's going to be like this. It's going to be, this is the recessive allele. So if it's a recessive allele, obviously this can't come from the father because the father already gave the what? The Y. Therefore, this came from the mother. So it means that the mother, since is normal, but a carrier. So the mother is going to be, or R is going to be capital A, capital X, capital X, capital H, small h. And that becomes the answer for that. Yes. Let's continue now. Let's continue. So we are done with this. Sex determination and biotechnology. How do I explain sex determination? Sex determination is like describing a karyotype. When asked you to describe sex determination, it's like describing a karyotype. But here, if they ask you the genetic cross, you have to do the cross. Ne? So, for example, if it's a cross, you tell us that P1, F1, everything, ne? Male crossing with a female, yeah, and then you tell us that male is X. You tell us the phenotype here, ne? And then you tell us the genotype. Male is X, Y, and then female is XX. Then you cross. Then you tell us meiosis here. You tell us gametes. They come out here. And then I'm gonna show you how to write it if they say using a genetic cross, and then also show you how do you describe if they ask you to describe. Then these are gametes. These are gametes. Then they cross, then this and this, cross Y, and then this and this, they cross like that. So this is the offsprings, meaning that you have 50% chance of having boys, and 50% chance of having girls. What about those who have girls, or boys? Yes, even if you have a boy here, the next time you have 50% of a boy again. Even if you have another boy, the next generation, you have the next reproductive cycle, you're gonna have still 50% chance of having a what? Having a boy. So it depends on how do you roll your dice and how many chances do you have to have a boy. There are 50, but you are just lucky that you have what? Boys, or you are lucky to have what? To have girls. So in this case, we are saying that 50 boys, 50 girls. Yeah. If they ask you to describe, you tell us that individuals, humans are made up of 46 chromosomes. Ne? The 23rd pair is called gonosome. It's either X and X or X and Y. If X and X, if a male sperm with X fertilizes the female ovum, it will result in X and X. The individual will be a female. If a male sperm having Y fertilizes the ovum, remember ovum is always X, the individual will be X and Y, and therefore will be a male. There are 50% chance of having a male and 50% chance of having a female. That's how you can describe sexy linked. So let's finish up cloning, and then we go to evolution. We finalize it with the evolution. All right. Biotech. Cloning. What is stem cells? What are stem cells? Stem cells, these are cells which have ability to divide to form other body structures. Imagine you are just a sperm. You are just a sperm, ne? Sperm fused with the ovum. You formed a zygote. The zygote, remember this paper one formed a ball of cells, ne? Which called a morula. You see, a morula is a stem cell. Because here, if I ask you, show me the eye, show me the legs, show me the chest, show me the, you can't show me. It's just a ball of cells. So these cells, they have ability to divide again to form other body structures. You understand? Yeah, you form other body what? Structures. And then, yes, you become, yes, you have the eyes, you have the ears, you have everything. So we call them stem cells. Those are cells which have the ability to divide to form other body structures. All right. Give me 2 examples of stem cells. Umbilical, adipose tissue, bone marrow. So you have to know those stem cells. Sometimes we bring this as an extract. Let me check and see. If I have any question regarding 2 stem cells. This is gene. Calculate the percentage of, okay. This is gene. Oh, I skipped something which is very important. That is the dihybrid cross. Yes. Let me leave it there so that I don't forget again. Dihybrid. I want to show you how do you identify the genotype. How do you identify the genotypes? Using the gametes, because that is a very problematic question. Okay. So in this case, I can't find the question there. All right. Now, let's go to cloning. I only want to discuss about cloning here. Ne? Yes? About the what? IA. Cloning. Yes? I have sheep A. I have sheep B. Ne? I get a cell of sheep A from the skin. Let me say somatic. And then this one I get it from ovum. Ne? What I do, I remove the nucleus of this. I go with the nucleus. Ne? I throw away this shell. Well, here, I go with the shell, but I throw away the nucleus. I fuse them using electrical method, and then I form a cell which looks like a zygote. It looks like a zygote. Now, this zygote, I put it into another animal. A foster mother. Animal. Yes? Yes. And then what I do, I produce a lot of these animals. And these animals are identical. Question, why are all these animals identical? They're identical because when this forms a zygote, it forms a ball of cells. Now this ball of cells, now I divide these balls. Now I'll have small balls, small balls, small balls, and then the small balls, I keep on going and then insert them in different other shape. Ne? So it means that because I use the same genetic material, obviously these ones are going to be identical. It's because it's the same genetic material which was used. Now, a question. Will these babies look like B or will look like A? They will look like B. Why? Because I use the nucleus of B. I didn't use the nucleus of A. Why did I use the nucleus, the shell or the casing of B? It's because I wanted this to look like a zygote, because what is outside is an ovum. You understand? It's like drinking alcohol in a cup. I won't say that you're drinking alcohol. I will say that you're drinking tea. So because the outside is a casing of the nucleus, therefore this will look like a zygote. And because this one is diploid, 2N, they can ask you how many chromosomes are found in this. Because it's a somatic cell. Somatic cells are always diploid. You see? Somatic cells are always diploid. Someone is asking me to do the part of evolution. I'm coming there. I'm finalizing genetics here. And then I'm coming to evolution. So because this one is what? Is a somatic cell. Therefore, automatically, this is going to be a diploid. And then, because it's brought in the a casing of the ovum, so it's going to look like a what? A zygote. And now I split them in two small pieces, and then I insert them in different shape, and each shape will produce identical what? Identical shape. Yes, I think I'm done there. Sorry. I'm done there. All right. Now, lastly on genetics is this question of the dihybrid cross. Identify what is Z. You just have to know. Cross this, and then you write what is Z. What is the genotype of the parents? Now, how do I identify the genotypes of the parent? Genotype of the parent. Look at the gametes. You'll find the genotypes from there. Start with F. For example, you write the F. If it is repeated, leave it. Is there any other small f? Yes, the small f is there. Then go to capital H. Yes. Is there any other small h? Yes, is there. Done. Look at them. If there was no, if all of them were capital H, ne? All of them were capital H, ne? For example, capital H, even here capital H. I would have said, do I have F? Yes. Do I have small f? Yes. Do I have capital H? Yes. Do I have small h? No. They are all capital H. Done. That's how you can identify. Simple. No stress. They're saying that number of the genotype that could result in offspring that is short finger, and short is dominant, ne? And then this one is recessive. Because it's recessive, I need to have with the small h. As long as I have capital H, is 1, is 2, is 1, is 2, yours, and then yeah, is 3. So there are 3. And then allele that for continuous hairline, allele, not genotype. So it's gonna be small h. The genotype of the child that is heterozygous recessive for both alleles. So is this one, which is small f. Yes. So this dihybrid cross, simple. You should not go to the exam when you don't know anything. Welcome to evolution. Now, evolution. Okay, evolution. Let's look at evolution by natural selection. This one does not have too many questions, which I'm gonna speak about, but it has many questions in human evolution. However, these days we are setting evolution by natural selection than human evolution. I think Mutasi, now we are on you now. Ne? Share the video so that now people can join for evolution. Ne? Because they find a lot of challenges with the evolution. Evolution, what is a hypothesis? What is a theory? You have to go when you know the difference. Ne? Yes. Hypothesis basically is a tentative statement which is subjected to be tested. It can be proven correct or wrong. And then a theory, basically what you need to look at is a statement which has been proven by facts. You understand? Situation which is being proven by facts. So it means that it must have evidences to support it. Then it becomes a what? It becomes a theory. Now, before I go there, I think this one is a little bit clear. The 6-mark questions or 7-mark questions, they are going to come from here. Number 1, theories of evolution. Of evolution. Number 2, that is Lamarckism or Darwinism. Number 2, speciation. Speciation. Number 3, present day evolution. Present day evolution. Now this difference between Lamarck and Darwin, yes, but present day evolution. You can't go to the paper without, because these are six marks. 6 marks each. Yeah, I told you, you go back, you come back and then you tell me what you found there. Ne? Yeah. All right, let's continue. Theories of evolution. Lamarck's theory, Darwin's theory. Lamarck, what you need to know is how to apply it, but I'm just going to talk about it. Ne? I'm not going to take too much time on it. I'll spend the last few minutes on evolution, human evolution, but I'm gonna explain everything so that you don't lose anything. Lamarck's theory. State 2 laws of Lamarck's theory. It's law of use and disuse. That's number 1. Number 2 is law of inheritance of acquired characteristics. So it means that Lamarck has 2 laws. Number 1, law of use and disuse of a structure. Number 2, law of inheritance of acquired characteristics. Why is Lamarck's theory not accepted scientifically or by scientists in the world? Number 1, there is not enough evidence to support his theory. Number 2, organisms don't change because they want to change. Organisms change not because they want to change. You understand? Yes. For him, he says that if you want to be tall, you just become tall. If you want to be short, you just become short. No, organisms don't change because they want to change. Number 3, what is the difference? You have to know how to use how how Lamarck's theory is applied. For example, how did the big elephants with the big trunks came up with the big trunks? Using Lamarck's theory. So meaning that the more you use the structure, the more. At the beginning, all elephants had short trunks. Then you tell us that these elephants, they use their trunks frequently, maybe to defend themselves or to reach the trees, and then their trunks became bigger and longer. They passed their genes. Yes, they produced babies with big, long trunks. But if you are using natural selection, let me just use this as for natural selection. You have to start with, there is variation among the elephants. Those with long trunks and those with short trunks. Those with long trunks, they could survive. Yes? Why? Because they are able to defend themselves. They could reach the tall tree plants. While those with short trunks, they could not survive. They died in large numbers. You understand? This is called natural selection. This is called natural selection. I think. Now, those with big trunks, they pass their genes to the next generation. After many generations, all elephants had big trunks. Let me show you. Let me see if we can get any question here. But you can't go to the exam without knowing that. using sex, show gender in human, how it is. I see, I explained this. I explained this. You see, 6 marks. 6 marks. Okay, when I find it, I'll try to show you that example. Speciation. Now, students confuse speciation with natural selection. What is speciation? Speciation is the formation of new species. Please don't confuse those two. Look, speciation is the formation of new species. What's a species? It's a group of organisms that can interbreed, meaning that they have similar characteristics. Group of organisms with same characteristics that can interbreed to produce a fertile offspring. What is a population? The population is the group of organisms of the same species that can interbreed to produce a fertile offspring. That's it. Now, speciation is the formation of new species. When they talk about speciation, they will bring about, for example, one area, and then this area is divided into 2. You understand? And then after that, if you try to mix them, they will form different organisms. They will form different organisms. They will form different organisms. So now, let me explain to you. You have a population of A, so you have to tell us that there was one this mark. There was one population. Yes. Now the population is divided into 2 by a geographical barrier. You have to tell us the geographical barrier. Let me show you, because I saw a question here. Yup. So what is the geographical barrier here? The geographical barrier describes speciation of poto and lemurs. How did they become different species? There was one population of these organisms. There was one population of these animals. Did they talk about the ancestor? If they talked about the ancestor, then you have to speak about it. Scientists believe that poto and lemur share a common ancestor. They didn't mention the ancestor. So you have to tell us that, don't say that there was one population of poto and lemurs. They have not yet become poto and lemurs. No. So they have not yet become those new species. You have to start with what they started with. There was one population of these animals which shared a common ancestor. Yes. And then what happened? The population was divided into 2 by 2 subpopulation by a geographical barrier. What is the geographical barrier here? Ocean. So you don't need to tell us by geographical barrier. Tell us by the ocean. If it is a river, tell us by the river, unless the question is just open. And then now, you formed 2 subpopulations. That is east coast of Africa and Madagascar. And then what happens? There is no gene flow. There is no, this one can't come this side. Gene flow means that there is no sex between them. There is no gene flow between these populations, whereby these 2, they experience different environmental conditions. Are we together? So because they experience different environmental conditions, natural selection occurred differently. Oh, someone is asking me about punctuated equilibrium. I will come there. They formed, I'm going to explain it. Different environmental conditions, therefore natural selection occurred differently, whereby they became genotypically and phenotypically different. And if you remove the river, if you remove the ocean, they cannot mix to interbreed. Therefore, they formed 2 new species of poto and lemurs. They formed 2 new species of poto and lemurs. You can only talk about these 2 new species. Don't talk about 2 species before you talk about genotypically and phenotypically different. If you talk about it, you're gonna lose marks. You're gonna lose marks. Even if someone is marking, it will pain, but nothing we can do. You will lose the marks. So what do? Is you only talk about different species after talking about genotypically and phenotypically different. If you don't do that, and you talk about you form 2 different species, that the population was divided into two different species by the ocean, you are gone. You have lost the marks. The paper has killed you. You didn't kill it. I think you understand what I'm saying. Yes. So now, I repeat. Let me repeat it for you. There was one population of a common ancestor. One population of common ancestor. The population was divided into two subpopulations of Eastern Coast Africa and Madagascar by what? By the ocean. There is no gene flow between the two subpopulations. You see, they are still populations, not yet new species. Remember, a population is a group of organisms of the same species. And then now, they experience different environmental conditions. Natural selection occurred independently or differently. And then now, they became genotypically and phenotypically different. If you remove the ocean and you let them mix, they cannot interbreed to produce a fertile offspring. Therefore, two new species of Poto and Lema have formed. They can bring when they are more than two or more than three. So in this case, you tell us three new species have formed. So don't stack on two new species. Don't stack on one new species. So two new species. So I'm coming for punctuated equilibrium. Charlotte, yes, you are saying hi. Are you still here? All right. Now, let's go to punctuated equilibrium before I go to present day evolution. Oh, let me just finish the present day evolution and then I'll talk about the punctuated equilibrium. If I forget, you remind me. Present day evolution, it deals with the bacteria, virus, black peppery moth. We are talking about present day evolution which is taking place, which we see. You understand? DDT on mosquitoes. Yes. So, you know, if they are bacteria, it means that you're going to use antibiotics. Use the same principle of natural selection. Please don't confuse natural selection with the speciation. So in this case, you will say that there was one population of bacteria. These bacteria are separated by the variation which is there. So now when you're explaining, you say that there was one population, sorry. You say that there was variation among these bacteria. Those which are resistant to antibiotics and those which are not resistant to antibiotics. Then now tell us that the bacteria which are resistant to antibiotics, they survive on the application of the antibiotics. While those which are not resistant to antibiotics, they die. Those which are this is called natural selection. Those which are resistant, they pass their genes to the next generation. So the next generation will have more bacteria which are resistant. And after many generations, the antibiotics will become ineffective to bacteria. Let me repeat. There is variation among the bacteria. Those you can use for mosquitoes about DDT. You can use for virus about the antiviral or ARVs. There was one. There is variation among the bacteria. Those which are resistant, you have to tell us the variation. If you don't tell us the variation, you lose the mark. There was variation among the bacteria. Those which are resistant and those which are not resistant. Those which are resistant and those which are not resistant to antibiotics. Those which are resistant to antibiotics, they survive on the application of the antibiotics. While those which are not resistant, they die in large number on application of antibiotics. Those which are resistant, they pass their genes to the next generation. After many generations, the bacteria are going to become ineffective to antibiotics. Simple. There is no way how you're going to skip Oh, they're gonna bring a lot of things which you don't know here. Never. What is the difference between artificial selection and natural selection? And what is the difference between Darwinism and Lamarckism? Difference between artificial selection and natural selection. You know that artificial selection, what is basically on artificial selection is what is a selective force? First ask yourself, what is a selective force? Selective force in artificial selection is human, while in natural selection, the selective force is nature, is the selective force. You understand? Yes. And then Lamarckism and Darwinism, the major difference is Lamarckism, organisms change because they want to change. But in Darwinism, organisms change due to the environment. Yes. Then number two, in Lamarckism, individual in the population change. Like if me, I want to be big, I become big. If I want to be small, I become small. Not the whole population. But in Darwinism, only the whole population change. Remember, when Corona came, if you want or not, they say that you have to put on a mask. Natural selection. You don't put on a mask, you die. Whether you want or not, you have to do so. You understand? Therefore, that is natural selection. Is it clear? So, in Lamarckism, individual in a population change. While the Darwinism, only the whole population will change. So you can also talk about other differences between the two. Let me talk about punctuated equilibrium. And then after that, I go to human evolution. Someone is asking me, how do I draw a pie chart? How do I draw a pie chart? I'll go to the last question. That is the last question, question 3. The pie chart, how do you draw this at the end of human evolution? So that just I show you how do I identify independent variable, dependent variable. How do I get it from the question? How do I know the control variable? How do I know the conditions which are supposed to be put in place before you start? All right. Punctuated equilibrium. It is very simple. Gradualism versus punctuated equilibrium is gradualism versus punctuated equilibrium is saying like this. You have two scenario. Punctuate gradual. When you say gradual, it means that it's slow. The change is slow. While punctuated equilibrium, the change is fast. You have periods of no change and periods of change. The change is rapid. Periods of no change, periods of change, periods of no change, periods of change. This is punctuated. Punctuated. What this is gradualism. Here you require a lot of time. A lot of time. Here it occurs in short periods. So punctuated equilibrium, you have periods of no change, period of change, period of change, period of change. And in most cases, the punctuated equilibrium is caused as a result of mutation. While this one is changed, is caused as a result of natural selection. Yes. Natural selection here, yes. While here, mutation. Why? Because you just wake up in the morning and then you have something different which you didn't have before. Mutation maybe occurred. So that is punctuated equilibrium, or the other side is gradualism. This marks the beginning of the end of evolution by natural selection. Let's kill the human evolution now. Human evolution. Human evolution, guys, is the most important part where students sometimes lose marks is a phylogenetic tree. I don't know why, but we'll talk about it. Someone is asking, do you do calculation for pie chart? Yes, you do the calculation for pie chart before anything else, because you can't do percentage in a pie chart because the circle is not in percentage. All right, let me just see if I can find here a question. The first question in human evolution can be, to which family do humans belong? To which family do humans belong? They belong to family Hominidae. Hominidae. Differences between, let me tell you what are the short questions, guys, in human evolution. The short questions which you're gonna find, and then you have to give the answers. 6 marks. Number 1, it is out of Africa hypothesis. That's number 1. Number 2 is, so here you have to tell us the evidences, evidences of that is fossil. You have to tell us the mitochondrion, DNA, sometimes you call it genetics. Then you have to tell us cultural evidence, and then they can also talk about the differences between humans and apes. Yes, and then also they can talk about similarities between apes and humans. There is no 6 question mark they can bring here. Nothing. I repeat, nothing. Unless it's gonna come from heaven. Okay, now let me explain. There are some things which we changed a bit, and you have to know. That's why if you check my book of distinction material, you'll find out that it's always updated each and every year. It's not static. It's always a version for that year. There will be a version for 2023. All right. Now let's continue. They're saying that, now look at this. Position of foramen magnum. They're saying, which diagram represents the skull? There is a difference between a skull and a cranium. A skull is everything which is encloses the head. That's a skull. But a cranium is only the part which encloses the brain. Only the part which encloses the brain is what you call cranium. But a skull is everything which encloses the what? The brain. I think I'm clear there. Number 2. Number 2, they're saying that, which diagram represents the skull of a bipedal? Now we have 3 factors which